Ladies and gentlemen, may I have your attention? We would like to initiate the webinar in a few moments. The Honorable Dr. Iman Hidayat, Chairman of Research Organization for Life Sciences, BRIN. Honorable Dr. Rati Asmana Ningrum, Head of Research Center for Biotechnology, BRIN. Professor Dr. Durk J.O., Professor Zihua Jiang, Mr. Zahir Ali, PhD, distinguished speakers and moderators, and all distinguished guests and participants. Good morning. Welcome to the second day of the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering, or ISIBIO 2021, Global Platform on Biodiversity and Biotechnology. Before we begin today's events, let me read the conference guide.
Recording in progress.
For presenter and participant are expected to join at least 30 minutes before the conference started, and all participants are expected to join all sessions in this conference. And please use the virtual background provided by the committee and rename your Zoom username according to the following formats for presenters: code of abstract or paper _name _institution. And for non-presenters, you could use the format P_name _institution. And please to make sure, mute the microphone, especially during the presentation of keynote speakers. And in the parallel session, presenter should activate the camera. And questions from all participants are delivered during the raise hand, Q&A, or chat feature in Zoom, and use the format name, institution, to name of the speaker, and questions. And every session will be led by a moderator. All participants should fill the attendees list and abide all the rules listed on the screen. Ladies and gentlemen, before we begin the plenary session, there would be principle and mechanism of biotechnology product session 1. Now, we will hear presentation from Mrs. Dana Chow from Science Work, with the topic Accelerating Breakthroughs in Molecular Breeding with Innovative Genomic Technologies. So from Mrs. Dana Chow, the next 30 minutes is yours. Please proceed. Mrs. Dana Chow, are you here yet?
Yes. First, we will send a video of our distributor in Indonesia.
Introducing Bio-Rad's CFX Opus real-time PCR systems. Modern connectivity. With more flexibility than ever, CFX Opus can operate in a completely wireless environment. In addition to Ethernet connectivity and direct data transfer via USB for standalone operation, connect with Wi-Fi for wireless access and data retrieval. Program data and notifications to be sent directly to your email. The new network drive allows you to save and share data locally with multiple users. CFX Opus systems can also link to Biorad's BR.io cloud platform. Remote setup, instrument monitoring, and data management and analysis capabilities easily integrate with CFX Opus systems, minimizing time needed at the instrument. For more advanced data analysis, Biorad's powerful CFX Maestro software works seamlessly with CFX Opus systems. A simple visual interface lets you quickly create biological groups, technical replicates, and set up standard curves. Generate publication-worthy graphics and figures in real time. Biorad re-envisioned their CFX Touch Systems block to produce its best thermal uniformity and accuracy while retaining compatibility with consumables. Uniform well plate temperatures ensure consistent data from well to well. At each scan position, the robust optic shuttle is centered above each well. The optical system automatically collects all well data so that information can be entered or edited when convenient. CFX Opus real-time PCR systems. Powerful, modern, uniform.
The proven unmatched sensitivity you've come to expect from Biorad's family of digital PCR products has a new addition, the QX1. Taking its place in a familiar workflow, the QX1 performs droplet generation, thermal cycling and droplet reading in a single instrument. The search for fully integrated and automated digital PCR is finally over. With 4-color analysis and a new multiplexing supermix, the QX1 is one of the most advanced digital PCR systems on the market. Operating with existing supermixes, assays and kits, the QX1 is designed for you. And just like our other systems, you can be confident in its precision and accuracy. Load up to 5 plates. Oil bottles are loaded and consumption is monitored by the system. Experimental setup and data analysis can all be done using the onboard touchscreen. A simple 5-minute setup to configure the plate is all it takes, and the QX1 takes it from there. Once a run has started, the plate handler moves a single plate from the inbox into the droplet generator section. There, the plates are prepared for droplet generation. The droplets are created through the combination of sample and oil to create an emulsion of approximately 20,000 nanoliter sized droplets. The plate then undergoes thermal cycling, in which 20,000 individual PCR reactions occur in each sample well to amplify the DNA. The plate is then moved to the droplet reader module. Here, the foil seal is pierced by the droplet reader, and droplets are taken up and read by the QX1 with improved optics, fluidics, and motion control. The QX1 features complete onboard software support. Data for 4-color detection and analysis can be viewed for each well on the newly designed 3-dimensional amplitude chart. 2D dimensional amplitudes can now be visualized in a 96-well plate format. And concentration calls can now be made automatically by the QX1 software.
The QX1 makes the workflow so simple. It's the future of automation.
Extremely easy operation and data analysis. Data looks great.
Multiplexing using 4 colors on the QX1 enabled us to gather more data than we could before.
Brought to you by Biorad, the world's leader in digital PCR. From preparing reactions to reading droplets, the QX1 is the all-in-one integrated system that will change how you do digital PCR.
The NGC chromatography platform is truly customizable and grows with your purification needs. Collect precious samples into bottles and tubes and easily access them during a run on the high-capacity NGC fraction collector. Improve reproducibility and efficiency through automation. Choose a method template in ChromeLab software. Automate optimization or assess multiple conditions in one method with multivariable scouting. Set up a run, push a button, and walk away for routine single or multi-column purifications. The pharma-ready NGC system has tools that enable compliance. The NGC chromatography system from Biorad, the one platform that takes you anywhere.
Good morning, ladies and gentlemen. Please allow me to introduce Ms. Dana Chow from our principal LGC Biosearch Technologies, who will present about accelerating breakthroughs in molecular breeding with innovative genomic technologies. Ms. Dana Chow, time is yours. Dana, sorry, we cannot hear your voice. Hello, Dana. No, we cannot hear your voice.
Can you hear my voice now? Sorry about that.
Yes.
Okay, thank you.
Please
All right.
continue.
Yeah, sure. Okay, Tika, can you see my screen? Okay, the presentation slide. Yep, all right. Okay, very good morning to all of you. Thank you so much for inviting me as a speaker for this event. My name is Dana Chow, and I'm the Field Application Manager based here in Malaysia. And I'm with LGC Biosearch, hoping to present my presentation to you. My topic is accelerating breakthrough in molecular breeding with innovative genomic technologies. Okay. Right. So as an organization, we strive to deliver science for a safer world, which is our group mission statement. In agrigenomics, we deliver it through our enabling our customers to overcome the macro challenges facing the world, such as increased population to feed, reduction in available farming land. In order to rise to this challenge, it is critical to apply genomics to uncover novel traits and then incorporate these traits into the desired germplasm. The more accurate selection is made, the faster genetic improvement will be delivered. But this requires data generation, which also require resources. So the critical question is how to generate the right data needed to make the most accurate selection with the least amount of resources. And answering this question will maximize effectiveness and accelerate agricultural science, enabling you and me to meet the challenges we face. Through the collaboration, our technologies help improve breeding cycle, allowing customers to take their discovery to make the market faster. Our integrated genomics tool enable you to achieve more with your breeding programs and accelerate your mission critical science. So LGC origin can be traced back to 1842 when the Laboratory of Board of Excise was founded in the City of London to regulate the adulteration of tobacco, which was prohibited under the Pure Tobacco Act. But over the years, through new product and development, partnerships and acquisition, our portfolio has now expanded to deliver also critical genomics tools such as extraction, purification, PCR, qPCR, and GS technologies applied to plant and animal breeding. Okay, so coming to the simplistic view of a breeding workflow. Okay, different phases and different analytical techniques can be used and are routinely used, as you can see here on this slide. Typically, starting on the left with array, microarray, whole genome sequencing that has a higher marker and low sample number, and moving through the targeted NGS to PCR that has high sample and low marker. So each technology here has its own trade-off, okay, and also such as its own trade-off, such as cost per sample, okay, data content, marker number, and time to result. These are critical components most of the researcher or developer, breeding developer routinely look at, okay? So example here, whole genome sequencing could get all the information you require, for example, back crossing, okay, right here, all right? So, but it is more expensive, a lot more expensive actually, compared to PCR and slow to get to the data turnaround. Okay. Right, so many times we at Biosearch Technology here, we try to push the operating window wider to allow a technique that do more but still remain cost effective, accurate and fast. As we can see here, we have our TGBS, targeted GBS or multiplex PCR solution that is pushing the either way, okay? A lot of the ways that we do here is to partnership with our customer like you to develop and test this new technology. All right. In comparison of the technologies for the same application, one technology will win over another one, at least for one of this dimension: cost, speed, flexibility, and quality. Classically, when developing a new product, you will look at the 3 key factors, which is this 4 dimension. Normally, there is a trade-off. You can only choose not all, but some of them. You can be cost effective, fast speed, and quality, but you don't have flexibility. So genotyping lab requires high quality data to make those breeding decisions quickly and ideally at low cost. And at many times, this proved to be very challenging. It may require compromises and add in the flexibility requirement, such as marker panel change, and this can further complicate the whole workflow. Okay, so when it comes to PCR, example here, PCR in agrigenomics, so usually the application that researcher would use or developer would use is endpoint PCR genotyping, and also qPCR. So for endpoint PCR genotyping, application that is usually used here is marker-assisted selection and marker-assisted backcrossing, and it would require the technology to be fast, flexible, and economical. All right, so for qPCR, something like adventitious, present, GMO, trait integration, copy number variation, and this is detecting if traits of event in interest present or quantifying level of presence. So some testing is actually required by regulation, other necessary as part of the breeding program. And for this, it requires to be sensitive and specific with higher quality at affordable price. So when it comes to endpoint genotyping technology, we at LGC Bioresearch Technology here, we offer CAPS genotyping. In chemistry, as also as listed here, genotyping services. Okay, for us here, we have 3 components. We have CAPS assay mix. Okay, so we have a forward and a reverse primer. So the forward is the allele specific that has a universal tail end. And our proprietary technology with this universal tail end is combined with the master mix that has the frag quencher, frag cassette in quench here. Okay, so then we also have a DNA template right down here at C. Okay, so this template here is provided by you as our customer. So combining the assay mix and the proprietary master mix, CAPS assay, so with the DNA template. So it goes through 3 rounds of PCR, thermal cycling. So total of it is actually 26 cycle. Okay, and we generate a signal generation that can be read through endpoint. Okay, so using the CAPS genotyping chemistry here, okay, so one of the case study that I hope to share with you here, okay, so rice blast is a serious plant disease worldwide and it causes losses up to 100%. It can be treated with chemical. Out of 100 race-specific R gene node and 20 have been cloned. Okay, another disease is the bacterial leaf blight, which can reduce yield by 5 to 85%. This accounts for 40% loss in the worldwide annually. This diseases is not able to be treated by chemical. Out of the 40 race-specific R gene node, 6 gene has been cloned. Okay, so these clone-resistant varieties are therefore a major goal for rice breeder to counter blast and BLB using single or multiple pyramidal genes. Okay, so marker-assisted backcrossing has been used for the past 20 years and varieties incorporating resistant gene has been released in all these countries. So this is done on our CAPS genotyping chemistry here. Another example here that I hope to share with you is May streak virus. Okay, a single loci that is found to be tightly linked to this particular disease resistant, and it only needed 3 capsid marker to identify the introgression into elite variety. These samples were screened with within a 1536 plate format using our SNP line high throughput genotyping platform. Okay, we will show you the photo later. All right, and over 50 plant sampling kits, our BioArc plant sampling kits will also send out to gather 5,000 sample material, and we extracted all this sample with our Octopure DNA extraction high throughput platform and genotype them 5,000 sample all within 4 weeks. That's the capability of high throughput. So the implication of this is the fast turnaround means that there is we only need to sample the resistant variety, taking it forward. So time and money saved. All right, so our customer can approach us running CAPS genotyping chemistry, whether in their own lab, okay, or send it to our largest genotyping service in the world in UK here at Hoddesdon. Okay, this is our genotyping service lab. So we routinely every day doing 1 million genotyping data point. Okay. So on top of that, if you run PCR, CAPS chemistry in your own lab over the years and you wish to take it to the next level into high throughput. So we also work routinely with our customer providing the instruments, DNA extraction. So, and using with our chemistry. CAPS is not the only chemistry that we provide for genotyping. We have the BHQ and BHQ plus probe here and also the master mix. Of course, we have here the PCR workflow technologies products that is for downstream and upstream as well. So we will go into the leaf collection kit in a moment, but we have other type of extraction chemistry. So we are all fully integrated here. So we have the quick extract, SpeedX, the AutoPure, and also we have the PCR reagents, and also NGS reagent as well. But of course, we are talking about PCR workflow here. So for high throughput instruments, we have the IntelliCube, Nexar, Snipline, Array Tape. All right. So coming to down here, PCR still dominates lower marker number workflow here. Okay, when it comes to routine genotyping. So main focus here is speed and cost, of course, some flexibility here as well. And for NGS, it is unable to achieve this same price point per sample. You know, NGS is talking about having a certain coverage on especially like large genome size, so those can be quite costly. All right, but however, the market here, or in general sense, people are actually looking into higher density genotyping. Okay, so genomic selection is the thing that people are gradually moving on. Okay, so what technology does people use for genomic selection here? Okay, so genomic selection is a critical application to shorten the time to market. Okay, what does it mean by that? So if you know about breeding technologies here, so the people would look at genetic gains, okay, for marker-assisted selection as an example. So for marker-assisted selection, the genetic gain from it is actually like 0.4 to 0.6% per year. And to be able to address the food need in the future and the consumption need of the world in the future, statistics shows that it needs to be actually 4 to 6. So from 0.4 to 0.6 to 4 and 6 per year is 10 times. So how can we accelerate and meet this demand here? Okay, so we need to market and develop new breeds in a faster way. So genetic selection is actually showing potential and chance to accelerate this over time. Okay, so genomic selection accelerate breeding and provide potential solution as part of approach to solving global agricultural sustainability challenges. But genomic selection requires fast, flexible, cost-effective screening of thousands of markers. So this is the technology challenge. Okay, so when it comes to GS, for us here, we actually take the targeted genotyping by sequencing workflow, PGBS, which is an NGS technology. Okay, next generation sequence technology. So 6NIP here is TGBS, optimized for screening thousands of markers cost effectively in any sample number. Okay, so it is not GBS, not genotyping by sequencing. It is targeted. You need to know your reference genome and to design this panel. Okay, so it is optimized for several hundred to 10,000 markers, but scale up to 100,000 per sample. So it's cost effective because we designed the panel and in NGS platform environment. So it is flexible. So you design for this panel this time, and next round you can remove and add new panel, new marker into the panel, and it is lower setup cost. There is no setup cost for 6NIP here. So we design the panel for you for free. So we just order it as a kit. Okay, so it allows de novo variant detection in the target SNP region. So there is like a 70 base pair read line here. So then you can detect additional SNP at the upstream and the downstream here. Okay, so comparing marker-assisted selection to GS here, important point here is that like, although we're still looking at the same sample number, 100 to 10,000, so then we have here a low SNP number for 1 to 10,000 SNP, okay? And then we have here up to 3 to 50,000 here, okay? So GS is much quicker in that way. Okay, so we have it in the service process. okay, and then the data result. So we also have other product here, genotyping by sequencing by key gene, okay, and on top of that, we have our BioArc sample collection kit that helps you to collect sample quicker in the market, in the field there, all right, and with a BioArc kit, then we can have a better genotyping quality because the sample are properly dry. Okay, so of course we have our DNA extraction and purification platform. Okay, so as you can see for LGC Biosearch Technology, we have focused on all this area of the breeding technologies. So we have here from WGS, whole genome sequencing, discovery, upstream analysis, down to a targeted GBS technology, and then move on to PCR routine QC technologies. We have our solutions for you. Okay, so there's a couple of ways of how technology development can drive cost, flexibility, quality, and time factors required to accelerate breeding technology to solve the global challenges facing the industry. So thanks to our customer in collaboration with their support and inputs, LGC Science for a Safer World. Thank you so much.
Thank you, Ms. Dana, for your presentation. And for the ladies and gentlemen, if you have any queries about our technologies, please don't hesitate to contact us, PT Sciencework. Thank you.
Thank you, Mrs. Maria and Mrs. Dana Chow for the presentation. For the next, we will hear a presentation from Mr. Revata Utama from Nusantics with the topic Indonesia as a Global Biodiversity and Biotechnology. Mr. Revata Utama, the next 30 minutes is yours. You may proceed.
All right, thanks so much, Ms. Wiwit. Good morning, ladies and gentlemen. Let me just share a screen and introduce you guys to Nusantics. All right, so today's topic will be about introducing Nusantics and also Indonesia as the global biodiversity and biotechnology hub. Is it a far-fetched or reality in the future? I think first of all, we'll just start with all these topics for this morning. And let me start with introducing who we are. So Nusantics is a holobiome startup. What does holobiome mean? I think we made a lot of mistakes looking at human as a single species, and now with the advance of genetic sequencing, we realize that we are not only a human, but also the microbiomes and the microbes that live on us, be it on skin, be it in the gut, or be it in your lung respiratory organs. So genetics is part of the things that you can't really change because it's given by your predecessors, your parents, your grandparents, and it relates to your ancestry and also, the gender that you, the sex that you're given with, be it a male or female. While microbiome is something that is dynamics, where it's changed when and in where you were born, how you were born, be it through cesarean or even normal, birth, and then what you eat and drink also affects on your gut microbiome, the environment that you're living in, the air you breathe in, the kind of exercise that you do, and also the stress level. And when we talk about microbiome, we also need to know that they consist of the bacteria, the fungal, the archaea, and also the virus, which is very closely related to the pandemic that we are having right now. So pandemic accelerates biotechnology. We believe that the next technology is biology, not only digital, but it's also biology. And Indonesia should not waste the momentum because one of the things that we can do, we were established in 2020. And because of the pandemic, we helped the government to create the first local PCR test kit for COVID-19. And we released second generation in September 2020, where it's multiplex and increases efficiency by 3 times. And also in August 2020, we launched our BioScan service for skin microbiome. And recently, in March 2021, we released the air viral detection to help reopen the economy, where we tried to detect the COVID-19 concentration in the air and also give out the risk index for the infection in classrooms and also office. And as recent as June 2021, we released the Gargle PCR method to increase the testing capability across Indonesia, not just in Java, but also in the rest of Indonesia. And our product has been used about 5 million test kits by Indonesian government and also the Indonesian market. We also have about 20% of our team members from universities, overseas universities. We consist of highly capable scientific team. These are the universities where we came from, and also we have the Professor Carl Whitwer here as our scientific board advisor, where he was we can call him basically the grandfather of qPCR and fast PCR systems. Let's talk about biodiversity because it's an ISIBIO event, and we talk about biotechnology and biodiversity. So why does it matter? Biodiversity is something that we take for granted for a long time. We live in the ecosystem as humans. We always think of ourselves first and foremost, but we don't realize that ecosystem actually give us back all those things by giving us foods, provisioning function, fuels, medicine, and then it also we are actually part of the ecosystem where we help, pollinating, and also we have to give back to the nature by creating the cycle back where we don't just use them and also but give them the needs for the ecosystem to keep going. And the ecosystem itself also has regulating function, and also the most important part is actually cultural function for us, where we use cosmetics, we do rituals, we actually develop all these cultures because of the shapes of the environment that we live in. But we failed to value the biodiversity because as of 2020, we lose about 70% of the vertebrate species, 40% of the insect species, and also 50% of the plants. What we don't realize is that 50% of the world GDP is actually generated from nature, and 75% of pandemic is caused by emerging infectious diseases such as COVID-19 and also flu. And when we talk about a system like biodiversity, we have to really correlate it with second law of thermodynamics, where some things are irreversible. Once the entropy increases and the system actually collapses, there is no more life on Earth. That's why we have people like Elon Musk try to bring us to Mars. But what we don't realize is biodiversity is the key to ensure that everything is at peace, we live at peace, because all this vicious cycle of destroying nature is actually going to increase a lot of zoonotic diseases, extreme weather, and also resources scarcity. With that, it will lead to global conflict. But when we talk about biodiversity, we talk so much about the carbon metrics, where it's just about a cycle of where the carbon is absorbed and where the carbon is being produced. But what we don't realize is actually there's a lot of things that changes from the micro environment itself. So that's why it's time for Indonesia, where we have the world's biggest land and marine biodiversity. So let's take a look at the microbiome changes itself. For example, coral bleaching itself. When we talk about coral bleaching, before we see the visible changes in the color of the coral itself, there's actually a lot of changes in the microbiome diversity on the coral itself. And we realize that microbiome diversity itself reflects microbiodiversity loss. There's a paper in Nature in 2020 that talks about the microbiome and the many changes before this coral bleaching is actually observed. And what we do here, we are trying to raise biodiversity awareness through whole biome concept, where we realize in human species, we have 30 trillion human cells, but we also have 39 trillion microbial cells, which consists of like 57% of our body. And they actually correlates to a lot of diseases out there, especially non-communicable diseases. Like, for example, in our skin, there's acne, eczema, psoriasis. In the oral microbiome, we have the dental caries, periodontitis. And in the lung microbiome, is linked to various diseases where the imbalance of microbiome will cause a lot of lung diseases like COVID-19, pneumonia, asthma. And gut microbiome, as you know, this is the most well-researched topic, and we have type 2 diabetes and stunting correlated to microbiome dysbiosis in the gut. So our work is centered around the interaction between humans and the microbiome itself. The key is holistic and accurate diagnostics, because we are not just looking at the human itself. We are, as I mentioned, microbiome is also coming from the environment that we live in. So we look at the air, soil, and marine. That's why we also have the air scan for COVID-19. And here are some of the services that we have built for the past 2 years. We have BioScan, which is the first skin microbiome check in Indonesia. We look at your bacterial fungal ratio and also the microbiome composition to see the right skincare for you. We also do a lot of research on ecobiome, especially the latest one will be AirScan, where we work together with Commandicwood to scan the schools for COVID-19 presence and also give out the risk index. We did it in Cinema 21 and also Masjid Istiqlal to check whether there's a COVID presence and to protect the people who are doing the prayers there. We recently also worked with Pelataran, where we basically research on their air microbiome and also the environmental microbiome in the Pelataran area. So we believe by collecting all those data, we will be able to understand the microbiome composition and its effects on human environment and also the relation to the disease that we observe nowadays. So we would like to believe that these microbiome changes can be an indicator for the health of our planet, not just in humans, but also in the environment. So let's talk about Indonesian bio startup then. As we mentioned, the next technology is biology. So we have so much potentials, but lacking the execution and also the political will to do it. As we can see here, when we do go into the biotechnology, there's a lot of things that we can develop from there, especially from what we listened to just now. There's going to be a lot of improvement in agricultural side. We can build new therapies and cures by just looking into more of these microbiomes. We might be able to develop new food source, new food plant source by utilizing all these microbes and new materials and clean fuels. But the challenges are, there are a lot of challenges. As we can see in the market right now, there are two types of biotech startup. The first one is the pioneer and research oriented, where we build all this, we do go through all this workflow where we validate the tech and then we build a database and we find biomarkers and we develop device for diagnostics, for example. And then from understanding the problem itself, we'll be able to give a precision solution. So there's a lot of challenges that comes with this, especially with building databases where we have to, use, we have to build laboratories to collect samples and then process them. There's also the regulation where, all this lands is actually owned by the government and we need the ethical clearance to do all the research. But then there's also the type 2 where it's market driven. You can just buy things from outside, especially from China, and then you do the diagnostic service, for example, like what we have nowadays with the COVID-19 testing. Where do we stand for? Nusantics do the hard work, where we, for example, in the microbiome service, we do the validation ourselves. We build the sample collection method up to the detection ourselves, and we build the database right now where we can identify the biomarkers and also design the kit in the form of bioscan. We also do the production ourselves and we do the diagnostic service ourselves. In terms of COVID-19 pandemic, we don't need to build the database because we have the GIS-8 to do it. What we do is we identify the biomarkers so that our kit is safe from mutation. We can still detect up to the new mutation, the latest mutation. That is reflected in the Biocov-19 up to the Biocov 3.0 that is going to be released soon, as soon as December. And the diagnostic service, we are going to do it as soon as possible because we believe in providing affordable service and also accurate service for COVID-19 testing. But we still need help from the government, especially because Indonesia, I think, as we mentioned, it's a big land and also we have a huge biodiversity. So I don't think we should be looked at as market only, but we also need to be an innovation driver. So to do that, we definitely need to collaborate with the government institutes like BRAIN universities and diasporas, and also this other downstream companies like the BUMNs. So I think in conclusion, is it a far-fetched or realistic future? I think let's quote some of the statement that were made by Steve Jobs and Elon Musk. Steve Jobs mentioned you can't connect the dots looking forward, so you can only connect them looking backwards. So we have to trust the dots that the dots will somehow connect in the future. And Elon Musk also said, when something is important enough, you do it even if the odds are against you. So I'd like to believe that Nusantics will be able to achieve the Universal Biome Index for us to monitor the health of human and also the planet through biodiversity itself. So I think for a closing statement, these are our websites. We have nusantics.com as our main website, where you can take a look at our biome services and also personalized skincare that we have. We also launched the initial Indonesian microbiome initiatives recently, where we are looking to collect data from not just Indonesian people, but also the environment. And recently, we are involved with Commandicwood to basically reopen economy and reopen schools, where we do air scan and gargle PCR for schools. So I think that's it from us. I'll return it back to you. Thank you.
Thank you, Mr. Refata Utama, for the great speech. The following presentation, we will hear from Mr. Andreas Agustian from ELO Karsa with the topic Vaccine Development to Translational Research. So, Mr. Andreas Agustian, the next 30 minutes is yours. You may proceed.
Yeah, hello, good morning. Thank you, Ms. Wiwit, for the opportunity. I will try to share my screen. Okay, is my screen okay?
Yes, Mr. We can see it.
Okay, okay, let me start. Thank you for the ISIBIO committee or the chairman for the opportunity for PT ELO Karsa Utama to have the schedule for the presentation here. I would like to share some information regarding the vaccine development and translational research here. Okay, at the end of the presentation, I would like to invite all the participants to play some mini games at the end of the presentation here. I would like to introduce myself. My name is Andreas Agustian. I'm as the product manager from PT ELO Karsa Utama. And ELO Karsa Utama itself established in 1981. It's already almost 41 years now. Our head office in Jakarta and the regional office in Surabaya, Bandung, Bogor, and Yogyakarta. And our focus in the life science business, animal health, food safety, and also the medicals. Our vision is to become the leading distributor in life science and medical industry to the effective and synergy with the principles and achieving the best solution for our customers, and also improving our customer satisfaction by consistently providing the high quality innovative products and a better service experience. And as our motto or logo is the serving you better. This is our distribution network. We are have the head office in Jakarta at the Kabayan Rama Street, and also we have some representative office in Bogor, Bandung, Yogyakarta, Surabaya, and also in Semarang. Please welcome all the customers to contact our representative in the area, and also we have some warehouse in Jakarta, Serpong, Surabaya, and also in Bandung. This is our value client from the industry, biopharmaceutical, also the animal health industry, pharmacy, and also the government institution, university, and also clinical lab. And this is our brand partner. The product we represent right now is the instruments, from the general, imager, and all the essential instruments that you gonna be needs in everyday lab, every day, and also the reagents starting from the chemicals, media, from the bacterial, and also for the cell culture, and also the testing kits, especially for the COVID-19, and all these consumables starting from the plasticware, glassware, and all the special one. For the life science itself, we also providing our solution from genomics, proteomics, cell culture, and also the animal research. Now for the vaccine development itself, typically we start from the pathogen profiling. We can use the COVID-19 as the example here. We do a lot of NGS, and also we do the viral characterization using the viral culture, and also some the Western blot or imaging itself. And then we try to design a vaccine here, starting from the factor, or using what kind of platform we want to use. For instance, the viral vector or the mammalian cells and so on. And also after that, when we get the protein, and then we try to do the expression, then we do the in vitro testing here, using maybe the nucleic acid, real time, or the in situ hybridization, and also doing some ELISA technique or Western blot, and also the insal Western. Regarding the insal Western, I will explain more later. For the cells itself, we can do the normal one is the plaque reduction neutralization test, and also some of the imaging. For the in vivo testing, typically we do serology, like ELISA and so on, and also the animal challenge here to see the pathogenesis, and also the imaging to see whether the biodistribution or the target is targeted the correct organ, or can protect the organ that we want to save. For instance, lung here for the COVID-19. And also we can provide the scaling up technology, starting from the upstream and downstream, and also through the QC process. Okay, our one of our solution for the viral acquisition systems comes from the Agilence technologies. We providing the adenoviral vector here, and also we do have the other plasmid called sure vector here. So in the previous time, maybe some of the researchers doing the molecular cloning, like doing the ligation, but here with the sure vectors, it's like playing your Lego. What is your gene of interest, and what kind of selectable marker do you want to use? Is it ampicillin resistant gene, kanamycin, or chloramphenicol? You can choose what kind of gene do you want to put in your plasmid. So it's just playing your Lego, and it's very simple to make a circularized plasmid in a single reaction, only 20 minutes. All right, I will next for the insal Westerns. So in virology, there is some well-known technique that can be used, like plaque assay or PRNT, and also some of the Western blot or the ICC, immunocytochemistry. Now, typically in cell Western workflow like this, when we do the culture cells, and then we stimulate or inhibit, typically for the vaccine, we put the antibody neutralization here, then the viral can be neutralized by the antibody, and then it can be inhibit for infecting the cells. And then the cells we can fix and permeabilize, do the blocking solution here, detect with the primary antibody, and then we detect with the secondary antibody. Now, the secondary antibody here, we put the label here for the fluorescence, so it can be detected with the imager. Then the imager will excite the fluorophore, and then we can get the result. So the benefit of using this insal Western to study the virus is to have this high sensitive, due to the low sample autofluorescence. Also the high accuracy of the assay, due to the antiviral antibody specificity. Also works with the virus with or without CPE. Some problems in the CPE in some virus and the cells model, yeah, that cannot show the CPE. And also we able to do the multiplexing, yeah. So we can do the normalization there, and also we can use the 96 well pair or 380 well 84 well pairs to get more throughput samples, and we can remove the subjectivity from the analysis. Also, we provide the software to shorten the times for the analysis, yeah. And the other things, we can also do the on-cell Western. So what is the difference in these steps is we exclude the permeabilization steps. So we can do some detection on the cell surface protein. So we can detect on the receptor trafficking, for instance. Yeah, and then we can image. So in vitro application here for the virology, we can do the viral translation, we check on the viral replication, we can quantify the protein expressions, yeah, very easily, and then also the mechanism of actions and efficacy of the vaccines or the antiviral. Or we can go forward with the in vivo characterization with the delivery and the response. This is some examples or the parallel comparisons between the plaque forming units here for the plaque assay and the in-cell Western. And this graph showing the correlation is almost close, yeah, to each other. So instead of we counting the plaque number here, we can measure the fluorescence signals comes from the fluorophore or reflecting the amount of the proteins expressed protein from the viral, and then we can calculate very easily. Okay, this is also some examples for the non-CPE causing virus, yeah. So we can detect the viral protein, yeah, comparing with the other fluorescence technique. So here the correlation is okay, comparing with the conventional method. Now here also the some examples for the quantifying infectivity through viral replications doing the HTNV here, comparing with the beta-actin, and then we can overlay here for the normalization. We can see how the viral replicates per days. Yeah. Here also some examples for the assessing the vaccine effectiveness. Yeah, using these examples here, vaccination with blood boost and the boost vaccination, and how we can compare the neutralizing antibody titer here very easily using the in-cell Western technique. So how we can achieve that one? We can use the ODC CLX imaging system here. This is the instrument picture. It can do the near infrared fluorescence. Why only near infrared systems? Because on the visible, we can show here cells. Yeah, cells we culture, and then without any staining dyes, when we expose or we capture the image with the visible fluorescence, it can emit a light. So in the visible fluorescence, there is some autofluorescence from the cells itself. So when we want to detect some of the target proteins, gonna be very hard, yeah, because due to the autofluorescence from the cells itself. Yeah. So this is the other DLX applications from the quantitative Western blot. Yeah, protein gels, in-gel Western, in-cell Western. We just talk about viral titration or the EMSA gel chip assay. Tissue sections, maybe after we do the in vivo imaging, then we want to do the tissue section to check. We can do the scan on the ODC CLX and also the protein arrays. And the next is how to do the in vitro testing using the cells. Yeah, how to make your cells happy. Okay, I have the illustration here. The cells must live in the house. Yeah, we call it the culture vessel. Yeah, what type of house? How big the house? And also the media, what's the cells gonna eat? Yeah, what is the nutrition they need? The instrument is corresponding to the environments behind it or surrounding. Yeah, what is the CO2 concentration or the oxygen concentration and all the things. And after that, how do we analyze with the imaging here? Okay, so for the culture vessel here, we support for the thermal from NANG. Yeah, we have several type of the culture vessel. Also, if you want to upscale the production, yeah, somehow the for the in vivo testing, you gonna be required a lot of culture vessels, yeah, the bigger one. So we can provide that one. Also the media, starting from the media basic and also the serum and also the differentiation medium or the serum free media. Yeah, we also providing the cell line here from our principles. Yeah, for the instruments, we can provide the CO2 incubator here. Also the have the accessories for the cell locker, so don't need to worry about the contamination for each segment or compartment. Yeah, also we provide the BAC to handle your cells, and then the centrifuge to process and the autoclave to terminate. Then for the imaging, we provide the microscope from the Perkin-Elmer here for the live cell imaging, and all the confocal or the high content imaging we have. I want to talk more about the high content imaging here with regards to the what can be done with this instrument for the for your vaccine research. Yeah. So high content screening itself is the imaging and analysis from image to number to the knowledge. So basically it's image based screening, yeah, measuring the biological activity in cells. So in this picture, we want to see, we can see the mitotic cells here, and also almost separate cells and all the mature cells. Yeah. So we want to discover things using the confocal with the high content screening. And how we can do the experimental with the high content screening. This is the workflow. We can sit, yeah, we can try to grow the cells, and then we can do the treatment. And after the grow, we can do the staining, and then after the staining, we can image and then do the analysis. And after the analysis, we can do the data analysis. Why there is two things, yeah, here, the analysis and data analysis. When we do the image analysis itself, we can separate which cells we want to analyze and which fluorescence we want to detect. And for the data analysis, we want to see the graph and the histogram about the effectivity of what we have done in the experiments, like the treatment. We put the compound or the antibodies that inhibit the interaction to the cells. Yeah. And what high content analysis can bring you to do more samples, yeah, because we do it in, typically we do it in the 96 well plate, yeah, in the microplate. So there is more samples and can be statistically relevant, reduce the bias and achieve higher physiological relevance and gain deep insight into our models, yeah, biology models here. And all can be done with the single software. Harmony software can do the analysis and data analysis. For the high content imager with confocal here, we can see the difference, yeah, between the wide field and the confocal. Confocal can bring more details on the fluorescence and a bit dim, but it can be, also give a sharp and better resolution. So what kind of models we can do with the high content imager? We can do the HeLa cells for the cell line. We can do the primer cells, co-culture, or the iPSC, and also the spheroid analysis, the FRET for the protein interaction, and also the zebrafish tissue. And again, we need the image that contain information that we quantified. Yeah. And what is the other application? We can do the drug repurposing, development of new therapeutics, development of new vaccines with the high content imager from the viral, bacterial, and parasitic type of studies. And this is all the type of publication, yeah, from the viral disease, bacterial, parasitic, and also cancers. Now have a growing in stem cells, and 3D models also already done with the high content screening here. This is some examples from the virology, yeah, using the high content screening here. So it can be determined in this research, they determine the co-localization at the cellular level, yeah, and also how the receptor can inhibit the viral activity, and also with this instrument in the studies, we can see how host and viral interact each other, and also here for the COVID-19, how the cross-neutralization antibodies developed from the vaccination can inhibit all the circulating variants around the world. Yeah, this is the evaluating neutralization antibody, and also calculate the neutralization activity using the Columbus software from Perkin-Elmer. This is the current publication for high content screening using the for the COVID-19. And then for the in vivo research, we can support for the mice itself, yeah, from BioLasco here. We can provide the inbred mice, the outbred animals, and also the hybrid. So please contact us if you have any inquiries. And also we can provide the in vivo imaging here. So what is the benefit of using in vivo imaging? We can explore different aspect of the disease. Yeah, we can monitor the disease development, characterize the mechanisms of actions from the pathogens, and also assess the therapeutic efficacy here. This is some of the examples. So the bioluminescence done here to monitor real time, yeah, for the COVID-19 spread in vivo, and then with the neutralization antibody design here, they can do the proliferasis of therapy, and then the engagement here to reduce the viral load in the lungs, and then the monocyte also can be detected here, how the cellular immunity also responsible for the viral clearance. Here also some examples for the thermostable mRNA vaccine COVID-19. Yeah, hopefully it's gonna come to Indonesia soon, I think. Yeah, so this is the development of the encapsulated mRNA vaccine targeting the RBD, and then it can induce the neutralizing antibody and also the cellular immunity. And then how these mRNA vaccines also give the protections to the animals model, and the thermostable vaccine is also already in the phase 1 clinical studies. Yeah, this is some of these examples. Yeah, the storage days, and also the temperature to store. So it shows that it can be protected. So more than 12,000 publication in many applications for the IV system. And also we provide the 2D and 3D imaging. Yeah. So let me play here. Yeah. For the 2D, typically we can all detect the fluorescence or the bioluminescence from one side. Yeah. But the problem is how deep is these signals. But with the 3D tomography, we can detect how deep, because we do the 3D tomography to check where is the locations, and then also we can do localization and correlation to the mouse atlas on which organ this signal is detected. Okay, this is our system from Perkin-Elmer. So IFIS comes from the in vivo imaging system. Yeah, that's a brand from IFIS. So we have variant model here about what is capability of doing 2D, 3D, and also the X-ray. Beside the instruments, we also provide the reagents. So if all the attendees has the in vivo imaging system. It's on your lab. You can contact us and discuss further about the reagents we can do the research and which catalog that suitable for your research. Okay. For upscaling, we can provide from thermoscientific called Star Factory here. It's very simple workflow with the constant result because it's just upscaling like the apartment. Yeah. So this is the landed house and this is the apartment. Just increase the numbers of your flash here using the cell factory. No need to optimization, just do the scaling up with the cell factory. Very easy to upscale your culture and also easy to downscale. And the other things is for the suspension cells, for instance, you can using our bioreactor system from our BioSmart. Yeah, it can be optimized to your cells for the single-use technology. And also for the sample QC, we want to introduce the MFI, stands for the microflow imaging from protein sample. Typically for your research in the QC, at the end your finished product, you need to count the particles, and then because the size of the particle itself can change the immunogenicity. Yeah, so typically when you do the in vivo imaging, the in vivo research, we want to analyze or detect the serology, for instance, is gonna be changed when it go to clinical trial. So better you can start with the MFI to check the particles for your formulations, and also for checking your vaccine final finished product, or in the in the process. Yeah, in the downstream process, you also can do the the capillary electrophoresis, yeah, from Maurice, to separate the protein from the heterogeneity. Yeah, because some of the protein may be produced and have their own isoform. And also for the size itself, whether it's already degraded, we can do the capillary electrophoresis using the SDS to check the size, yeah, whether it's truncated protein or not. Okay, that's all for my presentations. Maybe is there any open questions? And we have some gifts for the participants who are asking questions, and also we have some gifts for the participants who are willing to fill our link. Yeah, maybe is there any questions? Miss Vivit, maybe we still have time for Elokarsa.
Yes, we still have time. Mr. Andres, if anyone would like to ask questions to Mr. Andres Agustian.
Yes, please, you can put on the chat if you have any questions. If not, then we can play with the game. We have several door prize.
Oh yes, for the questions, Mr. Andreas Agustian.
Okay, is there any questions?
May I ask?
Yes.
Oh yeah, Penny, please.
Okay, so I have a questions about the mice experiment that you did. I mean,
yes.
How did you induce COVID-19 to nut mice, if I'm not mistaken? How did you make a mice experiment for your this one for COVID-19?
This one?
I think it's more like to imaging. Yeah. Next, you can explain it this. I mean, for how did you make? Because you do it in mice, right?
Yeah. Okay. So in this research, I think, if I'm not mistaken, yeah. So this is the research from Zhang here. Then they using the induced mice with the H2 receptor. So I think it's from the Jackson Labs who do these modifications, genetic modification. So these mice already express the human H2 receptors. Then it can be susceptible for the COVID-19.
So I got it. So you already, so it's not your own research. I mean, you use it for from someone else research or something. Oh, I see.
And there is some of, I think there is one. There is one like company that providing the animal model for COVID-19 research. To do the challenge because the mice already induced with these human H2 receptors, so it can be susceptible. Then after vaccination, we can do the challenge whether the mice is protected or not, something like that.
Oh, I see. About this mice imaging. So you use 3 different temperatures. So what is the influence between different temperature with the mice metabolite?
So in this research, they do the bioluminescence for the what to say, the virus. Then the vaccine itself will be injected, and then will induce the neutralization antibody. Then when they inject with the virus, it will give the signals from the lungs, because the virus will target the mice lungs here. And then how they gonna be to be protected from the vaccination, using the thermostable vaccination. So basically this research more focused on the development on the uncapsulated mRNA vaccine. So they want to develop the because we know mRNA is very fragile, it's unstable, easily to fragmented, and how do we encapsulate the mRNA as the vaccine with the thermostable. So no need to have the low temperature storage, something like that. Just the
judging from this in vivo imaging, the fluorescence in liver is very high. Is it really effective to use this strategy? Because you target a lung, but a majority, the accumulation of the fluorescence is in the liver. Did they mention about this?
I think for this research, it only shows that the protections can confer the protection against the COVID. But for the organ itself, I need to check, Ms. Reni. Thanks for the confirmation.
Thank you very much.
Anything else from the chats? Okay, maybe Elokarsa, we play some games at the end. I will stop share, and then my colleague, Ms. Stephanie, will conduct with the door prize. We have already several people that already fill the form. Thank you very much for your participation. Then we can spin here, and we have two prize here. Okay, we will looking for 2. Okay, please spin. Who is the winner? We will contact you after this. Okay, Miss Yati. Then the next one. Okay, Elo Rifki Virginia. Okay, thank you very much. We will contact because already submit the email and the phone. Thank you.
Thank you, Mr. Andreas Agustian, for the great speech, and congratulations to the winner. And you can contact Mr. Andrian for further information. Ladies and gentlemen, we have finished principal mechanism of biotechnology product session 1. Now, going to our next agenda, we will hear keynote speech from our keynote speaker, Professor Duk Jo from Asian Federation on Biotechnology, or AFOB, with the topic Enhanced Production of Recombinant Human Interferon Beta through Signal Peptide Modification. Now, allow me to invite Professor Duk Jo. Good morning, Professor.
Good morning.
Good morning, Professor. Before we begin, allow me to read a brief CV of Professor Duk Jo, if you allow me, Professor.
Thank you.
Okay, so Professor Duk Jo earned his doctoral and master degree in Chemical Engineering Korea Advanced Institute of Science and Technology, and his bachelor degree in Chemical Engineering, Seoul National University, Korea. He is currently a professor in Sejong University, Department of Integrative Bioscience and Biotechnology. Professor Duk Jo research topic focus on bioreactor development for cell cultures, therapeutic protein production, including antibodies, cytokine, hormones, and many more. Cell culture process development, adult stem cell expansion, cell culture medium, including serum-free medium development, and gene therapy technology using virus and or non-viral delivery system. Now, without further ado, the time is yours, Professor.
Okay, thank you. Let me share the... Can you see?
Yes, we can see it, Professor.
Okay, thank you. Okay, thank you very much. I'd like to appreciate the organizing committee for giving me a great opportunity today to share our experience on the bioprocess and also cell line development. Okay, I'm Duk Jo from Sejong University. I work for Asian Federation of Biotechnology also. So I'd like to invite you, all of you, to Asian Federation of Biotechnology. Actually, membership is free, so you can join and enjoy your network with other people in Asian region. Okay. Today, my talk is enhance the production of recombinant human interferon beta through signal peptide modification. Actually, my background is engineering, and I used to have a strong interest in how to enhance the productivity from cell culture by bioreactor development or bioprocess development, media development, that kinds of thing I have a strong interest. But this time, today, I have a little bit different area. Recently, we have interest in this cell line development area, and we got some results. So I like to share this result with the participants in this symposium. Okay. Okay, what is the interferon beta? It is right now it's a biotherapeutics, and mostly produced by animal cells, and it can be used for treatment of multiple sclerosis. And still, it has a huge size of a market in the global. And then actually, since it is a kind of biologic product, so it needs to have high productivity to be competitive in the global market. So I have interest in productivity also in this case to get a high productivity for production of interferon beta from animal cell, but this time not the bioreactor system, not the serum-free media, something like that. This time focus on the cell line development, especially for vector engineering. But I'm not a specialist in the vector engineering, but we started with a small project to approach this area, and I like to share our experience with all of you. So as I told you, in order to produce a recombinant protein from animal cells, you need almost 3 steps. First one is cell line development that produce your product. And then after you get the cell line, you need to develop the cell culture process, especially for that cell line. You need some specialized bioreactor system, and you need to decide what kind of a culture mode you're gonna use, like a fed batch, perfusion, whatever. And also you need to develop the media to get a high productivity. Maybe it's gonna be serum-free media or chemical-defined media. And then after you finish the cell culture, you need to purify your product. So if you optimize and develop the purification process, you can get a good productivity also. So most of my research area is focused on here, and most of my published paper is focused on this area. But this time today, I like to talk about cell line development, especially expression vector development through signal peptide. Okay. What is signal peptide? I think some of you already know that. And here we have a target protein, our target protein, and the signal peptide is a little bit earlier than your target protein, and it has 3 reasons. This blue region is cleavage region. This sequence is you cannot change much because enzyme should cut this side. So if you change something, that enzyme cannot cleave this part by enzymatic reaction. And there is a hydrophobic region in the middle. There are hydrophobic amino acid to form the alpha helical structure. And there is an N region. This is positive charged. So this is very important part for especially the secretional protein. So I will show you one cartoon in textbook. So this is it. So this is a ribosome here, and the signal peptide is always translated first before your target protein. And then there is a signal recognition particle. They bounce together like this, and then they bind to a signal recognition particle receptor protein, and then your target protein can be translated and put into the ER rumen here, and then signal peptide can be removed and cleaved and removed, and finally you get the protein inside the rumen, inside the ER rumen, and then your protein can be followed by the post-translational modification, like cutting and then glycosylation or something like that. So the signal peptide synthesis is a translation is the first step important for secretional protein for secretion. So this is important. And then when you look at the interaction between signal peptide and the signal recognition particle, they have an interaction in M domain and with the signal peptide through alpha helical. This yellow one is alpha helical structure. They interact through this alpha helical structure. So in signal peptide, alpha helical structure is mainly located in the H region. That is a hydrophobic region. So we thought that we can change or increase or decrease the hydrophobicity of the hydrophobic region, that is H region. But in this case, we decide to increase because some paper already show that. So we decide to increase the hydrophobicity of the H region. So when we increase the hydrophobicity, we decide to use leucine instead of other hydrophobic amino acid. And also from the codon usage table like this, we decide to use a CTZ codon for leucine. So this one is the signal, human IL-2 signal peptide example. There are 20 amino acids, and this part in the yellow box, this region is hydrophobic region. And if you look at closely, then you will find these blue ones are hydrophobic. Okay, let me close this first. Okay, so the blue one is a hydrophobic amino acid, and the red one is a polar amino acid, and the green one, green one is basic amino acid. So in hydrophobic region, the H region, you still can find the non-hydrophobic amino acid. For example, here, serine and cysteine is here. And isoleucine and alanine is not that strong as like leucine in hydrophobicity. So in a previous study with erythropoietin, not interferon beta, in my lab, we changed the hydrophobicity by changing the amino acid in this hydrophobic region. For example, we replaced the serine to leucine, cysteine with the leucine, and both together with the two leucine, or 4 of them, not strong or not hydrophobic amino acid to the strong amino acid, this hydrophobic amino acid, all leucine like this. So this kind of a strategy we applied, and by something is wrong. Okay, so by doing that, in a previous study, we could get the EPO concentration, transient transfected EPO production was increased, like a 2.7-fold increase when you change hydrophobicity in signal peptide. So this time we would like to focus on interferon beta. Interferon beta is very difficult to produce. The concentration is very low, and it is very unstable protein. So it's very difficult protein. So right now, this time we try with interferon beta. So here, this is the signal peptide of interferon beta. Most of the the secretional protein has their native signal peptide. So interferon beta is a secretional protein, so it also has its native interferon beta signal peptide, which is this one. So, and when you look at the hydrophobic region, it's very similar to IL-2 we already done with previous research. So we follow the same approach. We apply same approach to interferon beta signal peptide. So we change glutamine to leucine and the QIA to 3 leucine as we done in previous research. And then we see the result of this approach. So this is introduction, and then I will explain the material method. For this research, we used the 4 different cell lines, HEC293 cells and CHO cells. One is HEC293A, which is adherent cells, and HEC293F cell is a suspension cell. And we have 2 CHO cell lines, CHO-K1 for adherent and CHO-S, XP CHO-S for suspension cells. because we try this, because what kind of a cell line is good for interferon beta production, we need to decide. So we have to use for all of these 4 cell lines to make to do this research. For audience cells, we use lipopectamine 2000 for PEC293A and CHOK1. We follow this process. And for suspension cells, we use the polyethylenemine or XP-fectamine for ex vitro cells. So by using this kind of transfection protocol, we could get the transient transfection of our vector developed, express vector developed. And to make a stable cell line in the final, because we want to make a final cell line that produce stably, not transient. So we need to make a establish stable cell line. So we did 2 different kind of selection protocol. First one is Geoshin. So selection 1, phase 1, 2, and we use MTX selection protocol. So we follow the separate the selection process, and followed by the limited deleting method to have a single cell clone. And then we actually developed sandwich ELISA method to analyze the interferon beta concentration. And most of the material, like expression vector, we use pcDNA 3.1, which one is commercially available. So there's no secret material. We use the commercially available product, and we used already known information to develop, to increase the productivity of the cells we produce. So result. So first one, we changed the hydrophobic region of interferon beta synapeptide. So we actually increased the hydrophobicity of the hydrophobic region like this. For example, here we change the amino acid like this, and then the results like this. This one is interferon beta synapeptide wild type. It's a native one. When we change it, the hydrophobicity by replace the non-hydrophobic amino acid to hydrophobic leucine, we could get this kind of result. And of course, when we use no signal peptide, there is no protein secretion. So that's pretty much acceptable. However, the data shows that the opposite to our expectation, interferon beta productivity was not successful. So what's gonna happen? So, but we have a good experience with the IL-2 signal peptide. So we decide to, maybe the interferon beta signal peptide is not that strong, so we decide to use the IL-2 signal peptide. So we went back to the IL-2 signal peptide that we already succeed in EPO. So we did the same approach, and then this one is number 1 is a wild type interferon beta signal peptide, and the number 2 is a wild type IL-2 signal peptide, and then we tested different combination of the hydrophobicity fortified signal peptide of IL-2, and then it did not increase much, but still we could get the 1.5, 1.84 increase compared to the native interferon beta signal peptide. So we could get almost 2-fold increase. Is that enough? Actually, if it is a bioprocessing optimization process, 1.8-fold increase in productivity is pretty much acceptable and very successful. However, we are talking about the cell line development and vector development. So we could not satisfy with this kind of small number. So what else we can do? And then we learned that the Kozak sequence is very important to get high productivity in vector engineering. And the Kozak sequence is the GCC RCC sequence. So R is a purine sequence, and so it can be adenine or guanine. So ACC or GCC, ACC or GCC sequence before the initiation codon. So if you put this sequence in there, Kozak sequence, we learned that you can get higher productivity. So we decided to apply this sequence in our vector. So this is the result. So when we do experiment with the HEC293 adherent cells, we could get, this is the number one is IL2 modified, SCIA changed to 4Ls. So this is modified IL2 signal peptide. When we put the Kozak sequence in there, we could get about 3.5-fold increase, but just by adding the Kozak sequence, it's good. And compared to the wild type, the interferon beta native signal peptide, we could, this number is about 6.5-fold increase. So this is good. And in the HEC293F cells, this is suspension cell, we did similar experiment, and then we could get the 3.5-fold increase. And then actually HEC293F, the suspension cell, has a more productivity compared to the two HEC293A cell line. So compared to these two, we could get 3.2-fold. So compared to the initial interferon beta native signal peptide in HEC293A, we could get 21-fold increase in Kozak modified IL2 signal peptide produced in HEC293F cells. So this is good. We could get 20-fold increase. So compared to the previous one, 1.5, 1.8-fold increase, we could get the pretty much increase in productivity. So this is good. And also we did in CHO cells, because CHO cell is mostly widely used in industry, biopharmaceutical industry. This is also important. So we could get good increase, 9-fold increase with the Kozak sequence, and the 3-fold increase in Kozak sequence, especially in CHO-S cells, and this one is CHO-K1. And then the concentration different, CHO-S cell line, we could get the, between these two, the difference is about 104-fold, more than 100-fold. So if we think about from the native interferon beta signal peptide, it is more than 1,000-fold actually. When we use XP CHO-S cell line with Kozak and modified IL2 signal peptide, we could get 1,000-fold increase before and after. So this is amazing. And then, okay, then we try to find is there any other way to increase the productivity. We already have a modified IL2 signal peptide. So we want to change a little bit more because we have in here, this is our modified signal peptide. We have 4 extra leucine, and originally there are 3 leucine here, and then we change it 4 amino acid to leucine. So we have 7 leucine in serial. So so we want to see if we change the number of leucine in this hydrophobic region, what's going to happen. We just curious about that. So we changed that. So this one is our modified signal peptide with the Kozak sequence. And then we changed the number of region. This one is 5 and 6, 7, 8, 9. So we changed the region number, and but we could not get the enough productivity as we have already. So it seems like the productivity produced by our hydrophobic region modified IL-2 signal peptide is optimal, very close to optimal condition. And actually this one, number 4, has a 7 region, and our the modified one also 7 region, but difference is a codon different. So codon is a little bit optimized already in our signal peptide, but CTG might not be best in this occasion. So that's the another thing. We could get the better productivity in our signal peptide. Okay, and then we actually this is the hydrophobic region. That's why we have interest. But there's another reason. This is a basic reason, and we cannot change all of this. But there is one polar amino acid, so glutamine here, and that here is a methionine. So we try to see, because the barrier between this is not clear. So we decide to change the glutamine to leucine to see the effect, and also we decide to change it both two to leucine like this. But the results show that if we change the M or M2 to leucine, we could not get the good productivity compared to the original one. So we should not touch anything about this basic region, and we should keep the size of the hydrophobic region as same as we have. So it seems like we have a optimal optimized the condition of the signal peptide already. And the next step is to make a stable cell line. So we start with the geosine selection system. We did with 400 microgram per milliliter of geosine, and we could get some cells survived already. And then we did the second phase selection system. And then after that, we could get, this one is signal Kozak sequence and signal peptide optimized, modified system, but it's a transient gene expression, transient transfection. And after we make a stable, selection system, second phase, we could get this one. And then when we compare these two, there's almost a 12-fold increase compared to the transient transfection and the stable pool, not a single clone. It's a stable pool cells. We could get a 12-fold increase. So we select this pool to isolate single clone. So we did a limited dilution cloning procedure. And then we finalized the 3, most productive clones, 3 of them, and then we finally cultivate them and then compare the productivity. So we have 3 different clones, so Z1, Z2, G3 clone, and then we could see the G3 clone shows the highest productivity, and like this number, and this number is compared to the transient gene expression system, this clone gives us a 35-fold increase in productivity. So actually, this the increase in productivity is more than 1,000, because we already know that when we use the native interferon beta signal peptide, This one is already 1,000, and then we have extra 35-fold increase. So it's pretty much quantum jump from the beginning to the current stage. And also we did the MTX methotrexate selection phase, and we could get the very similar productivity like this. So conclusion. So we, in order to develop the highly productive cell line, we have to touch the signal peptide vector engineering. From interferon beta wild type, we used IL-2 wild type, and we changed the signal peptide in hydrophobicity, and then we put the Kozak sequence. And when we did the IL-2 signal peptide, we could get 1.5-fold increase, and then 1.8-fold increase when we modify the hydrophobicity. And when we add the Kozak sequence, we could get the 6.5-fold. And then use HEK E93F cell, 293F cell line, we could get the 21-fold. And then also when we did the same procedure with the CHO cells, we could get the 104-fold increase in productivity. So this is pretty much high productivity compared to the signal peptide from interferon beta. So also when we do the stable gene expression, that means we can establish a stable cell line, cell clone. We could get the 35-fold increase from transient gene expression. We could get the 35-fold increase, and also when we use the MTX selection system, we could get 28-fold increase in productivity. So today I show that how we successfully increase the productivity in cell line engineering. Especially we touch the signal peptide, we change or modify the hydrophobicity, and we put some the important sequence, Kozak sequence there. And then after we finalize the selection system, we all together we could get the more than 1,000-fold increase compared to the non-treated original native signal peptide applied cell culture system. So this kind of thing, I don't know, but maybe some of this finding we got from here can be applied to the developed cell lines producing recombinant therapeutic cells, therapeutic proteins, which is a kind of major part of biopharmaceutical industry in these days. So this is the end of my talk. Thank you very much for your attention.
Thank you, Professor Dugdeo, for a very great presentation. And let us give a round of applause to Professor Dugdeo, and you can also give reaction.
Thank you.
Thank you, Professor. Ladies and gentlemen, we will continue to our next agenda, which is plenary session with speakers, Professor Zihua Jiang from Department of Animal Sciences, Washington State University, USA, and also Mr. Zahir Ali, PhD, Center for Desert Agriculture, King Abdullah University of Science and Technology. This session will be moderated by Professor Siti Subandia. Now, before we begin, allow me to read a brief profile of Professor Siti. Professor Siti Subandia earned her doctoral degree in plant pathology, the United Graduate School of Agricultural Science, Gifu University, Japan, master degree in plant pathology, Faculty of Agriculture, University of Queensland, Australia, bachelor degree in phytopathology, Faculty of Agriculture, University of Gadjah Mada, Indonesia. She is currently a professor in Faculty of Agriculture, University of Gadjah Mada, where previously she was also a faculty of member in 1983 to 2017. She was honorary professor in the university. She is currently the honorary professor in University of Queensland, Australia, guest professor in Gifu University, Japan, and visiting professor in Shizuoka University, Japan. Her research interests are in the area of diagnosis and management of plant diseases and molecular plant protection, and also agriculture biotechnology. Now, without further ado, Professor Siti, time is yours to lead the session.
Okay, thank you very much, Ibu Wiwit. A very nice introduction of me. And good morning, good afternoon, or good evening for everybody in this meeting. I hope everyone is stay healthy, and we have already having better condition. The pandemic already declining. Yeah. Okay. Thank you very much for this opportunity for me to lead this talk. And the first speaker will be Professor Ji-Hua Jiang from USA. Yeah. Can I read the CV of Professor Jiang? Okay. Professor Ji-Hua Jiang is a full professor at the Department of Animal Science, Washington State University, as a faculty member of the University of Guadeloupe in 1997 to 2002. And he was doing postdoctoral at Roslin Institute in UK, 1994 until 1997, and then as a faculty member at Nanjing Agricultural University in China since 1986 to 1997. So his research interest is focusing on the studying of the origin of phenotypes using state-of-the-art genomic and transcriptomic approaches in combination with bioinformatics and cell embryo biology. So it's very interesting field. So Professor Ji-Hua Jiang, sorry, you have 30 minutes to present your research, and we are looking forward for your great talk. Okay, thank you, please.
Can you hear me?
Yes.
Okay, wonderful. Just to let you know, we have kind of the heavy being outside, or you hear some kind of background noise. Sorry about that. First of all, thank you so much, Professor Dr. Suban Dian. Hopefully I can pronounce your name correctly.
Just call me Siti.
Oh, okay, Siti.
Siti is easy.
Okay, Professor Dr. Siti, for your nice introduction. I would also take this opportunity to thank the conference organizer, Dr. Bao, for inviting me to give a talk today. My topic today is biodiversity. Does RNA diversity matter? As you know, biodiversity has been a hot topic in biology for more than 35 years. Aims to study variety and variability of life on Earth. As you can see here, the research field mainly focus on three areas: genetic diversity, species, ecosystems. No doubt, the number of species on Earth is enormous, while the ecosystems are very, very complicated. So therefore, my talk today would focus on genetic diversity, particularly on RNA diversity.
The committee haven't presented the PowerPoint.
So, can you see everything clear?
No,
okay.
We cannot see it.
Can you see my slides?
I cannot see in my laptop.
This is clear in my.
Oh, sorry. What's wrong with
Can you see my Can you see my slides?
Yes.
Yes, we can.
Oh, sorry. I cannot
Okay.
see in my. Oh, sorry.
Oh, sorry about that. Okay, the question here is why RNA diversity? First, we can simply classify our human genome, for example, into the exonic, intronic, and intergenic regions. Based on ENCODE studies, it estimated that only 7% our human genome is silent. So that means that the remaining 93% of our human genome are actively transcribed and associated with at least one primary transcript. As you can see here, that includes 1.2% expressed from protein coding exons, 39% derived from introns, 3 UTRs, and also 44% mapped outside of the protein coding genes. That's kind of one thing we need to know. Second, in addition to protein coding genes, you can see here there are several types of non-coding genes, such as SNRNAs, RNAs, tRNAs, long non-coding RNAs, microRNAs, and pRNAs, for example. Third, you can see here a gene often use more than one transcript. And for example, alternative polyadenylation can occur at the tandem 3' UTR sites, alternative terminal exons, intronic sites, and exonic coding sequence size. So what's the consequence if you look at these, these alternative transcripts? For the first top two, you can see these two transcripts just have different lengths of 3 UTR regions. And they do code for the same proteins. That means no protein change for first two alternative transcripts. However, you can usually there are lots of regular elements residing in the 3' UTR regions. For this case, for example, and the short version of transcripts may lose the microRNA size, so the transcript should stay much longer than longer one. So from here, you can see, so-called quantitative effects when two transfers are used in the process. So again, if we look at bottom three alternative transcripts, they may code for different properties. So these properties may have a different property. As such, we'll see so-called qualitative effects on gene function. In some case, protein coding gene can be converted into non-coding gene. As you know, obviously, non-coding gene will execute epigenetic effects. So anyway, so you can see here when a gene uses different isoforms, it may functions quantitatively, qualitatively, or epigenetically. So anyway, so gene overall use of alternative transcripts makes a finite genome capable to coordinate an infinite phenome. Okay, somehow I got something here. I could get fixed right away. So you can see this slide shows you a typical structure about mature RNA. So in this case, if you would like to profile a gene, everything from the 5' end to 3' end. So you can use RNA-seq, conventional RNA-seq, or iso-seq. However, if you would explore how alternative transcription start size or alternative polyadenylation size are used in response to different conditions. So you need to use either 5' end sequencing or 3' end sequencing method. So here, I just show you our methods to profile alternative polyadenylation sites. That's called whole transform termini site sequencing, or just WTTS thing. As you can see here, our WTS-seq methods uses total RNA as starting materials. The assay involves fragmentation, poly-RNA enrichment, first strand cDNA synthesis by reverse transcription, and the second strand cDNA synthesis by PCR. Actually, I want to tell you a few unique features about our WTTS-seq methods. So you can see here, actually, our WTTS-seq methods as both 5' adapter and 3' adapter simultaneously by during reverse transcription process. That's number one. Number two, as you can see here, poly-A RNA ferment are enriched using oligo GT 25 bits. And also poly-A CDN, okay, poly-A plus CDN molecules are enriched by using RNSI and H treatment. Third one, where you can see our primers here, adapters. So basically, we use so-called poly-A anchored primers to do PCR. So that's avoiding any PCR amplification detour. Lastly, we try to avoid any over amplification. So basically, that helps us to maximize long-term coverage. Now, let me tell you a few advantages our WTTS-seq methods over the conventional RNA-seq method. So first one, you can see here, compared to RNA-seq methods, our WTTS-seq can directly measure the diversity and the dynamics of APA patterns. So you can see here, we use the embryos at the stages 6, 8, 11, 15, and 28. Use of frog embryos, and the same samples derived at stage 6, 8, and 11 were also done used for RNA-seq analysis. So from the top part, you can see here, you see two APA size. You can call them, one distal one, another one proximal one. So you can see at stage 6 and 8, distal APA site is more abundantly expressed than the proximal site. However, things are changing. So I would say, at 11 and 15, you can see the proximal site the proximal site is more abundantly expressed than the distal site. However, at the stage 28, looks like both sides are kind of balanced. However, if you look at RNA-seq profiles, you cannot see any such kind of changes. Obviously, if you look at transcripts, there are pretty long ones. There are pretty, short ones. For example, so this is Xenopus trabeculus ribosomal protein like genes 34 RPL 34 ribosomal protein L 34. So this gene, okay, basically you look at express mature RNA. It has just 449 base pair, so it's pretty short. So you can see, look at the RNA-seq data. So that's kind of, obviously, that's not well presented in the data set. So basically, conventional RNA-seq is biased against 3 palm ends of transcripts, so you can not see clearly their expression levels. However, if you look at our WTTS-seq, and that's pretty clear, and the gene expression increases along these developmental stages. Now that in our genome, there are overlapping genes, our design is very unique, which our WTS-seq just a sequence from the polysize, so that's kind of the strand-specific sequencing. So as you can see here, you can clearly assign reads to the overlapped genes based on the color. However, if you look at conventional RNA-seq data, and we have no clear way to assign reads to each gene. So as I said, our WTS-seq uses strand-specific sequencing, so that reads can be assigned to overlapping gene easily and clearly. And also, obviously, as you can see, APS size can be extended from exon regions to introns. They might be located in introns or expressed from the antisense strand. There are cases. So if you look at RNA-seq data, basically you have no way to identify them. If you look at our WTS-seq data, so that's pretty clear. For example, here is a DCP1A gene. As you can see, APS site is expressed at starting at stage 11, and that's pretty dominant at stage 28. Well, and here this slide shows you comparison of transcriptome distributions. As you can see, RNA-seq data produce pretty wider of the wider distributions than the our WTS-seq. As a consequence, RNA-seq analysis would exaggerate the numbers of differentially expressed genes. So for RNA-seq data, you may see some false positive results. Here's the last one, based on, so usually for RNA-seq data, and you need to have at least, RNA-seq requires at least 30 million reads per library. However, for our WTS-seq, usually we use, we just require 5 to 10 million reads. So overall, our technique will save some sequencing cost. Okay, now I will use a few examples to tell you some interesting story. The first one I want to show you the relationships from gene knockout, APA, and muscle mass changes. So here, as you know, MPK alpha 1 and MPK alpha 2 are two alpha subunits of adenosine monophosphate activated protein kinase, which serves as cellular sensors in regulation of many biological events. Here we investigate MPK, MPK-R1 knockout and MPK alpha 2 knockout mice on muscle mass changes. So the knockout mice was produced by Violet and colleagues in 2003. If I remember correctly, the deleted exon 2 for each gene, and so these genes are located. AMPK-R1 is located on chromosome 15, and MPK-R2 resides on chromosome number 4. So we used muscle samples to profile APA events. So as you can see, our analysis shows that down-regulated, you can see you can see upregulated and down-regulated genes, between two knockout mice. Actually, we use the wild type, as a control, as a control. So for the pathway analysis, so we used the either upregulated gene or downregulated gene for pathway analysis. At the end, we observed that downregulated APA size in MPK alpha 1 knockout, okay, knockout, and are downregulated, while many genes are upregulated in alpha, MPK alpha 2 knockout mice. So at the end, you can see MPK alpha 1 knockout mice have much smaller, muscle area, while MPK alpha 2 mice produce much larger muscle area. So kind of you can see here, profiling APA size is a sensitive and a powerful approach that can be used to detail the information flows from a genome to transform to proteome and explain the altered phenotypes between knockout mice and wild type animals. So here I use frog, embryos and young males and growing females to tell you a story about development. APA.
Can you have slideshow, Professor John? Slideshow to have bigger view.
Okay, yeah, thank you.
Yes, that's better.
Okay. Okay, so here you can see we use the embryos derived from, again, from 5 stages, 6, 8, 11, 15, and 28. So therefore, adults, so we have 2 stages, kind of young animals and growing animals. One question here, I put it in a box here. So first question, are males and females genomically equal? That's kind of a question here. As you know, in mammals, males and females differ in sex chromosomes. Males have XY sex chromosomes, and the females have XX, sex chromosomes. As you know, in order to balance the dosage differences between sexes, one of the X chromosomes in females is inactive, but with 15 to 30% of its genes activated to counterpart the genes held by the Y chromosome in males. So I would say males and females are genomically equal. However, gender difference exists. Gender difference affects many different processes. So as you can see from the top left side, so that basically tell you that's kind of principal components analysis revealed kind of relationships among these samples. And based on color, you can clearly see where embryos are located, where the few young males and young females, growing males and growing females are located. Overall, you can see the APA transcriptomes of young animals are more variable than growing adults. That's kind of one thing you can see there. And then you can see overall male frog APA transcriptomes are more divergent than females. Yeah, you can see if we look at the 6 growing males and females, obviously 6 females are clustered, together, while you can see pretty separations among 6 male animals. Why? So here I just use one pathway to tell you the story. So if we look at the genes involving sex differentiation, that's pretty interesting here. Do you see among 110 genes involving sex differentiation, only 20 are common to both males and females. As you can see here, 6 specific genes are involved in male sex differentiation, while only 30 genes are specific to female sex differentiation. And I don't think I have time to go through pathways in details, but here you can see a story you can look at the pathway, and I can, use the question here: why do females live longer than males generally? So also you can see a specific pathway for the telomere maintenance. So you can see females have much better situation in telomere maintenance than males. So that I don't think you need me to explain further. So that tells a story why females lives longer than males. Well, so let me probably need to speed up a little bit. So here you can see kind of issue obesity is a huge issue in the United States. The current obesity rate is for 42% in the US. So here we look at APA event in response to high fat diet. And you can see here, high fat diet triggered more genes upregulated in hypothalamus. As a consequence, so these animals ate more food and grew more body weight. So here's is another interesting case about cannabis exposure, APA, and brain functions. As you know, marijuana use has been legalized in many states in the USA. And so based on kind of reports, and 48 million people used marijuana at least once in 2019. Okay, that's a big number. That's a big number. So we use a rat and also one-time exposure, that's acute exposure of cannabis for 10 minutes. So as you can see here, and hypothalamus responded to cannabis exposure pretty quickly. So as you can see, many genes were downregulated in response to cannabis exposure. So if you look at the pathways, so basically cannabis exposure impairs learning, memory, mode, moment, sense, and thinking. That's pretty reflect what people feel when they use cannabis. This kind of last example, that's also a big issue in dairy. Okay, so that's try to tell you a story about pathogens, APA, and endometritis in dairy cows. Endometritis is one of the most devastating reproductive diseases in dairy cattle, resulting in annual loss of more than $50 million in the United States. So we screened dairy cows and collected samples from healthy and healthy and unhealthy cows. So basically, we look at here. We have collected 18 samples, and we could, based on APA profiles, we are able to classify them into clinically healthy animals and conditionally healthy dairy cows, clinically mild, mild endometritis cows, and clinically severe, severe endometritis case. As you can see here, and lots of pathways related to immune response are upper regulate. I want you to pay attention. So for healthy cow, you can see the pathway called psyllium moment is dominantly enriched. So in order to keep cow healthy, basically, psyllium moment function has to be normal. So anyway, these are kind of stories tell you how APA events in response to internal, external, and environmental challenges. So here is my kind of summary. And so if you look at the genome annotation, so we know how many number of genes in our genomes. However, if we look at numbers of alternative transcripts, the number is much higher than number of genes. Based on our research, I can tell you that alternative transcripts rather than genes are minimal functional units expressed by genomes. So I guess use of alternative transcripts makes sense. Makes sense. If you look at as I said, if you look at our phenome, okay, that's a phenome. That's pretty evolving, and you can see many phenomes. However, you look at our genomes, that's kind of the limited. So basically, use of alternative transcripts makes it possible for finite genome to coordinate infinite phenome. So overall, I would say alternative transcripts serve as bridges between genomes and phenomes. And if we want to understand, create, or maintain the biodiversity, so we need to pay attention to the use of alternative transcripts. The last things I would suggest here, hypothesis to be tested, loss of RNA diversity results in extinction of organisms. Thank you so much for your attention.
Thank you very much, Professor Xiang. So we have run out our time, and we will invite our second speaker, Mr. Zahir Ali, PhD. I think he's available now, and so we will have a question and answer after the second presenter finishes his talk. Is it okay? Okay. So, Dr. Zahir Ali, please.
Hi.
Hello.
Can you hear me?
Yes. Okay, thank you.
Yep.
Okay. Welcome, Dr. Zahir Ali. Yeah.
So, yep.
Can I
Zahir
introduce
Ali.
you? Yeah. Can I introduce you first? Okay.
Yes.
Mr. Zahir Ali, PhD. He got his PhD from Plant Molecular Biology and Biotechnology, Gyeongsang National University in South Korea, and then his master at Quaid-e-Azam University, Islamabad in Pakistan, and then his PhD, his BSc was in the University of Peshawar, Pakistan. Okay, his work experience as a research scientist at KAUST, Tual, Saudi Arabia, and then postdoctoral fellow to 2013 to 2018, right? At Kaustul, Saudi Arabia also, and then lecturer at Department of Biotechnology, University of Malacca, Pakistan in 2003-2007. So he has very good experiences. So his research interest is in developing tools for plant genome engineering using specifically the CRISPR-Cas system. And similarly, he likes to explore plant potential to be used as biofactories for the production of various bioproducts, drugs of therapeutic importance. Okay, very nice. Please, Dr. Zahir Ali, you have 30 minutes for your presentation. Thank you.
Yep, thank you. I will share my screen. Can you see my slides?
Yes, it's coming.
Yep. Yep. My name is Zahir Ali. Good morning. Here it's morning. I don't know what time is there. Anyhow, it's really a good opportunity for me, and especially Dr. Rekno. He told me to participate in this one, and I'm really thankful for this, making this event. So actually, I'm a plant genome engineering scientist, but as you know, the recent spread of the COVID, and actually the system was ready. We were using it for plants, but the system can be used for any pathogen or any virus. So we developed something also for the COVID-19. So I will present today our recent work, which is just accepted, and I will just present this work for this event. So the title is BioScan. We call it BioScan, a CRISPR-Cas9 based lateral flow assay for detection of pathogens. Why a BioScan? Like, for example, as we know, the COVID-19 pandemic, it's just started like 2 years back, exactly 2 years back. And then here you can see that actually in October 2020, the cases were just 34 million. But just in 1 year, the cases become very high, and you can see like 234 millions were there in October, and confirmed it globally from this pandemic. In October, it was in October 2020, it was like 1.08, but in October 2021, it is 4.8 million. It's a huge, and still in some countries, still the lockdowns and all these things and all the lockdown measures are there, and still the people are suffering. But the main thing is like how to constrain a pandemic. So this was the call from World Health Organization, like they call it the 3T call. What is basically 3T call? It is like test, test, and test. So they just have like, they just say, we have a simple message for all countries, test, test, and test. Why test, and test, test, and test? We make it a little bit more elaborate. Here you can see, like, for example, if there is a test that is affordable, fast, sensitive, and available to all the people, then you can trace. And if you trace, you can identify the cases and you can isolate the cases. And then if these are isolated and identified, then you can provide the treatment, supportive care, and in symptomatic cases. So the currently available method, which is normally used everywhere in the whole world, and it's a gold standard method, which is the qPCR method. What they do, like they just get the nasopharyngeal swab from the sample, and then they extract the RNA, and then they do the qPCR, and then they provide the results in the format of whatever. And here you can see that this is possible only in labs. It's not possible for individual. So what are the drawbacks of the RT-qPCR method? The main problem with this one, like you need a centralized lab where you need very specific designs and operations, and this is really a complex mechanism. And then you need the specialized people, like they should be trained very specifically for the qPCR run and for the result interpretation. Like the operators are not that much easy. Then the next one is like you have to purchase all the system, and as well as you have to hire the highly trained people, so which make it very costly. And normally, if you see the results are like from sample to the result outcome, there is a 24-hour gap, which is a very long time, especially in the case of pandemic like SARS-CoV-2. So what we think that a point of care, a simple test, are needed to accelerate the COVID-19 diagnostic, as we see very long lines when the people are going for the tests and all these things, even like by individual, or we see these lines everywhere when the pandemic started and when the pandemic was there. So these things, there is a huge need for point of care test to accelerate the COVID-19 diagnostic. An alternative should be there, which qPCR cannot do, because the qPCR, there is a limitation of the equipment, the labs, the people. So what we think, we want to develop a point of care diagnostic test. To start with, we look with the World Health Organization recommendation. They have an assured criteria for point of care diagnostics, which means it should be affordable to end user and affordable to health system. And S stands for like sensitive, avoid false negative, and the S specific, avoid false positives again. And user friendly, they should be simple. Like the qPCR people cannot use by themselves. So they should be user friendly, and the people are maybe the simple clinics, they can detect the virus. It will be very easy, especially in pandemic cases. And they should be rapid and robust. Like the results you should get like 24 hours is a lot. The results should be get like within 1 hour or maximum by 2 hours. And equipment free, like there is like everywhere or every setup, or especially the developing countries, they don't have these sophisticated equipments and trained people. So the test should be equipment free, and it should be deliverable to the end user. So we are following this criteria to make a point of care diagnostic test. And if it's affordable, sensitive, specific, user friendly, and rapid, this can enhance the testing facilities for the pathogens, specifically the SARS-CoV-2. So to follow this one, recently the CRISPR-Cas system, which is immune system of the bacterial system, bacteria and archaea, this comes to the diagnostic, and this is called as like the next generation of point of care diagnostic tests. Basically, what is cluster regularly interspaced short palindromic repeats? This is actually the immune response of the bacteria against viruses. So what happened in the bacteria is basically like when the virus attacked the bacteria, it sent its genome into the to the bacterial cell. And what happened in the bacterial genome? The bacterial genome have a repetitive sequence. That's why it's called like clustered regularly interspaced. So it's like there is a cluster which is regularly interspaced. So there is specific region that are repeated again and again, and there are specific protein related to these sequences, which is the case nucleases. So what happened when the virus is there, the bacterial cell is playing a very good play with the virus. It's just take a small piece of the virus, use it as a memory and keep it in the genome. So when next time the virus come, it have a memory for this virus. So in the next step, this is the immunization against the virus. Like the bacterial cell keep a specific sequence as memory against this virus. So in the next step, when the virus come, like next time the virus attack, so here this sequence is expressed as single guide RNA, which is the memory of the virus. And this protein is produced, is sgRNA, and this protein make the complex, and it will cut the virus genome. So the virus cannot attack. So this is really a good mechanism which is present in the bacterial system and archaea system. And then this system is this the targeting of the virus. And this system was recently adopted for genome engineering, because you need only simple sequence of the gRNA, and then you can cut, or then you can use this system for guiding this protein to any nucleic acid. So the system is very simple, and it can be engineered very well for genome targeting, for the virus targeting, or for genome engineering in any species, like mammalian cells, plant cells, bacterial cells, virus, whatever, for the virus resistance or pathogen resistance, and even diagnostics. So we want to take advantage of this simplicity. Like if we have a gRNA and if we have the case protein, we can use this for the detection of the virus as well. Not genome engineering and not virus resistance, but for the detection of the viruses. So to do that, if we look, like recently there are thousands of publications which show like how the Cas9 system is used for genome editing. It's need very simple, the requirements are very simple. You just need a 20 nucleotide sequence of the genome, which is followed by the 3 nucleotides like NGG. This is called as the PALM sequence. And this, if you have a 20 nucleotide followed by the 3 NGG, then you can target this sequence. You just design RNA, which is complementary to this target sequence, and then your Cas9 will make a complex. And then if you have this one, then there will be a cut in the genome. And once the genome is cut, then because there is a random mutagenesis from here, and then there will be insertion and deletion in the genome, or the gene will be knocked down. It can be used for the repair as well, if you provide the repair template. It can be also used for diagnostics, because here you can bind your protein to a specific sequence of the nucleic acid. So the system is very simple and it can be adopted. But how to adopt this system for engineering of a diagnostic test? For that, we develop a mechanism which we call the BioScan, and that is a CRISPR-Cas9 based lateral flow assay for detection of pathogens. And what is BioScan? What is our hypothesis, and what are the components of the BioScan? It's very simple. As we know, the lateral flow assay, which is commercially available, it have mainly 3 regions. Like, for example, it have the gold nanoparticles, which is conjugated with the antiform antibody. It have a streptavidin strip, or the streptavidin is impregnated into the lateral flow. And another line, which is called as control, is the antiform antibody. So this is the lateral flow strips, which is commonly available, commercially available, and it can be used. So we think that these commercial strips, they can be used for the SARS-CoV-2 detection very easily. But what we need a DCAS9, which should be biotin level, because we know that biotin can interact with the streptavidin. So if this is the case, so we have the biotin and we have streptavidin. So this biotin can localize the Cas protein here at this region. And then if this is the case, then we can design a gRNA, which is complementary to the SARS-CoV-2 genome. And then the complex will be localized on the streptavidin strip. And this guide RNA, we can easily make in vitro. This is just like you have a DNA sequence, then you can do the in vitro transcription. And if you produce the protein, then it can be used with the lateral flow strip. And if this is possible, you just need a simple heat block, or even a water bath can be used with a specific temperature. because we don't need any amplification, any PCR amplification. It can be coupled with the isothermal amplification. And we just need the simple pipettes or the simple and tubes and strips. So with this whole setup, you can do very easily the test for the SARS-CoV-2 or any pathogen. Next, we produced like how to produce the bioscan reagent. So we have a system where we put our DCAS9 with AV tag, and we have in the same cassette, in the same plasmid, we have another protein which is responsible for biotinylating this AV tag. So use the biotin is there in the system in the bacterial cell, and then we can purify the BioDCAS9. So to confirm, we induce the system and then we produce these proteins. So here you can see that the size of the DCAS9 is like 180 kDa, and here we can see very clearly that yes, the protein is produced. But the next question is like, is it biotinylated? So we use the Western blot and using the anti-biotin antibody. So using the anti-biotin antibody, here we confirm that yes, this protein is biotinylated. So if this is the case, then we can produce the gRNA, which will guide us to the SARS-CoV-2 genome or the SARS-CoV-2 nucleic acid. So we did the in vitro transcription. Now we have all the components, like the guiding sequence to the SARS-CoV-2 genome, and we have the BioDCAS9, and we have the lateral flow strip. So the whole concept or the working principle is that actually if you have the farm level primers, then you can amplify, first you can use the reverse transcription to convert the SARS-CoV-2 RNA into DNA, and then by the isothermal amplification, you can produce the DNA in 10 to 15 minutes in a single isothermal temperature, no need of any PCR. So we are using RPA method to amplify the DNA. And once this DNA is there, so we have a guide sequence to this DNA, and this is coming from the SARS-CoV. This DNA is made from the SARS-CoV, which is complementary here. So the complex will come here, and once this complex is on the DNA, in the next step, the complex will be bound with the gRNA and with the Cas9. And then we have the ends as farm level, and where we have the anti-farm antibody, which is coupled with the gold nanoparticles. So this whole complex will be made. And the next step, because we are using the lateral flow step, so if you put this complex on the sample pad, it will flow through, and if you have the SARS-CoV-2 genome or the SARS-CoV-2 is there in the sample, it will be stuck here because of the biotin-streptavidin interaction. But if not, it will flow through. So here is just a representation of the positive results. So in positive result, you can see on the test band that the appearance of the color band is here because the fragment will be accumulated or localized on this side. And some of them, like for example, the remaining nanoparticles with the gold in the form, they will move here. But in the negative, there is no band appearance here because this complex is not there, the SARS-CoV-2 is not there, and here on the test line, you will see nothing. So to follow this one, we have a simple sketch that you just collect the saliva from a patient. It can be done with the saliva or nasopharyngeal, but saliva is much more easy, and that you can just spit in a tube. And the next step, a simple RNA extraction protocol we developed. It's a 5 minutes RNA extraction protocol. And then you can incubate at 42 degrees C for RT-RPA, which will convert the RNA into DNA and also amplify the DNA at 42 degrees C. So we don't need a PCR, but we just need a simple heat block or a water bath. And here you are using the anti-farm, the farm labeled primers. So the DNA will be labeled with the farm. And then we are providing this complex. So this complex will be made in the tube, like the BioDKS9, sgRNA, and farm-level DNA complex. So then this complex is applied to the sample bed, and the lateral flow will show you that you have a positive result or you have a negative result. So following this simple procedure, we try to develop our system for the SARS-CoV detection. So like this is just the general image, and here is the real result. We select two targets, like target 1 and target 2. And when we are using the specific gRNA, we can detect the target. But when we are using a non-specific gRNA, there is no target detection. Means our system is working very properly for target 1, and then we confirm with the target 2, another nucleic acid sequence. But the next question is like the different controls. So like, for example, no SARS-CoV-2, no gRNA, no protein, and also the different like SARS-CoV-2 genome and the influenza virus genome. So here you can see that only the SARS-CoV-2 is detected, but all other negative control, they are clean on the test line. So means our test is very specific only for SARS-CoV-2 because of the complementarity of the sgRNA that we are using, the specific sgRNA for SARS-CoV-2. So to confirm that this can be used for the SARS-CoV-2, we select different region. Like for example, we want to amplify a short sequence from the SARS-CoV-2 genome. Here, this is the genome of the SARS-CoV-2, where we have ORF1A, ORF1B, and the spike protein, the envelope protein, membrane protein, and engine. So we select different region, like one in each gene. And here you can see that when we were amplifying these regions, two regions, which is like number 8, which is here from the ORFB, and engine, which is this one, the set 1. Here they are amplifying, or we are detecting these regions very well. Comparatively, these other regions, they were not that much efficient. And the main reason is like the primer binding. So the primer binding is very well at these two regions, but maybe the primers are not binding, or the efficiency of the primer or amplification is not that much good. So then we just follow only with this specific set of the primer to follow it for the detection of the SARS-CoV-2. But one of the main question is like, what is the sensitivity of this test? Because the CDC, they provide a CT value that only this much virus should be detected by a point of care test. So we put different amount of the copy number, like for example, 0 copy number of the SARS-CoV-2, 0.4 copy number of the SARS-CoV-2, 2 copy number of the SARS-CoV-2, 4 copy number of the SARS-CoV-2 genome, and 20 copy, 40 copy, and 400 copy. So here you can see our system is very sensitive, and even 4 copies of the SARS-CoV-2, like generally one microliter contain more than 1,000 copies in the patients. But here you can see that our system is very sensitive, and we can detect 4 copies per microliter very easily. This is just the conversion of this figure into the graphical format. So this shows that our system is detecting it very well. Then we validate our system with the clinical sample. So we collect like up to 94 sample from the clinical patients. And here you can see that on the test line, most of these clinical patients we can detect. Only 3 samples, like sample number 54, 69, and 73, they were not detectable. And these are the negative control, are the people who were recovered from the SARS-CoV-2 and they don't have the SARS-CoV-2 anymore. Next is the question, like there are different variants of the SARS-CoV-2. So is it possible that we can detect the SARS-CoV-2 variants? But these variants, as we know, that these are just point mutants. And the total system is dependent on the gRNA sequence. So let's say, for example, alpha variants have a deletion of 2 amino acids, 69 and 70. So if you design an sgRNA for this sequence, only the mutant, the alpha mutant, but not the wild type. So this can easily detect, or you can predict the prevalence of any specific virus or specific strain present in a specific region. And you can follow the distribution or the prevalence of the specific variants. So we detect, like, for example, the alpha, beta, delta, and one of them is common mutation in all of them. And then we confirm that our system can specifically detect the alpha, beta sequence and delta sequence very well. So next, we confirm that like the next, one of the biggest requirement of the point of care is it should be compatible with point of care, like specifically at room temperature or 4 degrees C, because at point of care, like for airports, stadiums, you cannot use -20 and -80 for storage. So we confirm that can our system work at room temperature, and can we store the reagent for a long time at these two temperatures specifically? For that, we confirm that yes, the system is working. And also the quick extraction of RNA, like for example, can we extract the DNA in 5 minutes? And yes, so we did. Here you can see that the system, when they are stored for weeks, like for example, the components, you can see like for first week, second week, third week, and fourth week, you can see the reagents are still working and they can be used very well at room temperature. There is no need of these storage conditions. And here you can see that when we are extracting the RNA by our method in 5 minutes, the system is working. So as a conclusion, I will say that bioscan reagents can be easily produced in the lab, because all the system you can make easily in any lab, in any molecular biology lab. But this is just the reagent production needs the lab. Other things don't need the lab. Bioscan is comparable in sensitivity with the commercial RT-qPCR because the sensitivity is very high. We see like 95% sensitivity. Bioscan doesn't require sophisticated lab equipments, no need of lab equipments, and it can be done with the simple point of care setup. The other thing is like Bioscan follow the World Health Organization assured criteria. It's affordable, sensitive, specific, user-friendly, rapid and robust, and it's equipment free. And you can deliver it into the users, and we are trying to develop it for like personal use, like if it's available in market, in clinics, so people can do it by themselves. No need to go to the labs. And bioscan detection assay can be performed in less than 1 hour. So like, for example, you collect the sample and then you treat the sample, and then you got your results within an hour. No need to wait for like these 24 hours and also wait for the main labs. So this is our team, the bioscan team. This is Professor Magdi Mahfouz. He is our principal investigator. And Shiva, Nurhan, Ahmad, and Ten, and Rashid. This is the bioscan team, and this picture was taken when everything was locked down and only these 7 people were allowed to come to the university. So I like this picture a lot. And thanks for your attention and questions.
Okay, thank you very much. Great talk. So can we have applause for Professor Ji Huan Jiang and Dr. Zahir Ali? Thank you. And now we have about 15 minutes, even 16 minutes to have some questions from the audience. You can raise your hand, or you also can write your questions in the chat box. Okay. Anyone? Any questions? Hello? No question?
Hello, can I ask? Hello? Hello, can I ask question?
Yes, please.
Okay, hello, Prof. Zahir. Okay, I'm Pir Muhammad from Malaysia. So thank you for your presentation just now. Okay, I just started to learn about CRISPR. Okay, so I just want to ask your your opinion based on your experience. Okay, I feel to design the gRNA is the guide DNA is one of the most critical component of a successful CRISPR. So based on your experience, what's your advice to design a good guide DNA?
Basically, like the gRNA design is not a big problem at all. It's very simple. You just need the NGG sequence, like any like AGG, GGG, CGG, or like you just need NGG sequence and then 20 nucleotide. The only thing, like all the gRNAs, they are not working equally. So you have to search out in your gene which gRNA is working better. You can select the top strand, you can select the lower strand. And the sequence is the same sequence as you are targeting. So because you will design, it will be in vitro transcribed or the system transcribed, and it will become complementary to the lower strand. So the design is very simple. Specifically, if you are designing for Cas9, the Cas9 one is very specific and very simple compared to the Cas12 or the Cas13 and 14 systems. The Cas9 one is very simple and it's very widely used. So you just need 3 nucleotide, which is NGG sequence. 20 nucleotide, you can select any sequence. And NGG, the presence of 2 Gs in the genome, it's very common. So any target can be designed. The system is very simple, like simplicity is there, and it's really robust. The Cas9 is really working very well.
Okay.
Thank you.
Okay. Thank you, Dr. Zahir. Anyone else?
Actually, I have a question for Dr. Zahir. And what's the cost for one test?
Okay. So actually, the good thing is like point of care. Again, there is one thing is affordability. Like it should be affordable. So like if you look at the lateral flow strip, so the lateral flow strip is the main cost. It's $3. The rest of the reagent we can produce in the lab. And like, for example, once you in vitro transcribe it, you can, like one in vitro transcription is enough for 3,000 samples. And one production of the DCAS9, biotin DCAS9 is enough for 10,000. So what we are expecting, it should be under $10. So this is what we are expecting, that the people can afford by themselves, like not by the governments or not. So it should be available in clinics, just like a normal test, like a pregnancy test. So the people can just take it and use it by themselves.
Great. Okay. Any more questions? We still have enough time. Come on. Okay, I would like to have a little question for Professor Chihua Jiang. So on your APA analysis, you in that case, when we would like to use something like antisense for stopping or knock down any unfavorable gene, maybe for the pathogenesis or whatever for the pathogen, how can we have the benefit from your APA pattern?
So basically, you can use our WTDSIC to profile APA changes in response to internal, external, and universal challenges. So that's kind of the tool to look at the kind of mechanisms involving relationships from genome to phenome. Basically, see if you look at kind of for biodiversity research in the past, probably many people have focused on DNA, variation. However, RNA, variation has not paid lots of attention. So obviously, and we have not thought about to use APA to do kind of the pathogen, diagnosis. However, basically, I just show you a few examples, for disease, so we can see, but I couldn't have time to explain everything in details. However, our technique can be used to study kind of the progression of diseases. In human, like cancer is a huge issue. Obviously, cancer is not a kind of one-day event. That takes time to develop. And so like from, something, you start something there and to the kind of pretty, kind of level 4 stage something, there should be lots of changes. So basically, we can use this sensitive tool to monitor kind of the progression of diseases or defects of some disorder.
Okay, thank you. Very nice.
Yeah.
From the audience, if there is any question, we still have some time. No more? No more? Question for Professor Silva. How to maximize the transcriptome coverage? Primers and adapter are designed for what's that?
Okay, that's a great question. That's a great question. When we started to develop our methods, we have tried different primer combinations, and so I mentioned that in my talk that we use so-called poly-anchored primers to avoid PCR amplification detour. That's the thing can help us to maximize the transcriptome coverage. That's kind of one thing you can do. Next one, try to use less cycles of PCR amplification. So as you know, so for our genome, we just have two copies, have everything. However, for transcriptome analysis, things are not equal. Some genes are expressed abundantly, some genes are rarely expressed. So if you do many cycles of PCR amplification, that will favor abundantly expressed, APA size, not a rarely, expressed APA size. So that's the thing we need to pay attention. So in terms of protocol, can be used for plants. I will say yes. Use of alternative polyannulation is an evolutionary conserved event from animals to plants. Yeah, you can see lots of papers published using plant as materials. So yes, our assay will work on plants. So that's not a question. If you have RNA, to the RNA as materials, just use. Also depends on your kind of sequencing platform. So at the moment, our assay is based on ion torrent sequencing platform, not Illumina. So kind of tricky issue associated with the Illumina platform, because Illumina platform requires so-called diversity libraries. So look at our assay, so we have, our reads begins with several T's. So that's also, these are not a diversity library. So that's kind of a unique library. So Illumina system cannot process reads for you. So that's a problem. That's why we use ion torrent system. However, not a problem. We can adjust the parameter design and see, so one day definitely we can use Illumina system to do API profiling. So that's not a problem. Even bacteria, fungi, I guess, many organisms, they use alternative polyadenylation. So definitely these methods can be used for different organisms.
Okay, thank you. So we have still only 4 minutes left. If there is one more question, it's okay. No more? Okay. If there is no more questions, so thank you very much for Professor Sia Hua Jing and Mr. Zahir Ali, PhD, for your very interesting talk, and hopefully we can have more communication outside there. Yeah, okay, thank you very much, and give applause.
Yeah, thank you, it's my pleasure. Thank you.
Thank you.
Yeah.
Right.
Thank you.
I send it to the committee.
Okay, thank you, Professor Siti, and also Professor Zihua Jiang and Dr. Zahir Ali for a very insightful session. Ladies and gentlemen, next we'll have a photo session with all of the participants. So if you could please kindly open your camera in Zoom so we could take a picture together. Okay, so the picture will be taken by the host, and there are 5 slides. Okay, so are we ready? On the count of 3, let me help for the first slides. Give your best smile to the camera. 1, 2, 3. The next slide. 1, 2, 3. The third slide. 1, 2, 3. The fourth slide. 1, 2, 3. And the last slide. 1, 2, 3. Thank you for your participation.
Okay.
Thank you.
Okay.
All right. So ladies and gentlemen, we would like to remind you to please kindly fill out the attendee list of the first session that has been informed in the chat. For the next agenda is Principle and Mechanism of Biotechnology Product Session 2. We will hear presentation from Mr. Pandu from PT Bioscience with the topic Next Generation Sequencing to Advance Microbiology Study in Indonesia. Mr. Pandu, the next 30 minutes is yours. You may proceed.
Yeah. Hi everyone, thank you for the time. Okay. Let me share my screen first, and then please, I want to talk in Indonesia. Okay, ya. Pertama-tama, terima kasih kepada Bapak Ibu yang sudah menyempatkan hadir dalam acara ISIBIO kali ini. Izinkan saya memperkenalkan diri terlebih dahulu. Saya Safitriani, merupakan Senior Field Application Scientist dari PT Pandu Bioscience, di mana PT Pandu Bioscience sendiri merupakan eksklusif distributor dari Next Generation Sequencing, yaitu Illumina System. Seperti yang telah disebutkan, kita akan membahas mengenai Next Generation Sequencing to Advance Microbiology Study in Indonesia. Oke, jadi di sini kita juga memiliki untuk giveaway untuk para partisipan yang hadir di dalam sini. Saya memberikan waktu kepada Bapak Ibu untuk bisa melakukan scan QR code-nya atau bisa langsung hubungi langsung di link yang sudah disediakan di layar Bapak Ibu sekalian. untuk mendapatkan giveaway. Adapun giveaway-nya terdiri dari wireless charger, plastis, and many more. Ini dia merupakan PT Pandu Bioscience. Untuk PT Pandu Bioscience sendiri berlokasi di Jalan Raya Pondok Gede nomor 10B, Duku Kramat Jati, di mana untuk informasi lebih lanjut mengenai PT Pandu Bioscience itu bisa diakses melalui info@bioscience.com atau di www.bioscience.com. Untuk produk dari PT Pandu Bioscience sendiri, kita merupakan eksklusif distributor dari next generation sequencing, terutama di Illumina system, seperti yang sudah Bapak Ibu ketahui sebelumnya. Lalu kemudian kita selain ada Illumina, kita juga memiliki Beckman Coulter untuk terkait dari liquid handling automation, dan ada beberapa dari molecular region juga terkait dari kit ekstraksi maupun kit PCR lainnya. Dan yang terakhir itu juga kita ada 10x genome untuk buat profiling single cell. Lalu selain itu juga kita ada beberapa brand yang kita distributor, yaitu ada Verogen untuk buat forensik NGS solution, ada Shimatsu, proteomic analysis, Major Science, General Lab Instrument, dan juga kita merupakan kerjasama dengan IDT, Archer, dan CarDX untuk LA Type B. Apa yang kita sediakan untuk buat solusi dari Bapak Ibu ketika ingin melakukan research terkait apalagi terhadap dari biologi molekuler. Banyak dari produk bidang keilmuan yang sudah kita bisa cover, baik dari onkologi, reproductive health, genetic disease, microbiology, agriculture, dan molecular cell. Yes, yang mana saja yang bisa di-cover, yaitu sudah bisa di-cover dari universitas, dari academic research center, pharmaceutical company, genome center, biotechnology, hospital, dan sampai ke government. Nah, ini dia. Jadi topik yang akan kita bahas adalah bagaimana NGS memberikan solusi untuk di bidang mikrobiologi tentunya, yaitu adalah salah satunya dengan menggunakan metode whole genome sequencing, atau kita sebut biasanya familiar dan disebut total genome organism, yaitu salah satunya adalah dengan menggunakan culture based whole genome sequencing. Nah, ini salah satu contoh yang diambil dari Journal of Clinical Microbiology pada tahun 2018. Salah satu paper yang dibuat ini adalah ketika melakukan whole genome sequencing dengan menggunakan culture base untuk buat bakteri Clostridium difficile, yang dimana untuk dari sini kita bisa melihat bahwa terdapat analisa pohon filogenetik yang mana menghasilkan adanya kesimpulan, yaitu adanya transmisi dari AMR genes atau antimicrobial resistant di antara farm animal and human melalui dari bakteri Clostridium difficile ini. Nah, untuk teknologi ini total whole genome sequencing lah yang menjadi solusi ketika Bapak Ibu ingin melakukan tracking adanya AMR genes. Nah, ketika melakukan culturing atau melakukan kultur untuk melakukan whole genome sequencing, apa sih konsiderasi untuk buat bakteri dan viral whole genome sequencing? Untuk bakteri sendiri, kita memastikan apakah kultur kita pure monoisolate atau tidak. Lalu bagaimana genome size-nya. Lalu pastikan kalau ketika kita melakukan kultur, dia 100% target organism. Lalu kita lihat di bagian virus. Ketika di bagian virus, kita lihat apakah dia berupa RNA atau DNA, apakah dia berupa single-stranded DNA atau double-stranded DNA, lalu apakah dia bentuknya host atau culture medium dari DNA atau RNA. Jadi kita bisa mengetahui dari sini, ketika kita mengetahui apakah dia pure isolat atau tidak, kita bisa melakukan metode selanjutnya untuk bisa mendapatkan hasil yang memuaskan, seperti itu. Nah, ini dia salah satu pipeline atau salah satu metode ketika Bapak Ibu ingin melakukan next generation sequencing dengan menggunakan Illumina system, yaitu kita bisa lihat di sini ada isolat dari virus, lalu kita bisa bagi yaitu apakah virus tersebut berupa RNA atau berupa DNA. Untuk DNA balik lagi tadi ada single strand atau double stranded. Nah, dari sini kita bisa lihat untuk buat RNA virus, kita perlu melakukan reverse transkriptase terlebih dahulu. Setelah dapat dari si DNA-nya, lalu kita bisa melakukan preparasi. Adapun tahap preparasinya dengan menggunakan kit Illumina, yaitu bisa menggunakan Illumina DNA Prep, lalu instrumen yang digunakan pilihannya antara NextSeq atau MySeq, lalu dilakukan data analisis.
Untuk buat DNA-nya juga sama. Pertama, kita bisa melakukan ekstraksi terlebih dahulu untuk bisa mendapatkan material genetik berupa DNA. Terus kemudian kita bisa melakukan preparasi seperti pada RNA, yaitu dengan menggunakan Illumina DNA Prep. Lalu instrumen yang dipilih yang ingin dilakukan apa, lalu dilakukan data analisis. Nah, ini dia library preparation atau kit yang digunakan untuk buat melakukan whole genome sequencing dengan menggunakan Illumina system, yaitu dengan kita sebut kitnya yaitu adalah Illumina DNA Prep dengan metode pigmentation atau pemotongan. Karena Illumina mengusungkan teknologi yang disebut short read sequencing atau pembacaan dengan jarak pendek seperti itu, maka dibutuhkan pemotongan terlebih dahulu. Nah, untuk itu, tahapan pemotongannya itu dilakukan dengan menggunakan enzim transposom. Illumina DNA Prep ini memiliki teknologi yang disebut sebagai bit link transposom, jadi transposomnya itu terlink dengan bits, sehingga hasil pemotongannya lebih seragam dan memudahkan para peneliti untuk melakukan data analisis untuk selanjutnya. Jadi Illumina DNA Prep bersifat versatil, robust, jadi mudah digunakan, reproduktif, dan fleksibel untuk buat berbagai jenis aplikasi yang akan digunakan. Nah, ini prosesnya, jadi berupa hasil dari genom DNA yang dihasilkan baik dari blood, saliva, atau dari genomic DNA hasil ekstraksi, kemudian input DNA-nya kemudian akan dipotong, lalu setelah dipotong baru kita lakukan preparasi untuk kemudian dilakukan sequencing. Seperti itu. Nah, untuk buat berapa sih input requirement-nya ketika melakukan next generation sequencing ini, itu dibutuhkan konsentrasi sekitar 1-500 nanogram, jadi sangat fleksibel sekali ketika Bapak Ibu memiliki material DNA yang sangat kecil sampai yang besar, baik dari culture maupun unculture, seperti itu. Lalu hasilnya nanti ketika di akhir tidak perlu melakukan normalisasi atau penyetaraan konsentrasi, karena di dalam teknologinya, di dalam library preparation ini, konsentrasinya sudah disetarakan. Sehingga nanti proporsi setiap sampel ketika dilakukan sequencing itu akan seimbang. Dan hasilnya akan lebih baik. Jadi ketika melakukan studi mikrobiologi, salah satunya adalah dengan menggunakan teknologi yang disebut sebagai, atau metode disebut sebagai metagenomic. Nah, untuk metagenomic sendiri, pembaginya ada dua, di mana kita mengetahui yang disebut pertama adalah targeted sequencing dan shotgun metagenomic. Untuk targeted sequencing, itu jadi kita mengaplikasi terlebih dahulu untuk target amplikonnya, itu ada disesuaikan dengan studi yang ingin dilakukan, salah satunya adalah 12S, apakah 16S, CO1, atau 18S, dan ITS untuk fungi. Lalu untuk buat shotgun metagenomic, kita melakukan sequencing untuk seluruh DNA yang kita dapat. Jadi total genom DNA dari lingkungan yang kita ekstraksi, terus kemudian langsung kita lakukan sequencing, baru lakukan analisa untuk selanjutnya. Nah, bagaimana dengan 16S metagenomic sendiri? 16S metagenomic banyak familiar digunakan dengan menggunakan instrumen yang disebut sebagai MySeq system. Sudah ada protokol dari Illumina sendiri untuk terkait dari 16S metagenomics itu sendiri. Nah, dilakukan sequencing dengan menggunakan 500 cycle, atau kita biasa lakukan dengan 2 kali 250 sampai 2 kali 300 cycle, di mana untuk regionnya kita ketahui untuk 16S sendiri memiliki panjang 1500 base pair, yang mana untuk regionnya, untuk wilayahnya itu kita hanya mengamplifikasi di versi 3 dari gene 16S, seperti itu. Lalu kemudian dilakukan analisa data. Untuk analisa datanya itu bisa menggunakan MySeq Reporter atau longkaran manager dari instrumen. Jadi di dalam instrumen Illumina itu sudah diinputkan dengan bioinformatics software. Bapak Ibu bisa langsung mendapatkan data ketika melakukan sequencing, atau bisa menggunakan base pair sequence hub. Yang mana base pair sequence hub ini merupakan software analisa data berbasis cloud. Jadi dibutuhkan akses internet untuk buat melakukan analisa data itu sendiri. Untuk protokolnya sudah disediakan oleh Illumina, tinggal mengikuti prosedur yang ada. Nah, ini bagaimana workflow-nya ketika melakukan 16S RNA sequencing, yaitu dilakukan sampel prep terlebih dahulu untuk bisa mendapatkan total genom dari lingkungan. Lalu setelah kita mendapatkan total genom dari lingkungan, kita hanya mengamplifikasi di version 3 sampai version 4 region amplification. Jadi panjang base pair-nya hanya sekitar 300 sampai 600 base pair. Lalu kemudian baru dilakukan tahap preparasi. Nah, ketika setelah dilakukan preparasi, indeks yang digunakan itu disebut sebagai indeks Nextera dari Illumina. Lalu dilakukan sequencing menggunakan MySeq system seperti ini penampakannya. Lalu dilakukan analisa data dengan menggunakan base pair sequence hub. Adapun softwarenya yang bisa digunakan atau aplikasinya yaitu Kraken atau 16S metagenomic. Hasilnya ini merupakan tampilan salah satu ketika Bapak Ibu melakukan analisa 16S metagenomic, kita bisa mendapatkan profil mikrobiota dari lingkungan atau dari sampel-sampel yang dilakukan untuk buat 16S metagenomic. Jadi memudahkan para peneliti untuk bisa melakukan profiling bakteria, sudah bisa mendapatkan komposisi bakterianya dan sudah bisa mendapatkan dari taksonomi atau klasifikasi golongan spesiesnya seperti itu. Nah, jadi ketika kita melakukan 16S amplikan metagenomic, kita harus mengetahui apa saja sih keterbatasan ketika kita melakukan studi seperti ini, bahwa akan ada selalu kemungkinan sequencing error di dalamnya, sehingga membutuhkan optimasi atau melakukan pengulangan ketika melakukan sequencing seperti itu. Kita ingin mengetahui apakah komposisi di dalam lingkungan tersebut sudah sesuai atau tidak dengan komposisi bakteri yang kita temukan. Lalu adanya keterbatasan dalam melihat species level identification seperti itu. Terus kita juga bisa melihat kalau misalkan abundance atau komposisi berapa persen rates yang didapatkan ketika melakukan 16S metagenomic ini dia bersifat relatif, jadi tidak bersifat absolut. Lalu ada kemungkinan dia menghasilkan PCR bias. Jadi bagaimana sih untuk bisa melakukan 16S atau melakukan metagenomic, seberapa dalam kita bisa melakukan sequencingnya, bagaimana saya menentukan rates ketika kita ingin melakukan 16S ataupun shotgun metagenomic, itu tergantung dari jenis sampel yang akan kita lakukan ataupun juga dari komposisi sampelnya seperti apa. Lalu kemudian kita juga bisa melakukan hipotesis terlebih dahulu, bisa mengetahui apakah kita ingin mengetahui di lingkungan tersebut harusnya sudah ada komposisi apa saja di bakterinya di dalam lingkungan. Lalu kemudian kita juga bisa melihat apakah rates tersebut cukup atau tidak. Di sini sudah ada beberapa paper yang menstudikan mengenai berapa sih jumlah rates yang dibutuhkan ketika kita ingin melakukan 16S maupun dari shotgun metagenomic itu sendiri. Nah, selain itu, di sini juga metode yang bisa dilakukan dengan menggunakan metagenomic adalah untuk penginjai metagenomic, yaitu dengan menggunakan reagent ITS. Nah, untuk Illumina ITS protokolnya, ini merupakan pipeline yang dilakukan dari collecting extract sample, lalu dilakukan library prep reagent yang bisa digunakan itu sebenarnya bebas, bisa dari manapun. Nanti yang penting cocok untuk di mesinnya, salah satunya adalah kita juga menyediakan dari Illumina. Lalu baru dilakukan organism test, terus dilakukan workflow-nya. Di sini juga sudah kita sediakan protokol untuk melakukan ITS metagenomic. Lalu dilakukan normalisasi atau dilakukan wet lab. Lalu setelah selesai preparasi, kita melakukan sequencing. Dari instrumen yang paling kecil itu ada ISIC, sampai yang paling besar itu Novasic. Lalu dilakukan report dengan menggunakan base pair sequence hub, yaitu aplikasinya dengan menggunakan
Mr. Satriani, are you here with us? Or that it seems like the connection from Mr. Satriani is rather bad. So perhaps another person from Pandu Bioscience, if you could please continue with the presentation, or while we're waiting for Mrs. Satriani to join us. For Mrs. Aulia from Pandu Bioscience. Are you here with us? Okay. Mrs. Safitriani, it seems like she already joined us. It's still unmute, Mrs. Safitriani.
Yeah, sorry for the interruption. So I can continue my presentation.
Yes, please proceed. Thank you.
Okay. Ya, terima kasih Bapak Ibu. Mohon maaf atas gangguannya. Jadi seperti yang sudah kita jelaskan di sini sudah ada gambaran mengenai bagaimana para peneliti melakukan ITS metagenomic seperti itu. Bisa terlihat dengan menggunakan ITS metagenomic pada base pair sequence hub, kita bisa mendapatkan profil dari fungi yang sudah dihasilkan dari suatu sampel lingkungan. Lalu kita bisa melihat komposisi atau seberapa banyak fungi yang bisa yang terdeteksi ketika kita melakukan ITS metagenomic ini. Nah, ini untuk bulletinnya. Jadi kita di Illumina juga memiliki application note terkait dari bagaimana profil dari fungal sequencing and classification with the ITS metagenomic, bagaimana prosesnya, terus kemudian bagaimana juga tahapannya dari awal sampai akhir, terus hasilnya seperti apa. Nah, selain itu juga dari tadi kalau misalnya kita bahas mengenai ITS dan Cysteine S, maka di sini juga ada yang disebut sebagai shotgun metagenomic. Jadi kalau misalkan untuk buat 16S dan ITS, itu kita hanya mendapatkan amplikonnya. Jadi prinsipnya kita mengamplifikasi terlebih dahulu region amplikon yang ingin kita dapatkan, terus kemudian baru kita melakukan sequencing atau NGS. Sedangkan kalau shotgun, hasil sampel dari lingkungan, kemudian kita lakukan sequencing untuk seluruh total genom di dalam lingkungan tersebut. Jadi kita misalkan dari soil atau dari tanah, kita ambil, kita ekstraksi, terus kemudian total genom dari tanah, kita langsung bisa melakukan preparasi dengan menggunakan kit yang dari Illumina, baru kita bisa mendapatkan seluruh informasi terkait genom-genom apa saja yang didapat di dalam suatu lingkungan tersebut. Seperti itu. Nah, ini dia keuntungan dengan menggunakan shotgun metagenomic. Keuntungannya adalah bahwa shotgun metagenomic ini culture independent methodology, jadi kita tidak perlu melakukan culturing terlebih dahulu. Kalau misalkan saya mau ingin mengetahui bakteri apa saja yang didapat di dalam suatu lingkungan, kita harus culture terlebih dahulu, lalu kita pisahkan setiap organisme di dalamnya dengan menggunakan shotgun. Kita tidak perlu melakukan culturing. Dia juga relative abundance of microbial taxa seperti yang pada Cysteine S metagenomic ataupun ITS. Metagenotyping artinya kita juga bisa melihat atau mengidentifikasi adanya gene loss, gain atau mutation di antara population cell atau microbial species. Terus kemudian kita bisa melihat dia hipotesis free testing. Jadi kita bisa mengetahui seluruh informasi di dalam sampel tersebut, apakah mikroorganisme apa saja di dalam sampel tersebut, apakah mikroorganismenya terdapat archaea, bakteri ataupun yang lainnya gitu, apakah sudah sesuai atau tidak, lalu bisa dilakukan pathogen detection, lalu juga bisa menghasilkan kelebihan-kelebihan lainnya. Nah, ini dia untuk buat application note dari shotgun metagenomic. Untuk kit yang digunakan adalah yang disebut sebagai Next-DNA Flex atau kita sebut sebagai sekarang Illumina DNA Prep, di mana untuk Illumina DNA Prep ini bisa melakukan preparasi untuk buat shotgun metagenomic dengan cara tadi, total genom yang sudah kita dapatkan langsung dilakukan pemotongan. Setelah dilakukan pemotongan, tambahkan indeks atau ditambahkan barcode, baru kita lakukan sequencing. Hasilnya itu bisa kita lihat bahwa dengan menggunakan NGS Illumina, hasilnya terdapat keakurasian data maupun klasifikasi untuk bisa melihat adanya perbedaan komposisi diversitas antar sampel di dalam sampel tanah. Jadi efisien dan komprehensif workflow untuk melakukan shotgun metagenomic dengan menggunakan Illumina sistem, memudahkan para peneliti untuk mendapatkan informasi terkait taksonomi, diversity comparison, juga bisa memungkinkan untuk dilakukan de novo assembly. Jadi kita bisa, salah satunya bisa mendapatkan total genome baik dari organisme-organisme yang terdeteksi di dalam suatu sampel lingkungan, lalu dilakukan functional profiling. Nah, untuk aplikasi-aplikasi atau software bioinformatik yang sudah kita gunakan, itu sudah ada berbagai macam dan disolusikan dalam base-pair sequence hub. Nah, ini salah satunya yang kita lakukan dengan menggunakan shotgun metagenomic, yaitu adalah dengan menggunakan sampel tanah, soil metagenomic. Ini bekerja sama dengan Perkin Elmer, dilakukan fully automated DNA extraction dengan Semagic 360 instrumen, DNA soil kit, lalu dilakukan tadi menggunakan Illumina DNA prep atau teknologi yang disebut sebagai bit-link transposon, dilakukan sequencingnya dengan menggunakan Nexus system dan analisisnya berupa Kraken. Yang kita lihat di sini, ketika Bapak Ibu melakukan shotgun metagenomic, maka visualisasi hasil akhirnya bisa kita lihat seperti ini. Jadi dia diklasifikasi, kita bisa lihat komposisinya. Di sini sudah ada organisme, lalu di sini sudah bisa dibagi organisme apa saja yang terdeteksi dalam sampel tanah tersebut. Seperti itu. Hasil akhir solusi dari slide saya yang ingin sampaikan adalah untuk microbiology workflow solution dari NGS itu sendiri, bahwa ketika kita melakukan studi mikroorganisme dengan menggunakan NGS, kita bisa melakukan berbagai pipeline yang dilakukan, yaitu salah satunya untuk mendapatkan apakah kita ingin melakukan total genome sequencing, apakah ingin melakukan targeted sequencing atau system DNA sequencing, atau ingin melakukan shotgun metagenomic. Tadi shotgun metagenomic jadi mengambil semua informasi di dalam dalam satu sampel, lalu kita melakukan sequencing, lalu dilakukan targeted sequencing virology untuk virus, atau dilakukan RNA sequencing. Hasil akhirnya material genetik yang kita dapatkan, terus kemudian kita lakukan amplifikasi ketika pipeline-nya berupa targeted metagenomic atau targeted amplicon. Terus kemudian setelah didapatkan target DNA yang kita inginkan, lalu dilakukan preparasi libraries, di mana untuk library preparasinya itu juga disediakan oleh Illumina. Salah satunya adalah Nextera XT ataupun Illumina DNA Prep. Ada dilakukan sequencing, instrumen paling kecil yaitu adalah ISIC, terus kemudian yang di sini yang paling populer yang digunakan di Indonesia salah satunya ada MySic system, lalu kemudian dilakukan data analisis dengan menggunakan base-pair sequence hub dengan aplikasi-aplikasi yang disesuaikan dengan metode yang digunakan. Itu dia untuk metode dari NGS itu sendiri. Adapun kita mendistribusikan Beckman culture. Untuk Beckman culture-nya merupakan produk yang digunakan untuk genomic solution. Jadi kita memprovide para peneliti untuk melakukan ekstraksi kit dan juga untuk buat purifikasi. Di sini Beckman culture memiliki beberapa jenis produk, yaitu salah satunya DNA isolation kit, ada RNA isolation kit, total
Recording in progress.
ingin mendapatkan giveaway dari tim kami, bisa melakukan scan pada QR code yang tertera di layar, atau bisa akses di link yang sudah disediakan di layar Bapak Ibu. Adapun nanti untuk buat pemenangnya akan diinformasikan kepada tim kami. Demikian presentasi dari saya. Jika ada pertanyaan, dipersilakan. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
I apologize for the connection problem. So for Mrs. Safitriani, is your presentation is done? Okay, it seems that you're still muted.
Oh, sorry. Yeah, I ended my presentation and thank you for the attention.
Okay, thank you, Mrs. Savatgani from Pandu Biosense. I'm sorry for the previous mistakes. Ladies and gentlemen, before continuing to the next agenda, there would be some documentary videos from our partnership that we can watch. So let us watch the video together.
Halo, kami dari PT ITS Science Indonesia. Hari ini kami akan memberikan penjelasan singkat mengenai pengujian porcine. Mengapa perlu melakukan pengujian porcine pada produk pharmaceutical? Beberapa macam bahan baku atau obat, termasuk tablet, kapsul, injeksi, dan sebagainya, dapat berasal dari produk hewani atau turunannya. Contohnya seperti gelatin, yang sering dipakai sebagai bahan utama kapsul atau stabilizer pada vaksin. Selain itu, antikoagulan seperti heparin juga dapat berasal dari porcine. Sedangkan berdasarkan Undang-Undang Nomor 33 Tahun 2014 tentang jaminan produk halal, produk yang masuk, beredar, dan diperdagangkan di wilayah Indonesia wajib bersertifikat halal. Oke, jadi bahan apa saja yang dapat mengandung porcine? Kebanyakan senyawa tersebut merupakan antikoagulan, digestive supplement seperti enzim lipase, amilase, insulin, dan sebagainya. Wah, cukup banyak ya. Tapi tenang saja, pengujian porcine ini tidak sulit kok. Ada beberapa metode yang dapat digunakan untuk mengecek kandungan porcine pada produk. Yang pertama, lateral flow device atau test pack. Metode ini cepat dan mudah, tanpa perlu banyak peralatan laboratorium yang canggih, tetapi sensitivitasnya rendah. Metode 2, ELISA. Waktu preparasinya dibutuhkan 2-3 jam. Preparasinya cukup mudah dan dapat menguji dalam jumlah banyak dalam satu kali running. Dibandingkan lateral flow device, ELISA lebih spesifik, tetapi masih memiliki batas deteksi sensitivitas yang sedang. Metode 3, real-time PCR. Metode ini adalah golden standard untuk pengujian porcine pada produk pharmaceutical dengan limit deteksi yang sangat rendah 1-5 kopi DNA. Pengujian porcine pada produk pharmaceutical sebaiknya dilakukan dengan metode PCR karena bahan baku yang diuji telah melalui proses pemurnian atau pemanasan tinggi sehingga kemungkinan yang tersisa hanya materi genetiknya saja. Selanjutnya, tahap real-time PCR ada 3, yaitu preparasi sampel, ekstraksi DNA, dan amplifikasi dengan menggunakan mesin real-time PCR. Untuk lebih jelasnya, mari melihat alat real-time PCR dari Roche, Light Cycler 96. Alat berkapasitas 96W ini memiliki 4 channel fluorescence yang dapat digunakan sekaligus dalam satu kali pembacaan. Sebelum memulai proses PCR, profil temperatur dan protokol PCR dimasukkan ke dalam mesin. Setelah itu, tekan tombol start. Secara perlahan akan terbentuk grafik sinyal seperti ini. Sinyal ini terbentuk dari proses denaturasi, annealing, dan elongasi yang berulang, sehingga fluorofor yang terkandung di dalam master mix dapat berpendat dan tertangkap oleh sensor mesin. Sinyal yang didapat kemudian dianalisis menggunakan software Light Cycler, sehingga terlihat sampel mana yang positif atau negatif. Demikian penjelasan singkat mengenai pengujian porcine dari kami. Sekian dan terima kasih.
Halo, kali ini saya mau memperkenalkan alat laboratorium dari Syngene. Ingenious Tree. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel elektroforesis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious Tree, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat sehingga sangat memudahkan untuk mendokumentasikan gel fluorescent ethidium bromida atau cybersafe DNA atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database, tipe gel, dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Since 2019, PT Wadia Prima Mulia has continued to develop in meeting the needs of basic laboratories for Indonesian territories. Until now, our company has been trusted as an authorized distributor for 11 companies that produce the best laboratory equipment in the world. Our highly skilled and experienced sales team spread across Indonesia from Medan, Jakarta, Bandung, Semarang, and Surabaya. Our products. Sales services, including application and technical support, installation and training, also service and maintenance.
In the life sciences, researchers are actively pursuing studies aimed at understanding life, the causes of disease, and achieving breakthroughs in regenerative medicine. To observe and accurately measure intracellular processes and structures, researchers need a microscope that combines sensitivity and speed. Increasingly, scientists require the capability to view cells and tissue at high resolution, as well as their larger context with macro to micro capability. All of these advanced features must be easy to use so that even novice users can acquire high-quality data quickly and simply. Olympus created the FV3000 series to satisfy these needs. This next generation of confocal laser scanning microscopes combines leading-edge imaging features with renowned Olympus optics.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
All right, ladies and gentlemen, for the next, we will hear a presentation from Mrs. Sri Hayuni from Biotech on end-to-end solutions for biopharmaceutical workflow. And for Mrs. Sri Hayuni, the next 30 minutes is yours. You may proceed.
Hello, good afternoon, everyone. My name is Sri Hayuni, and I'm going to share my screens first before I start my presentations. Here you go. All right. So first of all, I'm going to apologize because I'm going to deliver these presentations in Bahasa to make everyone in this room would understand and better about my my presentation. So allow me to deliver this presentation in Bahasa. Selamat siang semuanya. Perkenalkan, nama saya Sri Hayuni. Terima kasih untuk kesempatan yang diberikan kepada saya mewakili PT Merck Indonesia untuk mempresentasikan topik berjudul End-to-End Solution for Biopharmaceutical Workflow. Di sini presentasi ini akan saya saya coba. Just give me a second. I think I have a technical problem. Okay, just sorry. It seems like the PPT is closing. I'm going to reshare again. So sorry about this technical inconvenience. Okay. Oops. Just give me a minute.
Do you want me
Do you
the
have
end to end?
Yeah.
Yeah.
It's about to. Okay, cool. All right, cool. Does my share screen?
Yes.
Can you see
you
my
can.
screen? Okay, thank you.
Yep.
Okay, cool. Yeah. Sorry for this inconvenience, everyone. So yes, I'm just starting my presentation again. Jadi presentasi ini akan dibawakan oleh saya saya Hayuni M Biotech selaku Cell Culture Manager dari tim research PT Merck Chemicals and Life Science. Baik, sebelum saya memulai presentasi, saya ingin memperkenalkan sedikit mengenai portfolio PT Merck Life Science yang saat ini sangat ingin kita branding sebagai life science company setelah mengakuisisi beberapa brand seperti Sigma Aldrich. Di kita sendiri ada beberapa divisi, yang pertama healthcare, kemudian performance material, dan life science. Khusus kebutuhan laboratorium Bapak Ibu, baik itu dari akademi maupun juga industrial, produk yang akan kita usung kebanyakan berasal dari industri life science bisnis di Merck sendiri, di mana di sini saya dan juga beberapa tim saya akan mendukung Bapak Ibu untuk kebutuhan laboratorium dan juga penelitiannya. Khusus yang healthcare, biasanya ini lebih ke arah obat-obatan yang sudah memiliki hak paten, dan khusus untuk performance materials, industri ini bergerak di bidang-bidang yang lebih ke arah coating, kemudian juga beberapa electronics yang mendukung teknologi seperti OLED. Nah, untuk bisnis life science sendiri, di sini kita ada 6 brand utama yang biasanya sering didengar oleh Bapak dan Ibu. Yang paling utama kali ini adalah Sigma Aldrich. Jadi ini sudah diakuisisi lebih dari 3 tahun yang lalu, dan produk kita ke arah life science lebih banyak diusung dari brand Sigma Aldrich ini sendiri. Nah, kemudian di kita juga ada Miliki, di Miliki biasanya ini mensupport lab water solution, baik itu di laboratorium maupun juga industri. Kemudian juga brand yang sudah cukup lama, sudah mengakar di PT Merck sendiri ini adalah Supelco Analytical Product. Jadi biasanya memang chemical classics. Kemudian juga kita ada Millipore. Kalau Millipore ini dia sudah lama kita akuisisi under brand of Merck. Dan untuk Millipore sendiri ini produk-produknya lebih ke arah preparation and separations. Kemudian juga kalau untuk media-media bakteri sebagai kisi gitu ya, itu lebih banyak brandnya ke arah Millipore. Nah, yang terakhir ini kita ada SAFC dan Bioreliance, di mana kedua brand ini lebih ke arah mendukung raw materials untuk industri yang dilakukan oleh customer-customer kita. SAFC mungkin banyak yang berhubungan dengan biopharmaceutical juga, dan Bioreliance ini mungkin lebih ke arah biopharmaceutical yang mengarah ke biologics maupun juga vaccines yang rekombinan, di mana nanti mungkin kalau misalnya Bapak dan Ibu ada kebutuhan untuk engineer cells untuk menghasilkan protein tertentu atau antibody, monoklonal antibody, kita memiliki tim yang khusus untuk memberikan paten engineer cell line yang atau stable clone yang bisa memproduksi produk biologics atau ataupun biosimilar yang Bapak Ibu ingin produksi. Baik, without further ado, I will present the portfolio that we can support for the biopharmaceutical. Herein saya ingin memperkenalkan sedikit produk biopharmaceutical atau industri biopharmaceutical apa saja yang bisa kita support. Jadi dimulai dari compound atau industri yang biologics yang menghasilkan compound, ini dia small molecule, biasanya dia lebih kecil dari 1 kilodalton. Ini merupakan salah satu industri biopharmaceutical yang saat ini sedang sangat booming juga. Salah satu produk yang terkenal mungkin Lipitor dari Pfizer ya untuk high cholesterol. Kemudian juga new biopharmaceutical seperti monoklonal antibody MAB, ini dia berupa therapeutic protein. Biasanya untuk ukuran dari monoklonal antibody MAB ini sendiri berkisar dari 25 sampai 800 kilodalton. Dan ini salah satu produk yang dari Merck sendiri di life science-nya sudah luncurkan adalah Keytruda, ya ini berupa checkpoint inhibitor for cancer. Kemudian juga saat ini biopharmaceutical lebih ke arah vaksin, ya terutama untuk SARS-CoV-2, di mana vaksin yang dimanufacture juga bukan hanya berasal dari inactivated gitu ya atau live attenuated vaccines, tapi sudah lebih ke arah mRNA, kemudian juga bukan peptide vaccines, tapi kayak therapeutic atau rekombinan vaccines gitu, sehingga untuk bisnis-bisnis biopharmaceutical ini kita memfokuskan beberapa produk yang ingin di-highlight untuk kebutuhannya. Nah, di bisnis kita sendiri karena kita memiliki 6 brand pilar yang menjadi dasar dari bisnis ini, maka dari mulai end-to-end, dari mulai R&D sampai biopharmaceuticalnya kita dapat dukung Bapak dan Ibu. Nah, secara umum untuk prosesnya sendiri kita akan membagi industri biopharmaceutical ini menjadi 3 kelompok, yaitu di bagian discovery, development, dan manufacturing. Nah, untuk produk-produk yang bisa kita support, mungkin saya compactkan dalam slide saya di slide yang ini dan juga slide-slide berikutnya, di mana produk-produk highlight yang pertama dari untuk stages discovery sendiri mungkin ada beberapa stages ya, biasanya berupa disease target identification, kemudian identify dan isolate target yang sesuai, kemudian candidate design, jadi termasuk PEG glycylation dan sebagainya ini masuk ke sini. Kemudian di karakterisasi ini juga masih dalam discovery step by step, dan ini merupakan produk yang biasanya kita dukung. Jadi untuk discovery sendiri tahapan awal yang paling sangat sederhana itu adalah teknik kultur sel, di mana di sini Sigma Aldrich sendiri sudah sangat populer, sudah sangat tua, berakar gitu untuk mensupport produk-produk yang berhubungan dengan culture cell. Jadi dimulai dari koleksi-koleksi sel lainnya. Jadi di sini kita untuk collection of the cell line itself, we have a variety of brands. For instance, for cell line we have ECAC cell line. Kemudian untuk primary cell we have cell application inks. Kemudian in millipore we usually support for primary and also stem cell. Nah, kemudian untuk teknologi terbaru juga ada induced pluripotent stem cell. Di sini kita biasanya membawakan atau mendeliver sel yang berasal dari ABISC. Di sini induced pluripotent stem cell yang sudah yang didistribusikan di sini merupakan stabil dari iPSC yang berasal dari donor-donor dan dimodifikasi dari lab-lab di berbagai belahan dunia. Nah, selain sel line, kita juga memiliki sera, media, antibiotik, growth factor, freezing medium, the cell disattachment factor yang sangat mendukung proses biofarmaceutical Bapak dan Ibu. Di sini mungkin saya highlight saja produknya, jadi dimulai dari yang FBS. Kalau FBS sendiri, Sigma Aldrich sudah mendeliver FBS yang memiliki GMP certified, kemudian juga untuk proses biofarmaceutical sendiri karena memang sangat rentan ya terhadap virus, kemudian juga other kontaminan yang bisa merubah atau mempengaruhi end product dari biologik itu sendiri. Biasanya sangat rentan untuk virus, sehingga untuk FBS yang kita sarankan untuk biopharmaceutical itu adalah yang sudah gamma irradiated. Jadi kalau sudah gamma irradiated biasanya dia sudah eliminates virus-virus yang biasanya menginfeksi bovine itu sendiri. Jadi ada yang GMP manufacture dan gamma irradiated khusus untuk industri biopharmaceutical. Nah, kemudian juga karena industri biopharmaceutical ini nantinya akan dipakai oleh manusia, tentunya di Indonesia sangat concern dengan isu halal. Nah, untuk cell dissociation factornya sendiri biasanya menggunakan trypsin, di mana trypsin tersebut mengandung kandungan porsin, sehingga untuk tahapan awalnya kita sarankan tidak menggunakan tripsin gitu, pencacah sel yang berasal dari porsin. Di sini produk kita sendiri ada yang disebut sebagai tripzean. Jadi tripzean ini merupakan tripsin yang diambil dari gene pankreas bovine yang diekspresikan di jagung gitu. Untuk hasil disattachment-nya sendiri bila dibandingkan dengan kompetitor, tripsin memiliki kekuatan cell disattachment atau enzim yang lebih kuat dibandingkan dengan other competitor, sehingga sangat convenience untuk kebutuhan biopharmaceutical Bapak dan Ibu. Nah, terakhir untuk cell freezing mediumnya sendiri, tentunya proses freezing merupakan hal yang paling krusial ya untuk di industri biopharmaceutical. Nah, di sini kita biasanya mensarankan cryostore. Cryostore ini adalah jenis cryopreservasi yang sudah ready to use, sehingga nanti pada perlu mix lagi ya serum, kemudian medianya untuk memfreezing selnya. Di cryostore ini sendiri dia sudah ready to use, dan rate of survival sel ketika dibangunkan jauh lebih tinggi bila dibandingkan dengan kompetitor atau produk lainnya. Nah, untuk produk-produk ini juga media, kemudian juga cell dissociation factor, kemudian cell freezing mediumnya juga ada yang serum free. Jadi kalau misalnya Bapak dan Ibu ingin melakukan ingin melakukan production biopharmaceutical product yang bebas dari animal komponen, dari awal kita sebenarnya sudah ada beberapa produk yang mendukung serum free environment untuk perkembangan kultur pertama atau stabil sel yang ingin Bapak Ibu kembangkan di laboratorium. Nah, selanjutnya tidak kalah penting juga adalah filtranya. Di sini kita bisa bawa millipore, di sini ada Stericup, kemudian juga beberapa syringe filtrasi yang biasanya digunakan dalam praktik kultur sel untuk menghindari kontaminasi. Terakhir, untuk labware alliance-nya sendiri, dari kita memiliki brand partner premium dari Corning, kemudian juga dari brand sendiri, di mana kedua brand ini bisa mendukung konsistensi hasil dan juga pyrogenik atau endotoxin free untuk alat-alat kultur atau consumable yang biasa Bapak Ibu gunakan di laboratorium masing-masing. Nah, selanjutnya biasanya selain dimulai in vitro-nya dari kultur sel, mungkin juga untuk studi tertentu akan ada asai yang lebih advance berupa 3D cell culture. Jadi bukan sekarang ini yang lagi happening bukan hanya cell culture yang biasa yang ditempelkan di plastik, tapi juga ada yang 3D cell culture. Nah, kita juga sangat support untuk kebutuhan Bapak dan Ibu mengenai 3D cell culture, di mana seperti yang kita ketahui 3D cell culture membuat asai-asai atau studi yang Bapak Ibu lakukan untuk sel, misalnya untuk penyerapan obat atau mungkin untuk bahkan untuk menghasilkan organ seperti kolon atau alveolus gitu ya untuk SARS-CoV-2. Nah, ini biasanya didukung dengan 3D cell culture technology. Nah, di kita ada 2 produk unggulan yang biasa kita sering sarankan gitu ya untuk studi disease modeling. Yang tertarik untuk 3D, yang pertama hydrogel. Jadi kalau di kita itu ada hydrogel yang sering digunakan itu adalah yang max gel sama yang ECM gel. Jadi berdasarkan dari testimoni beberapa customer yang sudah menggunakan ini dengan menggunakan Maxit dan ICM gel ini dapat meningkatkan proliferasi dari selnya sendiri, juga hasilnya sangat konsisten dan sangat bagus untuk studi disease modeling yang 3D. Selain hydrogel, kita juga memiliki 3D organoids portfolio di mana Bapak dan Ibu bisa memimik organ gitu, jadi mengembangkan sel yang biasanya berasal dari induced pluripotent stem cell untuk dikembangkan menjadi organ. Jadi kalau yang ini adalah gambar 3D organoid berupa kolon, sehingga ketika Bapak dan Ibu berhasil mengembangkannya, kalau untuk studi absorption obat gitu, dia lebih memimik in vivo dibandingkan dengan hanya 2D cell culture teknik yang biasa dikembangkan di lab. Nah, selanjutnya dalam proses discovery juga nanti akan ada teknik di mana kita harus memodify selnya atau ya gene editing. Di sini saya ingin highlight gene editing kita berupa CRISPR di Merck sendiri. CRISPR gene editing memiliki portfolio yang cukup lengkap. Saya bisa infokan seperti itu. Contohnya seperti gRNA atau guide RNA, kemudian Cas9. Di guide RNA sendiri kita memiliki guide RNA yang hanya berupa protein maupun juga guide RNA yang sudah melekat pada plasmid. Selain itu kita juga memiliki sintetik gRNA atau guide RNA yang memiliki efektivitas lebih tinggi dengan gRNA yang biasa. Nah, untuk Cas9 sendiri, untuk enzimnya sendiri gitu, Cas9-nya sendiri juga sudah ada yang tercantum di dalam plasmid maupun juga sudah dalam vektor berupa lentiviral. Nah, produk unggulan dari Cas9 kita ada yang SP Cas9 GFP protein. Di sini adalah Cas9 yang sudah track melalui GFP, sehingga nanti ketika si Cas9 ini melekat dengan target binding yang berada di sel kita, maka dia akan menghasilkan fluorescence bahkan sebelum transfeksi, karena kalau biasanya menunggu transfeksi membutuhkan waktu yang cukup lama gitu ya, beberapa hari. Nah, ketika si Cas9 ini sudah bertemu dengan meminimalisir waktu kita untuk melihat apakah transfeksi kita ini berhasil apa enggak. Kemudian juga kita memilih Bapak dan
It seems like there's a trouble from the presenter. We're waiting for presenters. We have to wrap up the presentation because there's 1 minute left.
Sorry, how long is the presentation for this, Mark?
1 minute left.
1 minute left. Okay.
Oh, I'm sorry. 10 minutes left.
Yeah, so sorry about the connection. I'm going to reshare my screen again. Share screen, yeah. Okay, where were we? Okay. So I'm talking about the CRISPR before. Jadi kita memiliki custom oligos. Selanjutnya untuk produk highlight CRISPR ini sendiri kita memiliki all-in-one CRISPR plasmid, di mana Bapak dan Ibu sudah memiliki vektor plasmid yang di dalamnya sudah terdapat Cas9 dan juga guide RNA. Jadi ini sudah all-in-one, Bapak dan Ibu tidak perlu lagi khawatir untuk satu persatu masukin kemudian mix. Nah, di CRISPR ini sendiri apabila sudah oke nih bagian gRNA dan Cas9-nya, nanti mungkin perlu melakukan transfeksi gitu ya, transduksi. Nah, di sini saya menyarankan Extreme Gene 36 di sini. Mana Extreme Gene 36 ini merupakan reagen transfection yang khusus untuk melakukan plasmid DNA dan sangat cocok untuk aplikasi CRISPR atau Cas9 yang akan dilakukan oleh Bapak dan Ibu. Highlight produk ini sendiri adalah performance-nya yang bagus sehingga memiliki higher transfeksi dibandingkan dengan produk sejenisnya, kemudian juga low cytotoxicity, dan yang paling penting itu dia free dari animal derived component, sehingga apabila Bapak dan Ibu ingin melanjutkan ke biopharmaceutical downstream, dia jadi lebih aman untuk menggunakan Extreme Gene 360 ini. Nah, lanjut masih ke arah discovery yang nyerempet-nyerempet sedikit ke development. Setelah melakukan teknik berupa gene editing, tentunya kita perlu menganalisis. Nah, jadi ini ada beberapa produk analisis yang bisa kita dukung untuk kebutuhan Bapak dan Ibu. Jadi mungkin yang paling gampang kita bisa melakukan immunocytochemistry, yaitu berupa antibody ya, fluorescent antibody. Nah, antibody ini sendiri kita juga ada edisi yang, jadi kalau untuk Zoomec ini sendiri dia merupakan antibody yang bisa tahan di room temperatur gitu, sangat bagus. Nah, kemudian juga memiliki sangat banyak varian gitu di portfolio produknya sendiri. Dan ini merupakan rekombinan dari interspecies gitu ya, tapi bisa men-tailor atau menyesuaikan dengan kebutuhan antibody yang ingin Bapak Ibu gunakan di lab. Nah, selain itu kita juga memiliki live cell imaging reagents gitu. Jadi untuk beberapa reagen yang mendetek perubahan variabel berupa warna, kita memiliki beberapa kit reagensnya. Kemudian juga untuk analisis proteinnya atau antigennya kita bisa menggunakan SMC X-Pro dan juga Milliplex Kits untuk produk-produk Luminex. Jadi kita juga mendeliver produk-produk Luminex. Nah, selain antigen, di sini juga ada sel asik onyx, di mana ini merupakan live imaging dari sel yang biasanya kita gunakan untuk studi disease modeling. Kemudian juga ada beberapa cellular assay dan juga Scepter 3.0. Nah, cellular assay ini berupa kit ready to use, yang di sini MAK kit ready to use khusus untuk beberapa analit yang biasanya digunakan Bapak dan Ibu untuk penyakit tertentu. Jadi misalnya Bapak dan Ibu tertarik untuk sedang melakukan penelitian onkologi. Nah, kami akan dengan senang hati memberikan brosur MAK Kits karena biasanya dalam brosur ini akan berisikan komprehensif analit apa yang biasanya digunakan untuk onkologi gitu, maupun dan juga penyakit lainnya. Jadi di sini hanya gambarannya kecil saja, cuman kita memiliki beberapa penyakit yang sering diteliti oleh peneliti secara globally dan juga di Indonesia. Kemudian bahkan ada juga analit khusus untuk drug discovery ya, seperti NAHDS atau mungkin untuk melihat senyawa metabolit tertentu gitu, bisa dengan menggunakan ingin lebih ke arah protein gitu ya, misalnya melakukan penelitian berupa monoklonal antibody. Di sini memiliki beberapa workflow berupa ekstraksi dan purifikasi, konsentrasi, sampai analisis western blot-nya kita memiliki produknya. Untuk ekstraksi, produk yang saya highlight di sini Celitic kits, di mana Celitic ini ekstraksinya lebih aman, lebih tanpa merusak selnya, sehingga nanti hasilnya akan lebih. Kemudian untuk purifikasinya kita juga memiliki produk concentrate protein. Nah, mungkin yang saya ingin tampilkan di sini adalah Emicon state cell, di mana di sini kita bisa mengkonsentrasikan protein dalam jumlah yang lebih banyak. Dan yang terakhir untuk analisisnya itu ada western blotting ya. Kalau untuk western blotting bloating, tapi kita punya alat berupa Snap ID tool. Jadi di sini biasanya Western blotting kegiatan Bapak dan Ibu bisa sampai 4 sampai 24 jam, ini bisa disingkat saja dalam waktu 30 menit gitu. Karena teknologinya bisa menggunakan vakum. Nah, terakhir untuk gel pre-cake, jadi kita ada true page di mana dalam untuk true page ini sendiri Bapak dan Ibu tidak perlu lagi membuat gel pre-cake sendiri, sudah ready to use dan diklaim 10 kali lebih stronger atau lebih kuat dibandingkan pre-cast gel yang tersedia di market. Nah, selanjutnya juga mungkin ini highlight beberapa teknologi saja gitu yang kita punya. Kita juga punya automated 2D electrophoresis di mana di sini apabila menggunakan SDS biasa ya ini mungkin cuma bandnya hanya segini, dengan menggunakan automated 2D electrophoresis Bapak dan Ibu bisa melakukan analisis SDSP secara otomatis. Jadi tidak perlu tambah-tambah reagennya secara manual tapi langsung menggunakan alat. Dan yang terpenting dari sini adalah analisis atau band separation yang biasanya di SDS page itu hanya dalam turun ke bawah gitu dia molecular weight. Kalau di sini dia sudah menggunakan PA dan molecular weight. Jadi seandainya Bapak dan Ibu memiliki protein yang biasanya berada dalam satu band yang sama dengan menggunakan PA atau kinetik, nanti dia akan ke arah samping dulu pemisahan separationnya lalu ke arah bawah sehingga nanti untuk protein-protein yang jumlahnya sedikit atau protein yang memiliki band bersama, nanti separationnya akan kelihatan lebih jelas dengan menggunakan automated 2D electrophoresis yang kita punya. Kemudian juga untuk analisis antigen tadi sudah saya sebutkan, di sini kita bisa ada SMC X Pro, ini merupakan ultra sensitivity di mana bisa mendetek sensitivitas antigen sampai dengan femogram. Kemudian untuk Luminex platformnya sendiri, gitu dia menggunakan Luminex di Taylor di support Luminex dan SMC X Pro. Nah, yang terakhir untuk teknologi yang ingin detection yang ingin saya
It seems like there's a problem with Mrs. Sri Hayuni.
Plate-nya iya, sudah waktunya habis ya Mbak ya?
No, you still have 3 minutes left.
Okay,
the connection is kind of, yes, please proceed.
Okay, thank you. Okay, so the last part I think I was just summarize. Jadi untuk manufacturingnya sendiri kita juga memiliki banyak produk yang bisa mensupport. Jadi biasanya kalau untuk manufacturing sudah masuk ke tim Bioreliance, tapi untuk beberapa reagen kimianya, misalnya untuk vaksin maupun juga biologics lainnya gitu ya, biosimilar lainnya membutuhkan preservasi, kemudian surfactant, adjuvant, antibiotik, stabilizer, dan acidity regulators. Ini merupakan portfolio produk yang kita miliki. Silahkan bila Bapak dan Ibu tertarik bisa mendiskusikannya dengan saya sendiri ataupun tim Merck area Bapak dan Ibu untuk mendapatkan konsultasi yang lebih lanjut mengenai preservasi dan juga chemicals lainnya yang bisa mendukung produk akhir biologics Bapak Ibu kembangkan di laboratorium. Nah, mungkin sebagai highlighting saya sudah menyebutkan bahwa kita merupakan one step solution. Nah, di sini saya akan sebutkan bahwa produk Merck sendiri itu dapat dilihat dari MQ level. Jadi ini adalah quality segment dari produk kita. Biasanya di setiap produk ketika Bapak Ibu dan login ke Sigma Aldrich, di situ akan dijelaskan berapa quality levelnya. Jadi MQ 100 sampai MQ 600. Nah, MQ100 adalah komponen yang paling komponen yang paling rendah gitu karena dia hanya melakukan pengecekan berupa basic quality gitu ya, tapi tetap menggunakan manufacturernya masih dengan menggunakan ISO 901 gitu, 9001. Nah, ini mungkin cocok tapi ketika Bapak dan Ibu sudah mulai melanjut ke arah biopharmaceutical atau manufacturingnya, mungkin bisa mengecek produk yang berlabel MQ 500 dan MQ 600. Jadi misalnya Bapak dan Ibu mau menggunakan sodium fosfat gitu. Sodium fosfat ada yang MQ 100, tapi kalau sodium fosfatnya sudah mau ke arah sarankan menggunakan sodium fosfat yang menggunakan MK600. Jadi kita memang support memiliki wide variety dari reagent chemicals berdasarkan quality segmennya sendiri. Nah, terakhir apabila Bapak dan Ibu memiliki ingin cost atau menghemat budget gitu, kita juga memiliki kemampuan untuk bulk packaging. Jadi di sini Bapak dan Ibu bisa membeli packaging atau pre-pack yang lebih besar sehingga nanti cost untuk bahan-bahan kimia yang Bapak Ibu gunakan di biopharmaceutical tersebut di-cut dengan sangat efisien. Silakan menghubungi ke untuk mendapatkan penawaran produk untuk Bapak dan Ibu. Baik, ini saya terakhir hanya masuk ke quiz time gitu. Nah, untuk quiz time sendiri ini akan silakan menjawab pertanyaan tercepat dan mengirimkan emailnya ke saya dengan format nama, institusi, kemudian jawaban gitu. Nah, untuk pertanyaannya sendiri adalah sebutkan salah satu saja teknologi detection yang dimiliki Merck yang tadi sebutkan, walaupun dengan presentasi yang terburu-buru karena limitation waktunya. Jadi silakan Bapak dan Ibu bila memiliki jawaban untuk pertanyaan tersebut silakan kirim ke email saya. Nanti di situ untuk 5 orang tercepat akan saya berikan gimmick atau hadiah dari tim kita. Baik, saya rasa itu saja presentasi saya. That's all my presentation. So sorry about rushing the time. I have a technical problem, etc. So yes, thank you very much for your attention and hope to see you again.
Okay, thank you, Mrs. Sri Hayuni, for the presentation. Now, ladies and gentlemen, before continuing to the next agenda session with invited speakers and parallel session, we will have a break for an hour, and we will start the session at 1:30 p.m. Jakarta time. And at the end of the event, there would be announcement for 6 best speakers. So stay tuned and see you after the break. Thank you. Ladies and gentlemen, welcome back to ISIBIO 2021. In the following hours, we will have the first panel session that are divided into breakout room. But before that, there will be the session with the invited speakers. So for room 1, there will be Dr. Puspita Lisdianti. At room 2, there will be Dr. Pierre Muhammad Abdul. And at room 3, there will be Professor Yong Soo Kim. Parallel session 1 will start at 2 p.m., parallel session 2 at 2:40 p.m., parallel session 3 at 4 p.m. Jakarta time. And for all presentation of parallel session will be conducted according to the rundown given by the committee. And to enter the room, you could choose the square icon, breakout rooms, and on the toolbar right below and click join of the room that you choose. And now you may enter the breakout room. Thank you.
Good afternoon. Good afternoon, Professor Yong Soo Kim. Good afternoon, moderators. Good afternoon, participants.
Sorry, I was in different room.
Okay, it's okay, Prof. So let me try and introduce moderator first before we get started.
Okay.
So let me share screen for a moment. So here I will be introducing 3 moderators in this afternoon. Started from the first sessions that will also be followed by the presentations from Professor Yong Soo Kim. The first moderator is Dr. Renat Anis H. Mahsuna. Dr. Anis received her doctoral degree from Saarland University, Saarbrücken, Germany in 2007. She is now working at Biotech Center BPPT that is now becoming BRIN, Indonesia. Her research interest is in the area of analytical chemistry and purifications and bioprocess engineering. So let me follow for the next the next moderator for this session 2. Moderators in session 2 will be Dr. Fina Amreta Laksmi. Dr. Fina received her doctoral degree from Kagoshima University, Japan in 2020. She is now working at the Center for Biotechnology, National Research and Innovation Agency, Republic of Indonesia. We know it as BRIN. Her research interest is in the microbial-based enzymes, enzymes for production of rare sugars, enzymes for food, and so on and so on. So the last moderators is Dr. Hani Susanti. Dr. Hani Susanti received her doctoral degree from University of Tsukuba, Japan in 2020. She is also now working at the Center for Biotechnology, BRIN. Her research interest in the area of biotechnology for aquaculture, microalgae, biotechnology for aquaculture and microalgae culture collections. That's all our introductions to our moderators in the session. So I will be giving the first sessions to Dr. Anis to be a host and to moderate the sessions. Dr. Anis, time is yours. Thank you.
Thank you, Panaro, for the introduction. Ladies and gentlemen, good afternoon. Assalamualaikum warahmatullahi wabarakatuh. Can you hear my son?
Oh, yes, I can hear you.
Okay, thank you. Okay. Yeah. In this session, we will have Professor Yong Soo Kim. Is the spelling right?
Oh yeah,
Professor
this
Kim,
is correct.
okay.
Right, right.
Okay.
Yeah.
Okay. So please, Mr. Naro, please show his profile. Okay.
Oh, this is.
Yeah, Professor Dr. Yong So Kim is from University of Hawaii, and he got his PhD in physiology in University of California, Davis in 1988, and
Long time ago.
yeah, he got his master in animal science in University of California, and then his bachelor is also in animal science in Seoul National University, Seoul, Korea. The work experience of Professor Kim is since 2013 until now. He is professor in Department of Human Nutrition, Food and Animal Sciences in University of Hawaii, USA. And then, before this, he was affiliated in graduate faculty in nutritional science, also in the same university.
Yes.
Yeah. Yes. Okay. And then, he started his work in the same university in 1991 as assistant professor.
Right.
So, and his research interest in conducting researches in the area of animal physiology, with research focus being on muscle biology. So,
great. Yeah.
Okay. Yeah.
Okay. Thank you.
Thank you. And and in this opportunity, you will have a presentation entitled "Production of Recombinant Myostatin Propeptide, an inhibitor of myostatin in Ecology." Okay, so you will have 20 minutes for presentation.
Okay.
And then 10 minutes for answer and question.
So I'll share my screen then, right?
Okay.
So,
okay. Please, Professor Kim.
Okay. Thank you. Thank you for the introduction. Hello, my name is Yong-Soo Kim, and then let me share my slide. Where is it? There it is. Okay. Share, and then I think I have to go to the first slide. I don't know if I can do the. Okay, let me try slideshow. So let me see what I can do it or not. Just a second, the slideshow from beginning. Okay, so I can do that actually. Okay, so I'm gonna, this afternoon, I'm gonna talk about the production of recombinant myostatin propeptide, an inhibitor of myostatin E. coli. And okay, so this is just my table of content. I'm gonna cover first some background on myostatin, and then talk about the examination of E. coli expression of vector system for myostatin propeptide production. By the way, can you hear me clearly? Or?
We can see your presentation, and we
How
can
about the voice? My voice is clear or not?
The voice is okay. It's clear.
Yeah. Oh, okay, good. Yeah, okay. So then next, I'm gonna talk about the production of various size of myostatin propeptide. So it's truncated form of myostatin propeptide, and then look at the effect of administration of myostatin propeptide on muscle growth and performance. And finally, briefly talk about the production of immunoglobulin FC domain fused myostatin propeptide. Just a second. Okay, so myostatin is a member of the TGF-beta superfamily growth and differentiation proteins, and then it was first identified in 1997 by S.J. Lee's lab at Johns Hopkins University. And then this picture basically shows from their publication in Nature 1997, and then this upper pictures from a wild type animal, and then this lower picture from myostatin knockout mice. And then as you see it, there's a more than 200% increase of muscle mass when myostatin was knocked out. And, you know, this clearly shows that the myostatin is a negative regulator of muscle growth and development. And then they also reported that the myostatin is mostly expressed in skeletal muscles. And then following studies also shows that the naturally occurring non-functional mutation of myostatin gene cause muscle mass increase in many animal species, including, you know, cow like a Belgian blue. This is myostatin naturally occurring myostatin mutation, and also in dog and sheep, also in humans, myostatin mutation increasing the muscle mass. And then finally, subsequently, it is well established that myostatin is a potent negative regulator of skeletal muscle growth. And then also it kind of suggesting that myostatin suppression can potential strategy to improve muscle growth in meat animals, actually. Just some basic biochemical characteristic of myostatin. Myostatin produced as a pre-propeptide having 375 amino acid. And there are three domains. Here, the red one is that signal sequence, and then in the middle, this is a myostatin propeptide domain, also called prodomain. And then the C-terminal is active form of these myostatin domains. And then, you know, one of the characteristics is that this myostatin is evolutionarily well preserved. So there are only 1 to 3 amino acid difference, you know, among mammalian and avian species. And then this, you know, pre-propeptide, when they enter into the endoplasmic reticulum, this signal sequence cleaved. And then this propeptide also cleaved from myostatin. But after, you know, cleavage, it's not completely separated. It's still binding to this myostatin, you know, dimer. Then I think, so this, you know, diagram shows that just prodomain complex with the myostatin dimer. And then this prodomain suppress myostatin activity. And then this complex called latent forms of myostatin. So this myostatin to be activated, this prodomain is cleaved by some enzyme called BMP1 toluene metalloprotease. And then it just cleave this site amino acid, aspartic acid 75 position. And then this one completely separated this prodomain. So this now binding to its receptor and then do its physiological activity. So now, you know, this shows that the myostatin propeptide is a strong inhibitor of myostatin. So my lab and then my Dr. Jin's lab in Korea collaborated to produce this myostatin propeptide in Ecology. Then our first, you know, study in this myostatin production was that first we tried to examine different Ecology expression, you know, vectors to find out the optimum vector for this myostatin propeptide expression. So we chose 3 different vectors. One is PET32A having TRX tag, and then PML-C2X having MVP tag, and then PET43.1 having news tags. And then we just used a flatfish myostatin pro sequence in making a just the expression, you know, construct, and then basically transform the E. coli and express the proteins. And then this is like a SS page result of this expression. And then the T indicates that the total cell lysate, and S is the supernatant after centrifugation of this lysate, and N is the insoluble pellet. And then if you see like expression with PET32A, PET43.1, most of this expressed protein is in insoluble pellet. And here, PET32A and PET43.1. But if you look at the PMAL vector, the expressed protein is mostly insoluble fraction. So we found that this myostatin propeptide can be produced using this PMAL vector in Ecology. And then we purified using amylose affinity chromatography of this PMAL vector expressed this myostatin propeptide. So we have some purified this expressed proteins. And by the way, this one has a maltose binding protein fusion. So this flatfish myostatin Pro fused this MVP in the N-terminal side. And then we measure the activity, and then using called PGR, CAGA, luciferase reporter gene assays, and then this using the cell culture system. And then when the myostatin is being added, they induce luciferase activity, and so to show the luminescence. So you see the big luminescence here, and then we combining the myostatin plus our proteins, and it's completely suppressed myostatin activity, indicating this protein really suppressed myostatin. And then this amount of the protein decreasing, and then slowly we see the increase of the luminescence. So we found that the PMAL vector is a good vector for soluble expression, bioactive, and soluble expression of this myostatin propeptide. Okay, next one is that we just used this PMAL expression vector, and then we produced a porcine myostatin pro. But the difference here is that we have two forms. One is that the wild type of myostatin pro, and the other one is that like a mutant type. This mutant type is basically, as I previously discussed, for this myostatin being activated, this prodomain need to be cleaved at the 75 aspartic acid. So we induce the mutation of this aspartic acid into alanine. So we have two forms, and then we just express the proteins, and then there's a total lysate. This is supernatant. So both the mutant wild type forms and mutant form expressed in soluble forms. And then we did purification, amylose affinity chromatography. It is purified, but still there are a lot of contaminant in here in this process. So we did further purification. We like anion exchange chromatography. So I think here, this is much more pure than amylose affinity only. And then next one is that because we have a mutated form of this prodomain, we need to check whether the mutation was correct. So we basically incubated, this the propeptide with the BMP1 because BMP1 cleave this prodomain. So this is SSP analysis result, and wild type is just after incubating with the BMP1, it's separated. So this is a maltose binding protein, and this is a myostatin pro, peptide in here. But mutant one was not cleaved by BMP1, so it's just showing that it is correctly mutated. And then this is just a Western blot analysis, and then it also shows that it's just the same result. And then we also measure activity, and then this is percent inhibition of myostatin activity, and then this is ligand, this is our ligand, and then we also use the commercial myostatin pro as a positive control. So here, you can see that the myostatin activity is suppressed in the high concentration, and then the ligand concentration is decreasing. Just suppression, disappear as expected. So, and then also, from this graph, you can see that the potency of myostatin suppression, this propeptide is comparable to the commercial myostatin pro. So. Okay, then after this, I think what we try to do is that we try to find out the minimum size of this myostatin fragment having the full potency of myostatin inhibition. So using the same flatfish myostatin propeptide, we have 6 different forms of truncation, like amino acid sequence is 23 to 65. This is a full sequence. then 45-100, 45-90, and then so we have different truncated forms of this myostatin, and then expressed and purified. So this one shows the purification, and then same way we measure the bioactivity, and then it shows that until number 5. Number 5 is that like amino acid sequence 45-80. So up to here, it shows that the inhibition of myostatin activity. But when you use the 55-100, 65-100, it doesn't show any myostatin inhibitory activity. So from this result, it indicates that propeptide sequence from 45 to 55 probably play an important role in on the suppression of myostatin activity by this propeptide. To confirm this, we did the pull-down assay, and then we found out the 45-80 binding to myostatin in pull-down assay, but 55-100 didn't bind any myostatin here, so supporting this the result. And then we try to further confirm using the just a synthesized propeptide, and then this is the result. So peptide like a 45-54, 45-65, 45-70, 45-80, up to 5, it suppressed the myostatin activity. But peptide 80 to 100, it didn't show any inhibitory activity. So it's pretty similar result with this our fragment produced in Ecology. So once again, it shows that the sequence from the 45 to 55 is very, very important. The next one, this one shows that some domains of this prodomain. Sorry that I didn't have a fish here, but it's pretty similar sequence like that. And then this 45 or 55 is in something called this alpha-1 helix domain. So it kind of suggests that this alpha-1 helix domain is kind of important in this propeptide suppressing myostatin activity. So this one play a role, just prodomain binding to myostatin. Okay, now then I think we want to actually administer this propeptide to animals in mice, and then see whether they have any effect on muscle mass and function. So, but we used MVP fused myostatin pro amino acid sequence from 45-70. Actually, this one is selected from 16 truncated forms of this myostatin propeptide, and then this shows that good inhibitory activity. So we used this one in administration to mice. Before administration, we examined some binding activity, and at the same time we examined whether this one really will inhibit myostatin signaling. And then SMAD phosphorylation is the earlier signaling process for myostatin signaling. And then what we did was that we use some cells, HEP-G2 cells, and this one having myostatin the receptor. So when we using these cells, so when you added these cells, this myostatin, there's a significant increase of this SMAD2 phosphorylation. And when you add myostatin and our protein together, then this phosphorylation was suppressed. And then same thing with the SMAD3 and 2 and 3. And so this result clearly shows that this myostatin pro 45-70 suppressing myostatin signaling. So we administer these proteins to mice for 2 weeks, and this is the result of this administration. And we look at the muscle weight, body weight, and muscle weight, and then grip strength, and then swimming time. And then first, after 7 days after administration, there's some increase of this body weight, but after at 40 days, 14 days, 2 weeks after administration, there's no difference between control and treatment. And this is arm muscle after 14 days. It's just a little increase. Also leg muscle a little bit increase, and then bone weight basically no change. And then if we look at the grip strength, 7 days there is a significant increase in grip strength, but 14 days after administration, it just disappeared. And then swimming time was also 7 days. There's a significant improvement in swimming time, but 14 days, even though statistically significant, the increase is kind of decreased. So we guessed probably the reason after 14 days the effect slowly disappear is probably due to immunogenic effect of these proteins, and then this antibodies probably are neutralizing the proteins. So it's prevent the suppression of myostatin activity. So what we did was that we measure titer, and then this is the result of this titer measurement. And then 7 days there was a antibody produced against these proteins. For 14 days, so there's a significant increase of this antibody. So it's sort of supporting that, supporting our hypothesis, probably neutralizing this protein. So no further myostatin inhibition. And then, so we try to confirm this by administering, peptide having no maltose binding protein attached, and then the size we used was actually 45, 65, and then this peptide is not immunogenic, so we used this peptide 45 to 65, and then the result shows that 14 days just more increase of this body weight, and then 14 also more increase of the arm muscle and leg muscle, and then if you look at the grip strength, compared to the 7 days, 14 days is much more increased, and then also swimming time is just also increased 14 days. So that this result sort of supporting our hypothesis, the immunogenic response. Okay, and then next one is that I think I'm gonna this is pretty similar to truncated form of flatfish myostatin peptide. So I'm going to kind of skip a lot of this result. I don't have much time, I guess.
Sorry, you have only 2 minutes.
2 minutes left. Okay.
Okay.
Okay. So I can finish that, I think.
Okay,
so,
you can.
Yeah. In this study, we have 5 different truncated forms, and then this basically shows that purified, and then we measure their yield. And then, as you expect, when the size is smaller, the yield is low. I'm sorry, size is bigger, yield is low, but the size is smaller, the yield is increasing. That's why we tried to, one of the reasons, try to find out this, small size of. Then the other one is that, we did also this bioactivity, and then smaller, size, the potency is decreasing like that. And then, but we also confirm this binding to myostatin using pull down assay. And the other thing is that important, different point in here is that we try to look at the effect of MVP. So we try to remove the MVP from this propeptide. So, and then there is a this factor XA recognition sequence between the MVP and then this Pro-42-175. So we cleaved this MVP using the factor XA, this protease, and then this is a proteins, and this is after incubation with the factor XA, and then this is MVP separated, and then Pro-42-175 is in here, and then we purify and we measure the activity basically here. Now this is an MVP fused 42-175. The number, this one is MVP removed 42-175. The potency is almost 20-fold decreased. So indicating that MVP is a really, really important role, just enhancing this myostatin inhibitory activity. We did also similar the experiment to confirm that result. This time much smaller one, 4298. Then they have with the MVP, it shows the inhibitor activity, but when the MVP is removed, just completely flat, no myostatin inhibitory activity. So it shows that the MVP is really enhancing the myostatin inhibitor activity. Okay, now basically this is summary. I'm gonna just quick read it, and then this is summary. I think that's you can see that it's already discussed. Maybe I'll just start here. I put too many slides, probably for 20 minutes of presentation. Okay, thank you.
Thank you, Professor Kim. Any question? Please, if someone has question, please raise hand or write in the chat box, please. I think there is no question, but I have a small question, Prof. Kim.
Yes.
This research, this inhibition of myostatin is the product similar with this is available commercial?
Oh, this propeptide? Yes, propeptide is commercially available, but the reason we try to produce in this Ecology is that it's very expensive. So for the animal injection study, I think you need a huge amount of funding. But so that's why we try to, because Ecology expression is relatively easy compared to the eukaryotic expression. So we use this, we try to produce in Ecology, and then we are able to produce large amount. Yeah, quite large amount actually. Yeah.
Okay, you hope if the research is finished and can be produced commercially, and you hope it will be cheaper than commercial.
Oh yes.
Okay.
Yeah.
Okay. Thank you very much. Yeah. Is any question? I think there is no question. Thank you very much, Prof. Kim, for very interesting presentation. And we give applause for Prof. Kim. Okay, thank you very much.
Okay, thank you very much.
Thank you. Okay,
thank you.
And we will now have another parallel session.
Yes.
So
thank you. Okay, thank you. Let me see. How can I remove this? Stop share. Okay, great. Okay, now
thank you.
Okay, thank you.
This is a great presentation, interesting presentation. Thank you very much. So we will have a parallel session with 3 presenters. The first presenters is Miss Dwi Wulandari, and the second is Mr. Andri Wardiana, and then Dr. Adi Santoso. So, organizing committee,
Bismillahirrahmanirrahim.
We can start the presentation.
Thank you very much.
Please show the slides.
Today I will present my research about the development of human papilloma virus detection kit in Indonesia, optimization process of lipid kit prototype. My name is Dwi Wulandari from Research Center for Biotechnology, BRIN, and my teams are Pak Sukman Nuswantara, Ibu Alkirta Atikana, Ibu Ratih Asmananingrum, and Ibu Puspita Listianti. For introduction, cervical cancer is the second most common and leading cause of death in women in Indonesia. Cervical cancer related with SPV persistent infection. The Indonesian Institute of Sciences, now BRIN, has been developing a prototype for detection of SPV, namely KIT-LIPI, that able to detect 25 types of SPV, causes of high risk and probably high risk. The detection kit based on the multiplex PCR methods. The reaction is simultaneous in one tube and have been tested for swab and urine samples. KIT-LIPI compared with COBAS as a standard and showed approximately 10% of negative results using COBAS were detected positive with KIT-LIPI. It indicated a false positive in the KIT-LIPI. thus need improvement on its PCR formulation. The study aims to optimize the PCR formula for the kit lipid to improve the kit specificity and sensitivity, as well as to minimize primer damage. Material and methods. We use for DNA sample from cervical swab with LIPI positive and cobas negative, and one sample with LIPI positive and cobas positive. The ID samples are here, and the sample stored at -20 degrees Celsius until use. We have for activity, the first is optimization of annealing temperature. We use 65 until 72 degrees Celsius. And the second is the variation polymerase enzyme. We use KOD, DreamTech, and NGY mix and NGY premix. And then we use we make variation in primer concentration to get a new formula. We use 0.02 until 10 pmol, and we make dilution for the old formula from 2 times until 10 times. And the last, we make a confirmation by sequencing a gold standard. DNA amplification will result about 200 base pairs, and we use 2% of agarose gel, and the primer is SPV16 in-house, and the reagent is here. We use NC Biotech to master mix, and the reaction is here. For result and discussion, for the first activity, optimization of annealing temperature, we note that the success of specific amplification depends on the rate of each primer anneal to their targets, and the rate at which anneal primer are extended along the desired sequence during the early, middle, and late cycles of the amplification. So we must know the optimum annealing temperature, and for the kit lipid was 68 degrees. And for the second activity, we make a variation concentration of primer to get a new formula. We use 0.02 until 10 pmol, and we get a minimum primer concentration that still get a good result. A good band, clear band is a concentration 0.08 pmol. But we still get a primer dimer in all concentration. The presence of more than one primer pair in the multiplex PCR increases the chance of the formulation of primer dimer. Ideally, the primer should have nearly identical optimum annealing temperature, prime length, 18 to 30 base pairs or more, and GC content of 35 to 60%, and should not display significant homology either internally or to one another. But we have designed the primer as this recommendation, but the result is still we get the dimer primer. For this variation polymerase enzyme, we use KOD, DreamTech, and premix, NCY premix, and NCY mix. The good result is from DreamTech and NCY mix. But the primer dimer remains visible in all positive PCR results. PCR reagent type does not affect dimer primers. Solution to difficulties encountered in the development of multiplex PCR has been the use of hot star PCR or nested PCR. But because we want to make a detection kit that have a cheap price, so this is not a good choice. And then for the dilution of all formulation, we try from 2 until 10 dilution. From here, we note that kit lipid still shows false positive until 5 times dilution. However, the primer dimer intensity at 5 dilution was the lowest compared to other dilution concentration. Dimers are still visible at the smallest primer concentration. The last number, we suggest that the last number of primer in the mixed PCR, it is difficult to get with the dimer. And the last is sequencing confirmation. False positive sample using kit lipid showed that the nucleotides was indeed similar to the nucleotide of SPP, 99% similarity with SPP16. So this is not a false positive. For the conclusion, we get optimum inhalation temperature of the kit lipid was 68, minimum primer concentration 0 HPmol. The kit lipid still showed false positive until 5 time dilution with the lowest intensity of primer dimer. Different type of enzyme polymerase in the experiment did not influence the forming of primer dimers. The confirmation-based analysis showed that the result of kit lipid were not false positive as the nucleotide was indeed similar to the nucleotide of SPP. Therefore, the kit lipid is comparable to the COBAS 4800. Thank you very much for the attention.
And thank you for the presentation. Next is the presentation from Ms. Andri Wardi. Sorry, Mr. Andri.
Hey everyone, my name is Andri Wardiana. I'm from Research Center for Biotechnology Green. First of all, I would like to thank the committee for this opportunity to present our research. And today I'm going to talk about our research about development of in vitro binding of SARS-CoV-2 spike protein to S2 receptor. As we know, the COVID-19 has been an outbreak since 2020, and this disease is caused by by virus called severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, and this is the structure of the virus. And there is the crown one. I mean, this is the structure like a crown. This is a spike glycoprotein, which is very important in the binding to the receptor in our body. And this protein or spike proteins are the main target for the development of vaccine, because this protein binds to the receptor on cytoplasmic membrane called S2 receptor, and then it can internalize to the cells and then can load the viral genome into the cells. The binding of SARS-CoV-2 spike to S receptor triggers a large conformational rearrangement of the spike from a metastable pre-fusion conformation to a highly stable post-fusion conformation, which can facilitate membrane fusion. So in terms of the vaccine development, there are several analytical methods typically used for in vitro vaccine potency determination. So many bioanalytical methods for the in vitro determination, such as ELISA, HPLC, flow cytometry, and so many more. And in this research, we will focus on the transgene expression using immunostaining. And therefore, in this research, the aim of this research is to develop in vitro assay using fluorescence microscopy analysis using transgene cell line. So first of all, we need the human embryonic kidney 293, and then we did transfection of human S2 plasmid. We did the transfection using PEI, and after 2 days, we confirmed the expression of the human S2 using Western blot analysis. As we can see that this is we can see the protein vein. This is the Western blot analysis data using the anti-human S2 antibody and using the secondary antibody containing HRP. And also we did the confirmation using beta-actin as the housekeeping gene. And furthermore, we did the immunostaining and then tagging to the fluorophore agent, and then we observe under the fluorescence microscopy, as we can see that in this cell, the blue one is the nuclei, which was colorized, visualized by the DAPI, and the color is blue, and this is the red one. In the membrane cell is the S2 protein. So we confirm that this cell line has been express the S2 protein in the membrane cells. And to confirm the binding of the spike protein, we did the binding of RBD containing GFP protein as a protein model. And then we confirm the binding to the S2 protein. So in spike protein, there is a RBD region, which is a receptor binding domain, and then this domain, this domain contribute to the interaction of the spike protein into S2 receptor. And in this research, we combined the cells containing the RBD, SGFP protein, and also the S2 protein. And then we check the internalization and collocation of the RBD GFP into the S2 protein, because this RBD containing the green fluorescent protein, so we can see directly the green fluorescence if the protein is expressed. And this S2 receptor, which has been described before, containing the cell line containing the S2 receptor, and then we visualize by the red fluorescence, and then we can see, when we combine together, we can see the internalization and collocation of the RBD to the human S2 expressing cell line. And also we confirm the binding assay using the spike fusion protein. This is a recombinant protein containing a spike region as a vaccine candidate. We use the same cell line, which is S2, which is the S2 expressed in human embryonic kidney to 93 cells. And then we incubated our protein for 4 hours in 37 degrees in this transfected cell line. And then we observe the binding of this protein onto the cell line. And then we can see that our protein has the green fluorescence, the green fluorescence in the second antibody. We take with the green fluorescence, and we can see that there is the green one in the membrane cells, which is there is a binding of the protein into the ACE2 receptor, which is expressed in the membrane cells. And also we observe the internalization of the spike protein to the S2 receptor expressing cell line, and then we can see there's an internalization and collocation of the spike protein into S2 expressing HEC cells. As a conclusion, transgene human cell line expressing S2 as a transient expression can be used as in vitro binding assay. Based on our results, using RBD GFP as a model protein and also the recombinant spike protein as a vaccine candidate could be internalized and colocalized with S2 receptor expressing a cell line. And for further research, we will develop a stable expression of S2 in human cell line for routine in vitro binding assay. And that's all, and thank you everyone, and thank you for the COVID-19 vaccine group as well. And I'm happy to take any question. Thank you, and good afternoon.
Yeah. Thank you, Dr. Andri. Now is the presentation from Dr. Adi.
Hello, everyone. First of all, I would like to say thanks to organizing committee for organizing this conference. And also I would like to say thanks to you all for watching these presentations. My name is Adi, as written there, and I'm from LIPI, Research Center for Biotechnology, and now they call it PRIMS. And in these presentations, I would like to discuss about development of a cell line, a cell that produces human erythropoietin. Okay, since EPO is biologics, I think it's important for me to mention a bit about what biologic is. Biologic is basically it's a bigger molecule, bigger and more complex molecule. Okay, bigger and more molecule, and then as you can see over there, the acetyl salicylic acid only 21 atoms. However, the biologic can be up to 20,000 atoms, even bigger, even can be up to 150,000 atoms. Okay, and then this is the structure of EPO itself. This is the structure of EPO. EPO is a glycoprotein. It means the EPO contains sugar moiety. As you can see over there, EPO contains 3 anilines. The one that just like a tree, 1, 2, 3. Okay, the one like a tree. That's 3 aniline, and then also one small O-ling over there. Okay, and then on the right side, that is a structure of aniline and O-ling in the bottom. Okay. Okay. Now I'm going to explain about regulations of EPO in the body. Regulation of EPO in the body. Basically, is that if you have decreased blood oxygen in the body, kidney will produce EPO. Okay, and then the EPO that produce will go to bone marrow. Inside the bone marrow, EPO will stimulate productions of a red blood cell. Okay, and then red blood cell will be increasing, and then finally will increase blood oxygen in the body. Okay, so basically EPO will increase blood oxygen in the body. Okay, and then what is EPO used for? EPO is used in many critical settings. Again, EPO is used in many clinical settings. Overall, EPO is used in the treatment of anemia, including anemia caused by chronic kidney disease, cancer chemotherapy, rheumatoid arthritis, and post-emergency surgery. Okay, and then the main idea of my research, is that we add 2 additional aniline, as you can see over there. Native EPO in the body contain only 3 aniline. Okay, and then we add 2 more. So we modify the native EPO, and then the new EPO has 5 aniline. As you can see over there, right? 5 aniline. And then the first one, the one that 3 aniline only contain 12 salicylic acids. And then the one that contains 5 aniline, it contains 22 salicylic acids. The idea is that by adding 2 more aniline, it will increase the half-life. It will increase the half-life of the protein. So by adding 2 aniline, it will increase the half-life of the protein. So the protein will stay longer in the body. That's the idea of this experiment, the idea of this study. And then this is steps in cell line development. The steps in cell line development can be divided into 3 basically. One is molecular cloning, which includes basic research, right, on the very left side, and then in the middle is cell line development, and followed by process development. And then the one that we are dealing with these presentations is the one that in the middle, cell line development, which has to do with the expression analysis and clone evaluations. Okay, as written there on the bottom, development of a cell line for the productions of a therapeutic protein drugs is complex, laborious, and expensive. So that's very true. Doing this thing is can take months or years, sometimes with no good result. And then this is the illustrations of a typical process to develop mammalian cell line for recombinant protein manufacturing. So basically, these illustrations can be divided into 4, I think. First is transfection, transfection, and followed by selections, and then clone evaluations, and then last one is expansion of selected clones. So basically, in that selection will include a technique called limiting dilutions. Basically, limiting dilution is that we try to get one single cell that that can uptake the DNA of our interest, and finally will express our protein of interest, which is human erythropoietin. But to get that things, it's not easy. It can be complicated. So anyway, it takes can take months. So if you are not lucky, it can take years. And then this is analysis of the protein that we obtained from the experiment. We call it Western blot and gel sieve analysis. It's basically is that, as you can see in the middle, the one that like a ball over there, that's the amino acids. This is the EPO, contain sugar moiety, contain sugar. And then with molecular weight is about 40 to 50 kilodalton. We are hoping that this molecular weight is a high 40, close to 50. That's a good one. And then after we digest this sugar moiety using PMGSF, and then the sugar moiety is gone, and then the protein will shift. The protein will shift from 50 to become about 20. Okay, so that's the idea of this this analysis, gel sieve analysis. So this is a proof that a protein that we got is glycoprotein. Okay, so the protein shift. And then this is my last slide. This is the titer of the clone itself. We have 3 good clone, 193, 145, and 223. And then the first one is about 2 gram per liter media. The second one is about 1.5 gram or 1,500 milligram per liter media. Last one is about 1.3 gram per liter. 1.3 gram per liter media. Okay. And well, hopefully this protein that we got is we can continue doing this thing, and then hopefully this protein become a biosimilar product. So our dream is that we can make our own biosimilar. Hopefully. Thank you.
Okay. Thank you for the presentations. Now it's time for question and answer. Please ask. Anyone has question, please write in the chat box, or you can raise hand. If Miss Penny, please.
Thank you very much. I would like to ask to Dr. Andri Wardiana. So I'm asking about that you are doing research on ACE2 receptor and the correlation with the spike. So I would like to know the correlations between spike in a SARS-CoV-2 with the H2 and the correlations with mutant of the SARS-CoV-2? Thank you.
Okay, thanks for the question. Yeah, in this research, actually, I just we did the in vitro assay, so to confirm whether the spike protein binds to receptor. So the aim of this study is to develop the in vitro assay, the binding assays of spike to S2 receptor. So related to the mutant of spike protein, yeah, we have to determine further if we want to let's say if we want to confirm whether the mutation in spike 2 has a different binding to the S2 receptor. But in this case, we just use in our protein, we use the Wuhan strain. So the first strain. So we do not yet if there is a mutant of spike protein and the results in binding to the S2 receptor. Maybe.
So I have a further questions about this one. So we don't know that there is correlations between the mutant for SARS-CoV with the S2 correlations, right?
Yeah, actually, the one that binds to the S2 is the RBD, receptor binding domain. So in as far as I know that if the RBD itself will correlate with the binding to the S2 receptor. Yeah, and also maybe the design of the protein as well. So if our protein has a special design, for example, and then our protein can emerge more the epitope in the RBD domain, we can see the binding to S2. But in this assay, just the binding assay for the further analysis, if we want to know the further binding analysis, we have to do I think cryo-EM is better method to confirm whether the mutation in the RBD domain or other spike protein region which can make a different structure of the epitope, and then the results in the binding to S2, we have to do more analysis using the cryo-EM, so more to the structural protein analysis.
So if I would say, so the important thing is RBD. So regardless the mutant itself, RBD is very important.
Yeah.
Is it right?
Yeah.
Oh, okay. I got it. Thank you very much.
No worries, thanks.
Good afternoon, Bu Anis. Are you still in the room? Bu Anis, okay. I think I will handle this for a moment, Bapak Ibu. We may continue to have a discussion sessions, to have a question and answer sessions. Anybody would like to ask questions to the three presenters? Just raise your hand or type in the chat box. So if I may ask to Dr. Adi Santoso, just a short questions, Dr. Adi, about the development of cell line.
Yes, sir.
You said that the development of the cell line, any cell line development, is really complex, laborious, and time-consuming, right, Dr. Adi? And and it's really high, how to say, high risk of failure of this research, right? So what kind of things does make you still strive to develop EPO, especially cell line that produce EPO in these situations? What kind of things that make you still there? If I may ask question, sorry for this.
What's the main question is? Can you repeat, please?
Yeah, development of cell line is complex, laborious, and time-consuming, right? And it's very high risk of failure. So, what kind of things does make you stand still to conduct this kind of cell line development research?
Well, I think it's every researcher have their own ideas or will what to do with their research, right? And then I think this EPO actually last time when we sent this the research when we send the cell to a biopharma and then we check it and then we check in electrophob. See, I forgot. And then when we check it, and then the molecular weight is kind of low. Molecular weight is kind of low because of salicylic acid is not as many as what we want, you know. And then for the last few months, actually, our colleague Safety and I we do it again. And then actually about a few months ago, we have a better EPO. You know, we have better EPO with a higher molecular weight. And then we are hoping that the last EPO that we got, this is likely higher molecular weight of EPO. Okay, higher molecular weight of EPO is of course they have more salicylic acids, and then more salicylic acid means that the half-life of the protein is higher, right? So that's actually what we really want. So why I still do this because of the latest results show that we got a higher molecular weight. I think that's the answer.
Okay, thank you, Dr. Adi, for the answers.
Okay.
Thank you so much. So we are still waiting for Dr. Anis to join us. Bapak Ibu, please ask questions to the three presenters. I think they delivered a very informative presentations. Okay, Miss Penny, please, Dr. Penny.
Okay, thank you, Mas Naro. So I have a question to Ibu Dwi Wulandari. So I just want to know, how do you differentiate between to detect high risk and probably high risk? Because it's like not so much different, and how you really distinguish between these two kits. Thank you so much.
Thank you very much for the question. Actually, in this kit, we designed this kit only to get a qualitative result, and we didn't difference the HPP, the high risk HPP or probably high risk. Just only get the result, positive or negative result.
I'm sorry, but how you distinguish between high risk and probably high risk, HR and PHR?
Oh, we get this from the paper. I mean, high risk consists of 12 genotype, 16, 18, and 31, 45, and probably is 26, 30, and 34, and so on. So we designed the primer based on this genotype.
Okay, so it means when you design primer for HR, you use a lower phenotype, and when you design kit for probably high risk, you use a higher phenotype. Is that right?
No, we get the genotype from the gene bank. We get the sequence, and the genotype is sorry.
I'm sorry.
We get the data about the high risk from the paper. High risk consists of 12 genotype. Sorry, we get the data of the high risk from the paper that consists of 12 genotype, and the probability high risk consists of 13 genotype. And the sequence we get from the NCBI, and so we can design the primer based on the sequence.
Okay, thank you very much, Ibu.
You're welcome.
Yeah, now we have Dr. Anis here in the room. Dr. Anis, we have one more questions, if any. If no, I think time is up. Dr. Anis, you may ask to the audience.
Thank you very much for the presentations and for the questions. Thank you also for the participants for the active participation. So we will close this session. Thank you, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Dr. Anis Masuna, for hosting these first sessions. So thank you so much for the presenters and the participants. Bapak Ibu, we'll have video from our partners from PT Wadya Prima Mulia and ITS. Please watch. Thank you so much. Please wait for a moment, Bapak Ibu.
Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut. Plant Genetic Engineering and Breeding Workflow, supported by ITS Science Indonesia. Plant genetic engineering can be done in many ways. One of them can be done as follows. First, growing bacteria for transferring the targeted genes into the plant. By using incubator from member, you can make sure the accuracy and stability of the optimum condition for bacteria to grow with simple user-friendly control button, just like this. Second, DNA extraction and isolation. With nanodrop from brand Thermoscientific, you can be assured the extracted DNA is free from contamination such as phenols and others. With feature called Ecology, we can count the concentration of extracted DNA easily without hassle. Super easy. Third, cloning and designing genes. Targeted genes then cloned with real-time PCR, like Cycler 96 from Roche. We can monitoring the amplification of targeted genes directly. Cycler 96 has 4 different channels and can differentiate 4 different targeted genes simultaneously. And then, we can separate the amplicons with electrophoresis. The targeted genes on agarose gel can be documented very well with gel dot ingenious from Syngene. This gel dot can capture fluorescence signal from dyed DNA band, and then from there we can verify the targeted gene and splice the gene into plasmid. Step 4. Transformation. Plasmid then are inserted into bacteria that we grow earlier. These bacteria will infect plant cells and insert recombinant plasmids into plants. Transformed cells then will be detected with flow cytometry. Meet Aurora from Cytek. This flow cytometry can help you to identify cells that are successfully transformed. Transformed cells with marker will be shown and counted by the software. Step 5. Cell and tissue culture. Transformed plant cells then are cultured into agar in petri dish. The growing transformed plant cell can be captured and documented with cell imaging and multimode reader. Citation 5 from Biotech. Equipped with reliable microscopy module, Citation 5 can offer detection in fluorescence, bright field, HNA, and phase contrast applications. Citation 5 also equipped with magnificent lens up to 60 times. With Gen 5 software, image processing and analyzing are never been this powerful. We can also measure the optimum parameters for the transformed plant. Step 6. Plant breeding. Do not let your precious transformed plants die after all those steps above. Meet Confarent, a reliable climatic chamber for important applications such as growing tall plants, short plants, tissue culture, and incubation. With easy-to-use control panel and many other important features such as broad-spectrum LEDs and dimmable light. Alright, those are 6 steps of plant genetic engineering and breeding that ITS Science Indonesia can work with. Thank you for watching. Contact us on our socials for more info. Stay safe.
Halo, di video ini saya mau pamer alat lab canggih. Lihat deh, ini Magnapure 96 dari Roche. Bisa buat bantu ekstraksi RNA virus COVID-19 secara otomatis. Sebanyak 96 sampel dalam waktu 2 jam. Setelah diekstraksi, sampel dapat diuji dengan metode real-time PCR menggunakan Light Cycler 96 ini. Hasilnya langsung terlihat positif atau negatif. Cepet kan? Hubungi teman-teman di ITS untuk info lebih lanjut. Bye-bye, stay safe.
Oke, Bapak Ibu, now we may continue to the second sessions of the second day of the symposium. Before we start, let me introduce our moderator for the session 2, Dr. Vina Amrita Laksmi. As I have shown it before in the beginning, that Dr. Vina received her doctoral degree from Kagoshima University in Japan in 2020. She is really interested in research related to enzymes for COVID-19 detections, enzymes for food and nutraceuticals, enzymes for rare sugars, and so on and so on. So for the next 5 presenters, we will be moderated by Dr. Vina. I'm calling Dr. Vina. Dr. Vina, are you here in the room now?
Yes.
Okay, great. Thank you. Time is yours.
Thank you, Dr. Naro. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Thank you for joining our conference today. So as already introduced by Dr. Naro, my name is Vina Amrita Laksmi, and today I will lead this session. In this session, we have 5 presenters, and after all of the 5 presenters finish their presentation, we will have Q&A session. You can either writing your question on the chat box or you can ask directly. And because time limitation, I would like to soon start the presentation session. And for the first distinguished presenters, we have Dr. Ratias Maraningrum. Without further ado, Dr. Rati, time is yours.
Very good afternoon, everyone. I'm greeting from Cibinong, Bogor. On this event, I will present our research activity entitled Expression of Receptor Binding Domain, Spike of SARS-CoV-2 as a fusion protein in CHOK1 cell line. A novel coronavirus induced pneumonia named as severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2 that causes coronavirus disease 2019 or COVID-19 was first detected in Wuhan, China in December 2019. So the disease has rapidly increased in epidemic scale and has been announced as the public health emergency of international concern on January 2020. According to WHO, a total of almost 250 million COVID-19 cases have been detected worldwide with 2% of mortality rate. Indonesia has reached 4 million cases with mortality rate 3%. Currently, the pandemic is being controlled worldwide by implementing regular health protocol and carrying out massive vaccination. Because effective therapy against COVID-19 is not yet available. COVID-19 is caused by SARS-CoV-2 infection. Coronaviruses are positive strain RNA genome virus with 27 to 32 kilobase insides. Viral infection begins with the binding of spike-shaped protein on the virus to receptor or co-receptor on the target cell surface as mediated by the receptor binding domain or RBD. The main receptor for SARS-CoV-2 is angiotensin converting enzyme 2 or S2. Spike protein or S protein is the most potential candidate for COVID-19 vaccine. In this study, we developed the RBD of SARS-CoV-2 fusion protein to increase protein half-life. The protein was constructed by linking RBD and the Fc domain of human immunoglobulin G1 to obtain the RBD-IgG-Fc fusion protein. The mammalian cell expression system was used to produce the fusion protein. In this study, the CHOK1 cells were thawed and cultured in the media, and the cells were processed and seeded. After overnight culture, the plasmid was transfected into the cells using lipofectamine, and the transient clones were characterized. The stable transfection was performed using G4.8 as selectable marker. The stable clone also characterized. We use limiting dilution to obtain the single recombinant clone. So the recombinant plasmid was cloned into E. coli, isolated and visualized by agarose gel electrophoresis as shown in this figure. Furthermore, the recombinant plasmid was transfected into CHOK1 cells. This research using RBD green fluorescence protein as a control. The results show that the RBD green fluorescent protein transfected cell produce the GFP, indicating that the protein was successfully expressed in the cell. So although that expression of RBD IgG-Fc cannot be directly observed, the GFP expression indicated that the transfection process is successful. The media from transient transfection was monitored by Western blot method. The results show that the protein was successfully expressed in CHOK1 cells. The supernatant containing fusion protein gave specific reaction with RBD spike monoclonal antibody and also anti-human immunoglobulin monoclonal antibody with 55 kilodalton protein band in size. The transient transfection doesn't initiate the integration of genetic materials into cell genome, so the foreign gene usually lost due to cell division. In order to obtain long-term expression, stable transfection was performed. Limiting dilution was applied as well to have recombinant CHOK1 as a single clone. Based on cell colony size, there were 10 potential clones that selected, and protein expression screening informed that the highest expression was produced by clone number 8 and clone number 9. So to conclude that the RBD IgG Fc fusion protein was successfully produced in CHOK1 cell line as transient and stable expression. That's all my presentation. Thank you.
Thank you very much, Dr. Rati, for the presentation. We will proceed to our next distinguished presenters, Dr. Christina Safirawini Lestari, with the title Prospect of Dengue Vaccine Development with Halal Process-Based Technology. Without further ado, Dr. Christina, time is yours.
Thank you for this opportunity which given to me to participate in this conference. My name is Winnie Lestari from NIHRD. I would like to present my review study with the title of Prospect of Dengue Vaccine Development with Halal Process-Based Technology. Dengue disease is still a global health problem, especially in tropical countries with the highest cases in America and Asia. WHO reported more than 8-fold increases in cases over the last 2 decades. It reaches 5.2 million cases in 2019. Dengue is transmitted through mosquito, mainly Aedes aegypti as a vector. Although the vector control has been developed, the dengue disease outbreak is still hard to prevent. Meanwhile, there is no dengue antiviral drug until now. Hence, vaccination is an alternative way as a preventive that needs to be carried out. Currently, there is only one type of commercial dengue vaccine, namely Dengvaxia, but it still had risk of severe dengue for the group of seronegative status. At present, the technology of dengue vaccine development is varied, but the halal portion-free technology of vaccination should be considered, especially for the country with most Muslim population such as Indonesia. This consideration is to prevent the possibility of community rejection. Therefore, the study aims to explore the prospect of dengue vaccine development using halal process-based technology, which is accepted by Muslim majority countries. This paper is a literature studies using the last 20 years of article journals, conference paper, standards, guidelines, and relevant website. The discussion about the halal material in vaccine development can be studied one by one from the raw material at its stage of the vaccine manufacturing process. In general, the conventional vaccine manufacturing process, example live or inactivated virus, consists of antigen, adjuvant, stabilizer, and other excipient usually intended for vaccine delivery systems. Antigens are source of pathogen viruses that can trigger the immune response. Adjuvant are immunogenicity enhanced, usually use chemicals that do not contact animal products, example aluminum hydroxide or alum. Stabilizer protect vaccine product against other chemical reactions, such as sugar derivatives, which function as a cryoprotectant for vaccine that need to be frozen. Among these components, the antigen is the most crucial component and is related to many material and preparation processes. The antigen is a source of pathogens such as the dengue virus. They forced to fulfill halal technology. The equipment must also comply with Sharia principles and GMP requirements and be free from contamination or contact from nuts. It must go through a cleaning process routinely. Virus cells as a culture cell is widely used to multiply viral antigens. In the cell culture process, there are universal steps from medium preparation until purification. Currently, trypsin is isolated from various sources, but the vaccine industry needs a large quantity of trypsin, ultimately still uses animal trypsin as the material instead of invertebrate or microorganism. Porcine trypsin is commercially used because of its higher selectivity and greater efficiency of about 78% compared to bovine trypsin, around 57%. Therefore, it makes vaccine often question about halal legality. Trypsin is one of main concern in this discussion because of its use in the most vaccine products, especially live attenuated vaccine or live attenuated recombinant vaccine, which are already commercially available at this time. Standard for halal vaccine are emphasis on two potential non-halal aspects, namely raw materials and equipment. According to halal pharmaceutical regulation, the halal aspects must meet the six following conditions. Doesn't contain any parts or products from non-halal animals or animals slaughtered not according to Sharia law. Doesn't contain nuts, safe for consumption, non-toxic, non-intoxicating, harmless to health under the recommended dose. Not prepared, processed, or manufactured using equipment containing nuts. Doesn't contain human parts and derivatives that are not permitted by Sharia law. During the preparation state, processing, manufacturing, packaging, storage, and distribution must be separated from other materials that do not meet halal requirement by Sharia law. According to Fatwa MUI number 4, 2016 concerning immunization, the non-halal or haram products described as product that containing porcine following QS Al-Baqarah 173. IUC states the immunization with haram vaccine is not allowed unless used in emergency condition or Arabian call al-hajat. halal vaccine ingredients have not been found and no available of halal vaccine based on information by medical expert. IUC chairman also stated vaccine is halal even though had no contact with the porcine trypsin enzyme because it was sought chemically in purification process. Even though the IUC has stated it, there are still disagreement between the ulama and society, especially in rural area. So the demands for halal process-based technology is still essential in Muslim majority countries. So we must to take a look how about the dengue vaccine development technology that we have until now, which have a prospect for halal or no porcine-based technology. We have at least 5 approaches to develop dengue vaccine until now. The live attenuated vaccine and activated vaccine had a similar technique using viral cells in culture process. Likewise, the viral vector vaccine that still need a cell culture process. So those technology high possibly using porcine trypsin industry. Recombinant subunit and DNA vaccine had prospect for halal technology, but we have to pay attention in Ecology or yeast medium composition for recombinant subunit vaccine technology because it may use animal-derived substances, including porcine. DNA vaccine using PCR technique for amplification, so it can avoid porcine material. We show some example of ongoing dengue vaccine type in that have been published. Just Dengvaxia that had license until now, but it is known containing porcine trypsin. Some of live attenuated vaccine and inactivated type also contained porcine trypsin. Likewise, the viral vector type that had developed containing porcine trypsin, recombinant subunit and DNA vaccine that developed from NS1 or pre-ME plasmid had prospect to
Dr.
be
Christina?
halal vaccine. Prospect halal vaccine in Indonesia. Indonesia is an archipelago country with a population more than 200 million in 2020. The majority of Indonesian population, nearly 90%, are Muslims, covering 13% of Muslims worldwide. Indonesia has a primary immunization national program for 8 infectious diseases. Vaccination were starting from the group of infants to the adolescents. In addition, vaccination are also given to travelers and prospective pilgrims for Hajj or Umrah. The economic growth rate in Indonesia before the pandemic was 5.3%. After the COVID-19 pandemic, it fell around 0.4 to 2.3%. According to large quantity of vaccine requirement and a relatively good economic growth rate, halal vaccine have good prospect in Indonesia. And as the conclusion the technology platform that use mammalian cell for culture processes need to pay attention to porcine-free trypsin enzyme. So the platforms that have the prospect of becoming a halal are recombinant protein, subunit, and DNA vaccines. Therefore, Indonesia is a prospective country to use the halal dengue vaccine. Thank you for your attention.
Okay, thank you, Dr. Christina, for your very insightful presentations. We will proceed to our third distinguished presenters. Is Dr. Annie Kusumaningtyas. She will present her research with title Determination of protease-producing bacteria for bioactive peptide production. Without further ado, Dr. Annie, time is yours.
Hello everyone. Today I would like to present my research about determination of protease-producing bacteria for bioactive peptide production. Food protein are currently being studied not only their nutritional characteristic, but also their positive impact on human health related to peptide sequences encrypted into native protein. Goat meat has less alchemicity compared to bovine milk. Nutritional values and has many biologically active compounds such as casein, whey protein, and other minor constituents like mineral and vitamin. Proteins are successfully considered as alternative to chemical and eco-friendly for bioactive peptide production. Among different protein producers, microorganisms are mostly exploited for proteins. So the aim of this study is to determine protein-producing bacteria for bioactive peptide production. For the method, first for milk preparation, mare and goat meat was obtained from Bogor, and for goat meat, 100 ml goat meat was defatted by centrifugation, and 6,000 G 4 degrees Celsius for 15 minutes. And defatted milk was hydrolyzed immediately or was stored in -20 degrees Celsius until used. For isolation of protease-producing bacteria, mare milk was inoculated in water agar containing 0.05% skim milk, incubated at 37 degrees Celsius for 20 hours. The presence of protease was indicated by clear zone around the colony. For DNA sequencing analysis, bacterial genome was isolated using bacteria DNA barcoding kits from phosphase, and molecular identification was conducted using 6-dsR RNA. The sequence obtained was then analyzed using MEGA7 and PLAS program. Phylogenetic tree was constructed using neighbor gaining method with 1,000 replicates in the bootstrap test. For crude enzyme preparation, Bacillus thuringiensis crude in the Luria botany crude containing 0.05% casein. And casein, and then to find out the effect of substrate, casein can replaced by fibrinogen, bovine collagen, and fish collagen. Enzyme prepared by precipitation with good ammonium sulfate overnight, and then the enzyme obtained was air-dried and stored at -20 degrees Celsius until use. And enzyme activity assay was conducted according to Barrel et al. For goat meat hydrolysis, goat meat was hydrolyzed using crude protease at 55 degrees Celsius pH 11 for 30 and 60 minutes. And the reaction was stopped in boiled water 95 degrees Celsius for 5 minutes, and then adjusting pH to 7 degrees. And then centrifuge and supernatant was filter sterilized using 0.45 nanometer membrane. For antioxidant assay, firstly prepare working solution of APTS, and then for assay, the peptide was mixed with APTS in microplate, incubated at room temperature for 15 minutes to allow the reaction. The absorbance were resulting mixture was recorded at lambda 409 nanometer. The antioxidant activity or scavenging activity was counted by this equation. For result, based on the gram staining and shape of the cell, bacteria are included in Bacillus group. universal primer has been designed to target conserved site and would amplify 60 sRNA gene and they obtain 1,358 base pair. For sequence alignment showed that bacteria has 100 similarity with Bacillus thuringiensis and Bacillus cereus. Phylogenetic tree analysis showed that the bacteria is closer to Bacillus thuringiensis. For enzyme activity, this was influenced by substrate. When Bacillus was grown in the similar condition but in different substrate, the bacteria produced protease with different activity, and the highest activity was shown by casein. For scavenging activity of the peptide was determined by reduction absorbance after addition of the peptide. 100% of scavenging activity correspond to complete scavenging of APTS. Peptide hydrolysis produced by Bacillus protease showed high activities. It's indicated by scavenging activity was 99 until 100% at 50 time dilution, and the activity was still very good. This means more than 90% at up to 2,000 times dilution. Peptide produced from 30 minutes hydrolysis showed higher activities compared to 60 minutes. Conclusion: the molecular identification using 60 sRNA of protease producing bacteria isolated from horse milk showed that the sequence related to Bacillus thuringiensis and Bacillus cereus. Analysis of phylogenetic tree indicated that bacteria closer to Bacillus thuringiensis. The protease has highest activity in casein substrate. Hydrolysis of goat milk protein using protease for 30 and 60 minutes produce peptide with a has scavenging activity 99 to 100% in 50 times dilution and more than 90% in 200 times dilution. That's all, and thank you.
Thank you very much, Dr. Annie, for your presentations. We will proceed to our next distinguished presenters, which is Dr. Danang Waluyo. He will present his research with title Isolation of Antimalarial Active Compound Derived from Indonesian Soil Actinomycetes streptomyces sp. BioMCC 83335. Dr. Danang, time is yours.
Good afternoon. My name is Danang Waluyo from National Agency for Research and Innovation. I would like to present our work with title Isolation of Anti-Malarial Active Compound Derived from Indonesian Soil Actinomycetes streptomyces sp. BioMCC 83335. Malaria is known as a parasitic disease caused by parasites from genus Plasmodium. World Health Organization reported more than 229 million malaria cases with 4,009 deaths in 2019. WHO recommended artemisinin-based combined therapy as first-line treatment. However, artemisinin resistance cases were reported in Cambodia in 2008. Thus, development of new anti-malarial drug became an urgent task. In other side, natural resources are important for drug development. 25% of currently discovered natural compounds are active, and 40% of them are produced by microbes. Fungi and actinomycetes comprised 85% of active compounds from microbes. In BioMCC or Biotech Center BPPT microbial culture collections, a microbial collection managed by Biotech Center BPPT, which is currently become BRIN, there are more than 27,000 microbial isolated from collected and preserved. These collections composed from fungi and actinomycetes by roughly half by wild. Thus, this microbial collection is very attractive source for drug discovery. We subjected 14,700 microbial botanic extracts prepared from BioMCC for phenotypic screenings against Plasmodium falciparum 3D7 cells. Relative inhibition activity of this extract were quantified by LDH assay. We also assessed the toxicity activity of active extracts against mammalian cell. We used colorectal adenocarcinoma cells line for this purpose. Toxicity activity were measured by CCK8 assay, which is more insensitive than MTT assay. As shown here, more than 600 out of 14,700 extracts were active against the parasite. 270 out of them were not toxic against the LD1 cell line. And after the replication process, 83 extracts were selected as hate. 44 of them were produced by actinomycetes. This study aimed to isolate and identify active compounds from extract of actinomycetes, streptomyces, sp, BioMCC AT3335 that inhibits proliferations of Plasmodium falciparum 3D7 cells. Actinomycetes, streptomyces, sp, BioMCC AT3335 was originally isolated from soil sample taken from Madura Island by acid treatment method. During screening process, extract of this microbe inhibited the growth of Plasmodium falciparum 3D7 cells as much as 140% compared to atovaquone 1 micromolar and selectivity index of more than 100. Total 5 liters of microbes broth were prepared by fermentation as shown here. Before we purify the active compound from microbial extract, it is important for us to predict chemical properties of target compounds. We employed a pre-extraction test to examine the polarity, acid-basic nature, and stability of the target compounds as shown in here. From PET research, both supernatant and biomass layer showed inhibitory activity. The active compounds were presumed to be stable at wide range of pH and could be extracted by ethyl acetate at both pH 2 and 8. For our convenience, we use the biomass as source for active compound purification process. We fractionated the biomass extracted using methanol with silica gel chromatography using several solvents. Bioactivity of each fraction were measured. From the result, fractions eluted by ethyl acetate acetone 1:1 solvent or more polar solvent showed high inhibitory activity. When we analyzed these fractions on silica TLC, fraction 1E, 1A, and acetone showed better separations compared to other fractions. Interestingly, the fractions showed different color when the pH was changed from acid to basic, indicating the fractions contain compounds from anthracycline group. We further analyzed separated compounds in fraction acetone using preparative TLC. Each band appeared on TLC was collected and extracted using chloroform methanol 9:1 and subjected into bioactivity assay. As shown in here, band 1 to 4 showed inhibitory activity. TLC analysis of these bands revealed that the compounds in each band were not well separated yet. This indicated that each compound has close polarity, so they were not well separated in silica-based chromatography. We analyzed band 3 and 4 using HPLC using octadecylsilyl column. From both samples, several peaks was appeared in chromatograms. We predict that the measured band could responsible for the activity. We then subjected these samples into LC-MS analysis. Similar to HPLC analysis, electropherogram of band 3 samples composed from many peaks. We extracted the ion pattern of major peak and we found that the peak has M/Z of 756.3863. The ion pattern of second major peak showed M/Z of 772.38813. We also subjected this sample to proton NMR analysis. Combined with LC-MS analysis results, the major peak was identified as cosmomycin A, and the second major peak was identified as cosmomycin B. We further measured cosmomycin A inhibitory activity against Plasmodium falciparum 3D7 cells and obtained an IC50 of 2.8 nanomolar. This IC50 is lower than that of artemisinins, 18 nanomolar. Cosmomycins possess anti-tumor activity against human and rodents tumors. It also induced the differentiation of trans leukemia cells and showed antimicrobial activity against gram-positive bacteria. To date, both cosmomycin A and B have not been reported as antimalarial agents. For the conclusion, we succeeded isolated an antimalarial compound, cosmomycin A, and its derivative, cosmomycin B, from an Indonesian Actomycet Streptomyces sp. BioMCC 803335 culture blood. We would acknowledge that this work was supported in part by a grant for satraps from JICA and AMED, and a grant from DIPA from BPPT. Thank you.
Okay, thank you very much, Dr. Danang Mario, for your insightful presentations. We will proceed to next presenters, which is Dr. Muhammad Mumtaz Zaki. He will present his research with title SARS-CoV-2 infection in certain groups, a review from COVID-19 study cases. Okay, Dr. Muhammad, time is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Thank you for the ISIBIO committee and all of the participants for joining us in this presentation. My name is Muhammad Mumtaz Zaki, and I would like to present our paper, which titled as SARS-CoV-2 infections in certain groups, a review from COVID-19 study cases. On December 31 in 2019, the China Health Authority alerted the WHO to several cases of pneumonia with unknown etiology in Wuhan, China. The most common symptoms are fever and cough, with severe outcomes requiring intensive care are associated with older age, a higher percentage of comorbidities, and a higher mortality. The lack of pre-existing SARS-CoV-2 specific protective antibodies led to the globally rapid spread of the SARS-CoV-2, even if the vast majority of the infections are reportedly mild or even asymptomatic. The emergence of various variants, such as alpha, beta, and delta variants, may increase the likelihood of viral escape from prior existing neutralizing antibodies. The extent to which host immune responses are able to protect from reinfection is an important factor to consider in estimating how long the COVID-19 pandemic will last. It's commonly regarded that an individual is better protected against the infectious agent following an infection that has been resolved, yet this might not be true in some cases of rapidly mutating viruses, such as COVID-19. With no specific treatments for COVID-19 currently available, a vaccine is expected to provide the best protection against SARS-CoV-2 infection. Thus, in this study, we aim To summarize various COVID-19 cases from vulnerable groups, including children and those with comorbidity, occurrences of reinfections and co-infections, cases in vaccinated individuals, and rapid transmission cases to elucidate if there are any specific SARS-CoV-2 variants that are associated with infection in those groups. For the methodology, we retrieved the data from various index journals online with the keywords of SARS-CoV-2 variants or nCoV or COVID-19, comorbidities, co-infection, reinfection, and rapid spread or highly transmissible. We focus our search on records available online within the last 24 months, only between 2019 and 2021. The initial literature search was conducted from August 6 until August 14 in 2021. Both the titles, abstract, and full text screening were conducted independently by the authors, and any conflicts were resolved by thorough discussion until consensus was achieved. Okay, for the results and discussion, the children and those with comorbidity. There were no patterns and significance of the susceptibility and transmissibility of COVID-19 in children. However, there is clear evidence that most children experience clinically mild disease or remain asymptomatically. It's still unknown whether children are less infectious than adults and the extent of their contribution to transmission. Children tend to produce SARS-CoV-2 memory T-cell responses that are weaker than adults, increasing the likelihood of the children to reinfection. Hypertension, diabetes, and cardiovascular disease are comorbidities in adults that were found to correlate with severity and a great risk of mortality. Immunosuppressed patients are at risk for a longer duration of infection, which may increase the likelihood of new mutations emerging. And then in the second part, the co-infections are SARS-CoV-2 with other pathogens like bacteria, fungi, or other viruses have been reported. An initial study originating from Wuhan, China, reported bacterial co-infections in 50% of patients who did not survive COVID-19. A detailed analysis of the co-infecting pathogens showed Staphylococcus aureus, Haemophilus influenzae, Streptococcus pneumonia, and Klebsiella pneumonia to be the most prevalent bacterial co-infections. There have been multiple cases of people who were infected with both the novel coronavirus from different variants at the same time. As an example, an elderly woman in Belgium was concurrently infected with two variants of the novel coronavirus in a rare case of viral co-infection. The 19-year-old woman who was not vaccinated against COVID-19 developed a simultaneous infection with both the alpha and the beta variants. And then for the third part, reinfection and cases in vaccinated individuals, coronaviruses have been known to cause reinfections where it's likely that the antibodies cannot recognize the epitopes of reinfecting virus. There are 15 cases of reinfection with SARS-CoV-2 have been reported, where sequencing data of the SARS-CoV-2 RNA from the first and second infections were available. Governor et al. Found that among all patients that are previously infected with SARS-CoV-2, unvaccinated people had significantly higher reinfection cases. However, the emergence of new variants might affect the duration of infection-acquired immunity. The lower probability of reinfection among the partially vaccinated group compared to the unvaccinated group suggests a protective effect. And for the fourth part, the rapid spread of COVID-19 cases. We found that the rapid spread of SARS-CoV-2 is caused by adaptive mutations in the viral genome that can alter the pathogenic potential of the virus. 3 of the variants of Delta lineage and sublineage have a potentially higher rate of transmission than the others. This aid showed that the Delta variant B.1.617.2 type of lineage have cumulatively become the highest cause of COVID-19 in the world. For the Alpha variant, it has 13 of the lineage dividing mutations that are hypothesized to increase its transmissibility. As of August 8, 2021, in GIS8, the Alpha variant B.1.7 occupied the second position in the world. And for the Gamma variant has been associated with higher transmissibility. As at August 8, 2021, the Gamma variant B.1 occupied the fourth position in the world based on GIS8 data. For the results and discussion, suggestion and further studies, collectively, these studies highlight the importance of genomic surveillance in the management of COVID-19, especially in certain groups. Genomic surveillance is also useful in informing variants circulating in particular areas at a certain time, therefore enabling mitigation of potential outbreaks. Combination of enhanced genomic surveillance and continued compliance of preventive public health measures, including vaccination, physical distancing, use of masks, hand hygiene, and isolation and quarantine when necessary, remains essential to limit the spread of SARS-CoV-2 infection and COVID-19 cases. And last conclusion, there are no particular variants that infect certain groups, as any SARS-CoV-2 variants can infect any groups with any immunity levels without any geographical boundaries. The severity of the disease is influenced by both the virus and the host conditions such as gender, age, comorbidity, etc. Monitoring of circulating variants of SARS-CoV-2 by whole genome sequencing is a strategy to control the disease, as well as the implementation of preventive measures, as SARS-CoV-2 variants are still highly transmissible.
Okay, thank you very much, Dr. Muhammad, for your very insightful presentations. So we already finished our presentation sessions. Next, we have Q&A sessions. You can either write your question in the chat box or you can directly ask by using raise hand. Because still no one asked, so first I would like ask a question to our third presenters, Dr. Annie Kusumaningtyas. So in your presentation, you explained that you already determined the activity of the protease. Can you give us the information using what kind of protein you use when you determine the activity? You using crude protein or already purified fraction of protein?
Okay, thank you, Dr. Vina. The antioxidant activity. This from cold meat protein was hydrolyzed using protease of Bacillus thuringiensis. This after hydrolysis, and we test the activity without purification or filtration. I mean, this may be contain many different size of peptide.
Okay, so Dr. Adi used crude protein for determination of the activity.
Yes, yes.
Okay, thank you.
Thank you.
Dr. Rani. So as we waiting for the question for the participant here, I would like to ask our second presenter, Dr. Christina, about the dengue vaccines. So would you please give us information about the commercial dengue vaccines already available in the market? Is there any?
Yes, thank you. Previously, I apologize on behalf of Dr. Christina's because she is on duty in the rural areas now, so she couldn't attend the session. I will represent her as one of author of our review paper. For a commercial dengue vaccine that present now is a Dengvaxia. It is a live attenuated vaccine type. This is the only vaccine that have license, but it use a yellow fever virus as a backbone. So we know that the yellow fever virus, it's not still reported on the Indonesian now. So we must find a new surveillance that could be better than it. And the Dengvaxia has known it didn't effective for seronegative status patients, so it couldn't efficient. But there is only one type of vaccine that we have now that have a license and pass the clinical trials. Thank you.
Okay, thank you very much. Okay, we have one participant who would like to ask Dr. Penny Ahmadi.
Thank you very much, Dr. Fina. So I have a question for Dr. Ratih. So in your presentations, you explain about you develop RBD with, you link the RBD with the antibody, if I'm not mistaken. So I'm wondering what kind of link between RBD with the antibody? Is it covalent bonding or chemical bonding like a thiol, or is it using like a binding pocket? Thank you very much.
Thank you, Penny. Sorry, I'm not feeling well currently, so apologize if my answer is not optimal. So in protein modification, there are several approaches that we can use to create like a fusion protein. We can use two different type of the protein, then we link it using the short peptide. Or actually, we can also chemically modify the protein by using like the big molecule like polyethylene glycol or something. Actually, the purpose is the same. It is for improve the half-life of the protein or to improve its antigenicity. So in this case, we use like the commercial plasmid. We didn't construct ourselves because this construct is we just use only to develop the if you already hear the presentation from Andri Wardiana, we develop like the system to see the interaction of the RBD with the H2 receptor. So we use the commercials and it's linked by the short peptide.
So thank you very much. So short peptide and big if we compare short peptide with and a big molecule, so short peptide it's more much more better compared to the big peptide. Is that correct? Or depend?
I think if we want to do the like protein modification, actually we have like to do like the bioinformatic study first because like the certain linker, actually it's not form like the expecting fusion structure. It also can form and cover like the active site of our target protein. So it's depend on your protein, the structure, the complexity of your protein. So sometimes we just trial and error to get the expected form protein structure.
I got it. Thank you very much.
Okay, thank you very much, Dr. Penny, for your question, and thank you, Dr. Rati, for your precise answer. Is there any other who want to ask? Okay, Dr. Nero.
Okay, thank you, Dr. Fina, for the opportunity. I would like to ask a question to Dr. Danang Waluyo. Pak Danang, you mentioned about cosmomycin A and B. If I'm not mistaken, most studies conducted using cosmomycins A and B to develop antibacterial drugs or agents. So in this case, you found that cosmomycin A and B can inhibit the growth of protozoan. I forgot the name, sorry. Can you speculate the mechanisms of the inhibitions of those protozoans by cosmomycin A or any cosmomycins? Thank you so much.
Okay, thank you for your questions. Okay, actually, yeah, we assessed the, we found that the cosmomycin A, especially could inhibit the growth of Plasmodium falciparum. Yeah, parasite cause the malaria disease, and yeah, so far we didn't did any investigations about the mode of actions of this compound in inhibiting of the growth of Plasmodium. But we also conduct the culture of the Plasmodium. Yeah, actually we also tested this compound against the mammalian cells, yeah, to measure the toxicity activity of this compound against mammalian cells in quite severe conditions. It means that we culture the mammalian cell in nutrient deprived conditions and also in quite low oxygen conditions. At that time, we found that the mammalian cell could grow, yeah, not so good, but it could grow even in the presence of the compound and also in the presence in the conditions in these severe conditions. It means that maybe the cosmomycins did not affect the respiratory system of the cells. It means that we know that the respiratory system between mammalian cell and Plasmodium is quite different. So maybe this compound could also inhibit the respiratory systems in the parasites. But you know, it is just my speculations because we didn't inspect more deeper about the action of mode of these compounds in the Plasmodium falciparum. But yeah, it is quite an interesting finding. So maybe in the future we would like to see more deeper about the action of mode in the parasites. Thank you for your questions.
Thank you so much.
Okay, thank you very much, Dr. Naro, for the question and Dr. Danang for your precise answer. Because of time limitation, unfortunately, we need to end this session. Hopefully, we can still meet in the next conference. Okay, thank you, everyone.
Thank you, Dr. Fina. Thank you, participants, Bapak Ibu, professors, doctors, for attending the second sessions in the room 3. So we'll be having 10 minutes break at your own home, yeah, just to enjoy your coffee or going to toilet. But please, going back to this room before 4 p.m. Thank you so much. Enjoy your break.
Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut.
National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies. National Research and Innovation Agency, a synergy in an orchestra. Established through the Presidential Regulation Number 78 of 2021 about the National Research and Innovation Agency. To enhance science and technology's contribution to the national development and to improve the research and innovation ecosystem in Indonesia. The National Research and Innovation Agency is a government agency that is directly responsible to the President of the Republic of Indonesia. Consists of 7 deputies, supported by various research institutes, and have more than 70 research centers and supporting units. Through the 3 directions: science and technology resources mobilization, providing a favorable research ecosystem, building an economy foundations based on research. The presence of National Research and Innovation Agency is to achieve Indonesia's goals as a developed country in 2045. Also to make Indonesia as a global reference for sciences, bringing advantages on science and technology development, policy making, and societies.
Hello, good evening, Bapak Ibu. Thank you very much for staying with us until this last sessions. But before we move to next sessions, let us see some video commercial from our partner from Vidya Pramamulya and ITS. Please, let's see.
Martin Chris, freeze drying system untuk penelitian, pilot project, hingga manufacturing produk Anda. Hubungi PT ITS Science Indonesia untuk informasi lebih lanjut. Halo, kali ini saya mau memperkenalkan alat laboratorium dari Ingenious 3. Sebelum pengenalan alat, apakah Anda pernah mengalami kesulitan saat mendokumentasikan hasil gel electrophoresis atau plot Anda di ruang gelap? Sistem dokumentasi gel secara manual terdiri dari ruang gelap, kamera, dan transluminator sederhana. Dokumentasi gel secara manual seperti ini kadang menyulitkan dan belum lagi kalau hasil foto gel yang didapat ternyata blur. Dengan Ingenious 3, Anda dapat memilih pencahayaan sinar UV, sinar biru, atau putih sesuai dengan pewarna yang Anda gunakan. Selain itu, software Genesis akan membantu Anda memilih pengaturan cahaya dan filter yang tepat sehingga sangat memudahkan untuk mendokumentasikan gel fluorescence ethidium bromida atau cybersafe DNA atau protein gel dengan composite blue atau silver staining. Untuk penggunaan mode otomatis, Anda hanya perlu memilih tipe gel yang sesuai, kemudian software akan secara otomatis mengatur aperture, zoom, dan fokus serta posisi gel yang sesuai sehingga gambar gel terekam secara optimum. Software Genesis dilengkapi dengan database tipe gel dan pewarna yang tersedia di pasaran. Jadi jangan khawatir tidak bisa menemukan pengaturan yang sesuai. Sangat mudah. Hubungi tim ITS Science Indonesia untuk info lebih lanjut. Terima kasih. Stay safe.
Microtomy requires my full attention and dedication to make sure that I deliver the perfect slides. And with the Tissue Tech Auto section, I started getting the job done easier, faster, and with better quality. The automated block alignment makes sure precious tissue is preserved optimally. It is also a great help for making recuts, while the perfect alignment helps me produce even more slides. Trimming and sectioning are done with high precision, each section the same exact thickness. Thanks to the easy operation of the auto section, every technician in our laboratory is quickly up and running. Whatever the need, we are all confident to deliver the perfect slide each time. My name is Mark Shanti, and I'm empowered by Sakura.
Okay, thank you very much, Bapak Ibu. For the next sessions, for the last sessions, it will be moderated by Dr. Hani Susanti. But before that, let me introduce her. I read her background. So Dr. Hani Susanti is she got a doctorate at University of Tsukuba, Japan in 2020, and her current institution is in researcher at the Center for Drug Discovery and Development and Research Center for Biotechnology, National Research and Innovation Agency. And her research is about biotechnology for aquaculture, microalgae culture collections. And let's let me invite her. Please, Dr. Hani Susanti, please lead this session. Thank you very much.
Thank you very much, Dr. Penny, for the opportunity. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Hello, everyone. This is my honor to be a moderator in the final parallel session. Even so, this is the final session. I do believe that all the attendee or participant will pay the best attention to all the presenters today. So currently we have 4 presenters. I would like to remain the timeline, 10 minutes for the presentation and followed by the question and answer session after all the presenters perform. So for all the attenders who has a question before the presentation ends up, you may write down into the chat box or just wait until the question and answer session comes up. And then without further ado, I would like to invite the first presenter, Miss Ibu Yuliawati. Ibu Yuliawati will present with entitled Construction of Multi-Copy Glucagon-like Peptide 1 Open Reading Frame and its expression in Ecology. So for Ibu Yuliawati, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. I would like to present our study entitled Construction of Multi-Copy Glucagon-like Peptide 1 or GLP-1 Open Reading Frame and its expression in Escherichia coli. The members of this research are Yuliawati, Alfi Taufik Fatur Rahman, Winkus Haryoto, and Ratih Asmananingrum. GLP-1 is a hormone produced by L cell in the digestive tract from the transcription product of the proglucagon gene and is classified as an incretin. GLP-1 undergoes limited proteolysis in its synthesis. The active form of GLP-1 are GLP-1 7 to 37 and GLP-1 7 to 36 amide. Under normal condition, GLP-1 will trigger insulin release and inhibit glucagon secretion in response to the digestive process. This activity makes GLP-1 very potential to be used in the treatment of diabetes mellitus. The use of GLP-1 has a limitation that has a half-life of less than 2 minutes due to the cleavage degradation by the enzyme dipeptidylpeptidase 4 or DPP4. Various approaches has been taken to improve the pharmacokinetic profile of GLP-1. Among them, by modifying amino acid to make analogs. As example, deoxyxanthate, albiglutide, and commercial products exenatide from Eli Lilly, analog of GLP-1 that more resistant to degradation by DPP4. The aim of this study is to provide a process for the production of recombinant GLP-1 in Ecology. The multi-copy ORF was added or extended in multiples of H copies so that production level expected will increase. The construction of H copies or multi-copy ORF encoding GLP-1 in the expression factor PDH64, namely GLP-P recombinant plasmid. It was made synthetically by Atom USA. The multi-copy ORF is added with a polyhistidine site at the C-terminal. This site can bind to a metal cation undergoing cobalt ligand binding to facilitate the purification process. To increase the gene copy from 8 to 16 copies, the multi-copy gene plasmid were isolated with BAGL2 and EXO1. Parallelly, but also cut with BAM-H1 and EXO1. Both of the isolated gene and the cut plasmid were then ligated to obtain the recombinant plasmid containing 16 copies. The method can be used to increase the gene copy from 16 to 24 or from 16 to 32 copies. The plasmid containing gene encoding GLP-1 was used for the transformation of Ecology DH5-alpha using the heat shock method. The recombinant plasmid was then used for the transformation of Ecology Nico21 with a similar process and section. The cell pellet were then separated from medium and lysed using lysis buffer. Then the cell component debris fraction was then redissolved using solubilization buffer. The lysate and supernatant were then characterized by protein electrophoresis using the SDS page and Western blot according to the method Sambrook et al. Production optimization was carried out to obtain the highest amount of protein. Transformants were cultivated at 3 different temperature, concentration variation, and the time point of production. Then, protein solution was applied to purified with Hispur cobalt resin. The construction of H copies ORF encoding GLPP and modification for the addition of H4 copies of ORF to 16 copies has been successfully obtained. The molecular weight characterization by restriction analysis are in accordance with the theoretical size of the truncated plasmid without the insertion gene. Protein expression was performed on the construct of 8 copies of the gene encoding GLPP in E. coli Nico21, of which the apparent molecular weight was consistent with its theoretical molecular weight. It's about 24 kilodalton. Clone number 3 was chosen for optimization for GLPP protein expression with the highest protein expression level among other clones based on the result of analysis using imaging. Optimization of GLPP recombinant protein production was carried out at various temperatures. In this figure, show that the GLPP recombinant protein expression at 30 degrees Celsius was higher compared to other selected temperatures analyzed by imaging. Several concentrations of rhamnose were used to determine the optimal inducer concentration. From this figure, it can be seen that the best inducer concentration is 0.5 millimolar, which can produce a higher GLPP recombinant protein expression level than other concentration. The optimum inducer concentration with the highest protein expression level determined by imaging analysis. Optimization of the production time of the recombinant protein was conducted at the optimum temperature and inducer concentration. The results show that the optimum GLPP recombinant protein expression level produced in E. coli Nico21 was at 22 hours. The optimum production time with the highest protein expression level determining using image analysis. In this figure, show the effect of protease inhibitor. We use PMSF or cocktail. Show that there was protein degradation in the protein isolation without the addition of protease inhibitor, which was marked by the appearance of several bonds several bands on the western blot result. Based on the optimization data, GLP was produced in Ecology Nico 21 on a 100 milliscale. The harvested pellet splitting by sonication to get the inclusion bodies protein. The IBIS protein was then purified. The results show that the protein isolation result from IBIS indicate a GLPP protein band with a size about 24 kilodalton. Furthermore, the purified IBIS figure in this figure also indicate a similar GLPP protein band size. For the conclusion, the recombinant plasmid carry out H and 16 multi-copies gene encoding GLP-1 were successfully constructed and characterized. GLPP protein expression only performed on H gene multi-copies and as preliminary study. The molecular weight of the recombinant H multi-copy GLPP based on the SDS page and Western blot analysis was the same size and the theoretical GLP-1, about 24 kilodalton. The recombinant H multi-copy GLP-1 were expressed as inclusion body and successfully purified by Talon superflow affinity chromatography. Thank you very much.
Okay, thank you very much for the presentation. And we next to the second presenter. This is going to be a very interesting topic related to the current situation of pandemic. Will be presented by Pak Kambang Saryaji. He will present with entitled Detection of SARS-CoV-2 in the air, surface and workers in receiving area of COVID-19 diagnostic laboratory at National Institute of Code Research and Development. For Pak Kambang Saryaji, time is yours.
Okay, thank you very much for the time that given to me. I'm really happy to be here. And I would like to say Assalamualaikum warahmatullahi wabarakatuh. And I would like to say thank you very much for the audience that already coming to my presentation. I think this is the best time for me to discussing about the SARS-CoV in the air, surface, and worker in receiving area of COVID-19 diagnostic laboratory at National Institute of Health Research and Development. And regarding of my presentation, I think that everybody know that the COVID or the SARS-CoV that already disaster in the world. I think this is the pandemic that had taking many people dead, and the traveling of from one country to the other country has a limitation restriction, and the activity of the work on the office or on the school of course has inhibition or have I think this could be can be stopped at the moment during in pandemic. And I would like to introducing my name. My name is Kambang Saryaji, and I work in the National Institute and Health Research and Development. And I am a researcher. And for today, I would like to present my research regarding of the SARS-CoV, detection of SARS-CoV in the air surface and worker in receiving area of COVID-19. And we begin from the introduction, as we know that the COVID-19 cases caused by the SARS-CoV-2 has become a worldwide pandemic in the early 2020. We have data from the WHO and mentioned that the number of people who are infected with the COVID-19 in the world has nearly surpassed 6 million. This is in the early June 2020. Maybe can be more and more people have get the infected with the COVID-19, and the death rate or CFR also varies in each country and is also continuing to increase. And currently, the world mortality rate for the Southeast Asia region and Indonesia are around 6 up to 7%, 3 up to 4%, and 6 up to 7%. Some cases of the COVID-19 are symptomatic or maybe mildly symptomatic, even though without symptoms, but the SARS-CoV itself still have the ability to transfer the disease from person to person. And SARS-CoV-2 also can be transmitted with spread primarily through the droplet generated when an infected person coughs or sneezes, or through the droplet of saliva or discharge from the nose. We know that the droplet something fall into an object surface. The surface can be floor, can be a wall, can be the table, or uh, can be anything. The surface of the object of anything that we find it, and the people may be or may also become infected by touching surfaces that have been contaminated by the virus when touching their eyes, nose, or mouth without cleaning their hands. And laboratory as a place to testing for COVID-19, I think this is an area who have a potentiality for the spreading and transmission the SARS-CoV to the worker inside. And we know that in laboratory also, especially for the specimen activity, such as the unboxing, arranging, identifying, and labeling specimen. Of course, we conduct this in the BSG to minimize of the contaminated. And the specimen packaging also has a potential to be a source of the transmission and spread of COVID-19 in the referral laboratory. And the purpose of the study is to now to detect the presence of SARS-CoV-2 in the air, water, and the surface of the object, specimen COVID-19. And we continue with the material and method. The team and place research, we conduct this in the July 2nd up to 12, 2020 in the National Institute and Health Research Department Laboratory. And we collecting the specimen was carried out in the COVID-19 specimen reception room. And there were 40. All of the specimen is 24 divided into 3. There were 40 surface area point specimens such as BHC, inside and outside, table receiving specimen, air conditioner, refrigerator, wall surface, door surface, handle door surfaces, and floor surfaces. All specimens collected by swab surfaces, then the swab insert to the tube contain VTM. And tree workers who are working in the set in the receiving room, we also have taking the specimen by using naso and oropharynx swab combination both. And 7 air sample in proportional point using the air sample with 1,000 liter 10 minutes every 10 minutes during 30 minutes. And the testing procedure, this assay detect target nucleic capsid and gene according to the CDC screening protocol. Extraction of viral RNA from the sample using the KM viral RNA minikit from Kiagen and the multi-pack PCR procedure SARS-CoV-2 detection protocol from US CDC Atlanta. And the result from the 24 sampling point area in the COVID-19 specimen, the receiving room, all of the specimen are negative. And the discussion, the activity of specimens such as unboxing, identifying, and labeling could cause places, leaking tube sample, surfaces contamination from the packing container and from document, or the result of swab examination at the several points such as the BSG, table receiving, specimen, refrigerator, wall, floor, door surface, and door knob show negative COVID-19. This result indicated that the activity performed while handling COVID-19 specimen, I think this is with good standard biosafety procedure that could be minimized contamination and regularly decontaminated. And the PCR screening from the 7S sample from the COVID-19 specimen receiving room revealed that they were negative for SARS-CoV-2. This is indicate the air in the COVID-19 specimen receiving room was free from the SARS-CoV-2 at that time. The PCR also result from the 2 air conditioner and 3 worker were all negative. It showed that no worker were infected while working in the specimen receiving room since last month. Because if a worker infected with COVID-19, the worker could also infect it. And conclusion, all the tests show negative for COVID-19. It could be that laboratory worker become infected if they are not appropriate PPE when handling biological specimen from the packaging specimen COVID-19 are received. They could also infected from the air surface in that room if the decontaminated of room was done unrootedly. Thank you very much.
Okay, thank you very much, Pak Kambang, for very nice information, scientific information regarding the detection of SARS-CoV-2 in the air surface and worker in the receiving area of COVID-19 diagnostic laboratory. So we will be heading to the next presenter, Dr. Rikno Harmoko. Are you ready for the presentation entitled Production of a Polyclonal Antibody against Arabidopsis thaliana ascorbate peroxide 2? So without further ado, let's watch the recorded video or presentation from Dr. Rikno. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. I would like to say thanks to conference committee for a chance to deliver presentation in this conference. My name is Rikno Harmoko. The title of my presentation today is Production of Polyclonal Antibody against Arabidopsis thaliana ascorbate peroxidase 2. For introductions, reactive oxygen species or ROS is unstable molecule containing oxygens that easily reacts with other molecules in cell. Hydrogen peroxide H2O2, hydroxyl radicals, superoxide, and singlet oxygen is classified into ROS. This molecule is byproduct of aerobic metabolism and recognized as toxic substances. Excessive frost accumulation is generated in response to abiotic stress. For example, salt, drought, heat, and cold stresses. High accumulation of ROS might affect imbalance of oxidant and antioxidant. This condition might cause oxidative stresses. Oxidative stresses ultimately might lead to molecular damage, cellular damage, and plant cell death mechanism, including necrosis and senescence. The most dangerous ROS is H2O2 or hydrogen peroxide in plant. Because this substance is the most stable ROS in cell with half-life 1 to 3 hours in cell. The H2O2 is soluble in water and can move from one cell to another cell via membrane. At low concentration, the H2O2 can activate signaling pathway that induce growth and development. However, at high concentration, this substance may cause oxidative stress that damage the cellular and molecular structure. The H2O2 is byproduct of cellular metabolism in mitochondria, chloroplast, and microbody. Plant cell has evolved a mechanism in neutralizing H2O2 by using ascorbate peroxidase enzyme and ascorbate acid as electron donor. Ascorbate peroxidase or APX enzyme play a role in peroxide scavenging by reducing H2O2 to water using ascorbate as an electron donor. In plant, especially Arabidopsis, has 8 homologue of APX gene. APX1 and APX2 localized in cytosol, APX3, 5 localized in microsomal. Stromal and thylakoid APX is localized in chloroplast. APX6 and APX4 localization is still unknown. Each APX gene or protein has specific role in metabolisms. For example, TAPX or SAPX, which is localized in the leaf of green tissue, is responsible to neutralize H2O2 produced by photosynthesis. ATAPX1, which is localized in the leaf, is major peroxide enzyme that responsive to abiotic stress. Today, presentation is focused only on ATAPX2, which is the role of metabolism also still unknown. To understand ATAPX2 role require a molecular detection tool, for instance, a specific antibody. The specific antibody for ATAPX2 is still unavailable. In the present study, we will isolate APX cDNA from heat-treated Arabidopsis thaliana. The cDNA was expressed in E. coli to obtain recombinant protein. The purified recombinant protein was used to raise polyclonal antibody against APX2. For a result, we have isolated APX2 cDNA from heat-treated Arabidopsis using RT-PCR method. The APX cDNA was cloned into pGMT-EC vector and the sequence was confirmed by Sanger sequencing method. Based on the Sanger sequencing method, the APX-cDNA, our APX-cDNA has 100 sequence similarity to the database. In the figure A, based on our sequence, we perform protein sequence alignment by comparing APX1, APX2, and APX6. The APX2 has sequence similarity up to 6.4% and 30.3% in comparison to APX1 and APX6. In the figure B and C, we perform predictive analysis using bioinformatic tools. Domain prediction analysis in figure B show that the APX2 has specific peroxidase domain from amino acid 191 to 251. However, in the figure C, structural domain show that this protein has 11 alpha helices and 2 beta sheets in their structure. For the production of recombinant APX2, we clone the APX2-cDNA in PET28 plasmid expression plasmid, which is fused to thrombin side sequence and his-tag amino acid. Both his-tag and thrombins will facilitate downstream purification of recombinant protein. The APX2 ascorbate peroxidase 2 protein was expressed at 30 degrees Celsius for 4 hours under the induction of 0.5 millimolar IPTG. As a result, the APX2 protein was expressed at soluble fraction in the Ecology protein lysate. The recombinant protein were then purified using NE-NTA affinity column. The histidine tag was released from recombinant protein using thrombin protease digestions. As a result, 28 kDa recombinant APX2 protein was successfully purified using this method. For the production of polyclonal antibody, we mix APX2 protein with complete adjuvant and injected into rabbit for immunization. The immunization was repeated 4 times for boosting at 2-week intervals. After 1 week after last injection, the blood was harvested from the rabbit and the serum was separated from the blood using by centrifugation. The serum was used as secondary antibody in the Western blot analysis. To check the concentration of antibody in the serums, we perform Western blot analysis using protein from Arabidopsis thaliana. The APX protein in the total protein of Arabidopsis thaliana were detected using serums at several ratio dilution from 1 to 1,000 and 1 to 100,000 ratio. As a result here, the protein signal was observed in all of dilution ratio. Interestingly, in 1 to 100,000, the signal still clearly observed. Taken together, this data indicating that polyclonal antibody was successfully generated in blood serum of rabbit, and the polyclonal antibody present in high concentration in blood serums. The conclusion is we have isolated APX2 cDNA from heat-treated Arabidopsis. The APX cDNA was successfully expressed in Ecology BL21 at condition 0.5 millimolar IPTG, 30 degrees Celsius for 4 hours. The antibody was successfully generated in rabbit and specifically recognized endogenous APX2 from Arabidopsis. This antibody could be used for molecular analysis to explore the molecular regulation and function of APX2. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for the precise presentation. So we will heading to the last but not least presenter, Dr. Desriani. She will present about the PTEN promoter methylation study in West Sumatra breast cancer patients. So, Dr. Desriani, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the time and opportunity. First of all, I would like to introduce myself. My name is Desriani. I'm from the Center for Biotechnology, PRIN. On this occasion, I would like to report our recent activity with the title P10 Promoter Methylation Study in West Sumatra Breast Cancer Patient. As introduction, in the world, female breast cancer and also in Indonesia is leading as new cases among of all cancer cases. That is one of the important biomarker in the breast cancer called as phosphatase and tensine homolog, or we call as P10, is a tumor suppressor gene located in the chromosome number 10 with negatively regulates PIK3CA active pathway. Loss P10 related with the tumorigenesis and aggressive tumor behavior in breast cancer, and P10 also correlated with a malignancy and poor prognosis. In some report, this molecule potentially as target for effective therapy and also for diagnosis of breast cancer. 31.2% of breast cancer have a P10 promoter methylation, that 64.3% of which have lost P10 expressions. Can also shown the same report, the same pattern, that is 34% of P10 methylation occurred in this cancer, and 60% were correlated with lost P10 protein. So far, there is no study about P10 promoter in Indonesia, especially in West Sumatra. And here in this research activity, our objective is to investigate methylation in P10 promoter in West Sumatra breast cancer patient, and to determine association of P10 promoter methylation with some of clinical pathological parameter. Here is the method how we collect the sample from the patient, tissue sample from the patient. Actually, this is done by our colleague, Professor Azamris. And then we did the sample from the sample preparation. Then we did the DNA genome extraction, DNA concentration measurement, and DNA disulfide conversions. And we did the analysis of P10 with the methylation specific PCR, or we call as MSP. And then we did the statistical analysis. Here is some of the kit and also some of the device that we use in this recent activity. As the result and discussion, in the beginning, first of all, we did the MSP, but we use non-bisulfide modified DNA. That we did the MSP starting from the 45 analytic temperature and 2 to 65 degrees Celsius. We did this activity in order to confirm for the DNA genome contamination probability that will be exist because of the failure of DNA bisulfide conversion, or maybe because of partial DNA bisulfite conversions. And as the result here, there is no band axis in the high temperature, while in the below temperature we can see the PCR product band that exists around 500 base pair, not in the targeted base pair for the methylated and unmethylated band. So after we did this activity, then we did the annealing temperature optimization starting from 450 that we have already known that the high temperature will show no band. Then we did the annealing temperature optimization in the high temperature, and we found, actually the data was not shown in here, but we found around 55-degree temperature as the best annealing temperature for both, for the MSP for the methylation and also for the unmethylation determination. And we can see the profile for the MSP both for the methylation or unmethylation, as we can see here in the figure 2. That is, for the methylation, we will show a band around 206 base pair, while for the unmethylation, we will show around 162 base pair. And based on the base MSP setting temperature, then we did the screening for the 36 breast cancer patient, and we found around 7% sample as the methylated sample, while for the 29 around 19% as the methylated sample, while the around 81% as the unmethylated sample. And further, we did the correlation between the methylation of the P10 promoter with the clinical pathological parameter in the patient from West Sumatra. And as a result, we can see here that the parameter of the age have shown a correlation with the P10 promoter methylation, while metastasis and stage have no correlation with the P10 promoter methylation. Finally, as the conclusion here, that is in West Sumatra, in our respectivity, shown detect P10 promoter methylation around 19.44% as methylated DNA, while for the 80.56% detected as unmethylated. That is, P10 promoter methylation correlates with patient age and show no correlation with carcinoma stage and metastatic. But since the sample used in the study were limited, it needs more sample and more complete clinical pathological parameter for more understanding P10 methylation in West Sumatra breast cancer patient. I think that's all. I have already report our result. Thank you very much for your attention.
Okay, thank you very much. Now time for discussion. So I invite all the presenter, all the speaker to open up the camera. And also I do invite the participant for the question or comment or clarification. Before that, I would like to congratulate Ibu Yuliawati and Pak Rikno for the achievement already generate the antibody and the recombinant protein. So, is there any question? So I have one. It's such a awkward question for Pak Rikno. Why you use the New Zealand white rabbit instead of Indonesian rabbit for your research? Is there any special reason for this?
Thank you for the question. why I use the New Zealand white rabbit, because
Yeah.
this is the standard in making antibody from rabbit. Because if we use secondary antibody, which we generated, then the first antibody should be against white rabbit New Zealand, like that. It should be connected. If we use another kind of rabbit, the first antibody and secondary antibody will not connect it like that. The available first secondary antibody is from white New Zealand rabbit. So that's why the secondary antibody should be from New Zealand rabbit, like that.
I see. Thank you so much. And then, is there any question for the other audience? Okay, Dr. Penny. Okay.
Thank you very much, Dr. Hani. Actually, I have two questions for Dr. Yuliawati and also Dr. Kambang Sariaji. So first question is about for Dr. Yuliawati. So I saw in your slide, it's about inducer concentrations. So I saw at concentrations 5 something, the band on your peptide or something, and on your band, it shows tighter compared to the higher concentrations. Is it the problem of putting the sample, or 5 is the best conditions? And for the second questions for Bapak Kambang, it's just out of my curiosity. So when you take a sample from the air, do you have kind of basic rule or just like standard rule to get a sample? Because the air, you have many spaces, you need to standardize your sampling method. Thank you very much.
Okay. Can I answer the question from the Ibu Penny?
Okay.
Okay.
Sure.
Actually, we have the method how to collecting the air. Of course, we have the referral from the WHO, and also some referral from the journal, and I think that's you're right, because the air is very wide, very very wide. So hard to taking all of the air in the room. But we have the justification for the collecting of the air. We're taking for volume the air, 10% from the all of the weight of the room. 10%. For the example, if the room is with the 40 meter cubic, and then we're taking the 10% from the area. This the point actually. Thank you.
Did you use vacuum or something to catch the air?
No, we using we use the instrument like the air sampler. Yeah.
Oh, I see. I get it.
Yeah. Thank you.
Thank you very much.
Okay, the question for Ibu Yuliawati. Ibu Yuliawati, will you answer the question from Dr. Penny?
Pardon? You asked about the concentration
inducer concentrations.
Oh, inducer concentration.
Yes.
We use we analyze the band by image analyze. So we membandingkan the band by the image analyze.
Jadi Ibu pakai foto di gel kan, gel concentration, various concentration with 2, 5 or something, and I show on your gel at 5 concentrations the band is very tighter compared to the other concentrations. Is it 5 is the best conditions, or because basically when higher concentration the band should be tighter if I'm not mistaken, but in this case in a 5 concentration the band is tight. So I was wondering, is it due to putting the sample to the gel, or the 5 is the best conditions?
I answer in bahasa. Kami di sini memproduksi protein dengan beberapa induser Lamnos dari 0 sampai 2 milimolar. Nah, setelah itu dari 5 kondisi kalau nggak salah itu, lalu proteinnya kita isolasi. Nah, lalu dianalisis dengan SDS page. Nah, hasil dari band di SDS page itu kemudian dianalisis dengan image. Seperti itu.
Oh ya, baik Ibu, terima kasih.
Okay, thank you very much. Very nice discussion. So I have one question to Dr. Desriani. You mentioned in the conclusion that there is a challenge to get more sample of breast cancer patients. Do you have any strategy for the future research to overcome this challenge?
Thank you, Dr. Hani, for the questions. Yeah, I think more sample, the result will be more reliable, I think. And maybe in the future, in order to get the more acceptable data, I think. So we need more sample to add more sample, and also more clinical pathological parameter. In order to get more comprehensive about the relation between the PTEN promoter methylation with the clinical pathological, especially here with the breast cancer patient in West Sumatra. Thank you.
Thank you very much. Is there any question? 1, 2, 3? No? Okay. I would like to thank to all the presenter and all the participants in this very nice session. So thank you so much. I do hope everyone get beneficial from the knowledge sharing from all the speakers in this afternoon. Thank you very much for all your kindly attention, and stay happy and healthy. And I return this session to the organizers.
Thank you very much, Dr. Hani, for moderating the sessions. It's really great discussions. Bapak Ibu, thank you very much for staying us until right now, but we need to come to the main room because we need to hear some informations and some announcements from MC, like a best presenter and something. So please be patient, wait for another room to be done, and please wait in the main room. And see you and take care. Bye.
See you. Bye. Thank you so much.
Recording in progress.
Welcome back, ladies and gentlemen, to ISIBIO 2021. So we're waiting for others participants to join us in the main room. And after that, we will announce the best presenter of ISIBIO 2021. So please keep waiting. Thank you. Ladies and gentlemen, welcome back to the main room of ISIBIO 2021. We would like to thank all the speakers and moderators for very insightful session. Okay, so for the next, we will announce the best presenter of ISIBIO 2021. And the best presenter are chosen based on the highest score of the presenter in each room during day 1 and day 2 of parallel sessions. Now we would like to announce for the first day, the best presenters of the first day of ISIBIO 2021 are Room 1, Nikadek Erosi Undaharta. I'm sorry for the wrong spelling. And Room 2, congratulations. Room 2 is Mr. Aryo Beta Juan Silvero. Congratulations. And Room 3, the winner is Fitria Dwi Ayuning Bias. Congratulations for the winners of the best presenter of the first day of ISIBIO. Now, move on to the second day. The best presenter of the second day of ISIBIO 2021 are Room 1, Mrs. Wayuni. Congratulations. Room 2, La Ode MM Davis. Congratulations. And last but not least, Room 3, Andri Wardiana. Congratulations once again. Let us give a round of applause to the winners, and you can use the reaction feature in the Zoom to congratulate the winners. Once again, we'll say congratulations to the best presenter for delivering such remarkable presentation in ISIBIO 2021. Ladies and gentlemen, we will have closing remarks by Dr. Mastur, Chair of Indonesian Biotechnology Consortium. Please welcome Dr. Mastur.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. Distinguished invited participants, organizers, ladies and gentlemen. First, let us express our praise and gratitude to Allah the Almighty for His grace and guidance that today we can successfully finish the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering. On behalf of everyone involved in this conference, I will also express our gratitude to the Chairman of National Research and Innovation Agency, Dr. Laksana Trihandoko, for his support, direction, and opening remark. His support motivates and boosts our energy to enhance research and application of biotechnology, to support national development in all regard sector and field. Biotechnology at the front line of science and technology is required to increase productivity, quality, efficiency on biodiversity, sustainable environment management and use, medical and industrial sectors, product diversification, and new innovation. Biotechnology is also expected to overcome the climate change through mitigation and adaptation to bring great benefit to human life and environment. During these 2 days, we had a fruitful discussion and sharing information. Thank you so much for all resource persons for your excellent ideas and sharing information. For all participants, thank you so much for your support in scientific papers. We highly appreciate all sponsor and support for this international seminar, especially for BRINS and Biotechnology Enterprises Partners. KBI management and all members believe everyone gain a huge benefits from this program. Therefore, KBI management will commit to promote biotechnology through the similar program, especially during period 2021-2024. We will improve collaboration with other stakeholders, universities, research institute and bureaucrats, and industry and other biotechnology practitioners. Synergy to support research, education, and industrial practices in Indonesia. At this opportunity, we will also invite all elements, universities, research institute, government institution, industry, and business related to biotechnology to join KBI. KBI management commits to bring real benefits to all members. KBI will facilitate research, engineer, university lecturers, and students to improve their research and publication. For bureaucrats, KBI will participate in promoting regulation. For industry and business sector, you will get opportunity to advertise your product. Finally, we hope collaboration between KBI members and all stakeholders will be strengthened and intensified. Lastly, thank you Research Center for Biotechnology BRIN for great collaboration. For all participants, on behalf of the conference organizer, please accept our sincere apologies for inadequate services or any shortage during the conference. We hope this conference will bring advantage for everyone who join for our organizations and national development. Praise to God, Alhamdulillah, I close this conference. Thank you and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Mastur, for officially closing the 8th International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering. We would like to give gratitude to Chairman of Research Organization for Life Sciences and Chair of Indonesian Biotechnology Consortium, all the keynote speakers, invited speakers, moderators, and presenters of the International Symposium, and Not to forget, the participant, thank you for the participation in ISIBIO 2021. Don't forget to fill out the list of attendees, and the certificate will be sent by the committee through email maximum 15 working days. Tomorrow, we will also hold the 7th Indonesian Biotechnology Conference, or IBC 2021. There would be talk shows with many great speakers, so we hope you can join the event tomorrow. And on the behalf of the official committee, I would like to apologize for any inconvenience matters during the event. So see you on ISIBIO next year. Have a good day. Goodbye.
Oke, thank you Mbak Wiwit. Thank you semuanya.
Thank you very much.
Thank you.
Thank you for all.
Ibu.
See you in next year.
Thank you semua panitia, seluruh komite. Live ya.
Iya, Bu.
Asep, Bayu, makasih ya.
Makasih. Selamat
panitia. Panitia
Siap, Bu.
semua.
Bayu, terima kasih banyak.
Sukses
Ayo,
ya.
sukses semuanya. Terima
Sukses.
kasih.